Hi, Namaste sir, good morning. Ashok sir, good morning. Rakesh sir, good morning. Can you hear and am I audible? Sir, I think your mic, wait, one sec. Can someone from my team open the mics for everyone? Oh yeah, now, yes, now
sir.
can speak.
Yeah, good morning.
Yeah.
Good morning.
Good morning, sir, good morning.
Namaskar and good morning to everyone.
Okay, Dr. Ashok, sir is here. Ashok sir.
Yeah, sir, good morning. Good morning. Can you hear me?
Your video.
Good morning.
Yes, good morning, sir. Good morning. Very good morning. Yes, now I can see. Yeah. Okay, so good morning, everyone, and I welcome you all to our second medical writing, clinical research, and pharmacovigilance conference. So we have done with the first day yesterday, and it was the amazing day and very informative, very fruitful day where we had with us our guest of honor, Dr. Ranjit Barsikar sir. We had Dr. Pramod Kumar Rajput. We had Mr. Hardik, and we had Dr. Hit Sarma from Serum Institute of India. We had Dr. Siwari Desai. We had Mr. Apoor Soni from JSS Medical Research. We had Dr. Kanav Khera, Dr. Sameer Dhingra, Registrar, Naipar Hajipur, and we had our multiple panelists, Dr. Jamal Beg from Sanofi. He's a director of pharmacovigilance there. We had Dr. Anupama Koneru, principal and professor at Sultan Uloom College of Pharmacy. We had Dr. Kiranbhai Mandava. She is serving as principal at St. Paul's. We had Dr. Hamantachar Joshi sir. He's principal and professor at Sarda Vilas, and we had Mr. Abjit ji, and then we had Dr. Isaac sir. He is professor and director for the Center of Career Planning and Counseling at University of Kashmir. We had Dr. P. Rajsekhar. He's the principal at St. Peter's Institute of Pharmacy, and we had Mr. Ashish Kumar Verma. We had Mr. Ravi Jogai. He's associate director at Syneos Health. And we had Dr. Mansa also. She is professor and HOD at East West College. And we had Dr. Arun Kumar Mishra. He's the professor and head of pharmacy and dean of student welfare, IFTM University, Muradabad. So let's start for today, guys. We are already 10 minutes delayed, but as Sunday morning, people are a little lazy to join. So we were just waiting. So today's our first topic is very amazing. And today's first topic is new drug and clinical trial rules 2019. And to take this with us, we have Dr. Ashok Shenoy. He's the professor and professor of pharmacology at Kasturba Medical College, Bangalore. Mahe, and we have Dr. Rakesh Somani sir. He's the principal and professor at Dr. D.Y. Patil University School of Pharmacy, Mumbai, Maharashtra, to chair this session. So let me take this privilege to introduce Ashok sir. Give me a second, and then our host will take over. So let me take this privilege to introduce our sir here. Okay, so as you can see, our Ashok sir, he's currently serving as the professor at Department of Pharmacology at Kasturba Medical College, Bangalore, Mahe. Sir is an honorable member of Mahe Ethics Committee as well, and he is experienced professor with a demonstrated history of working in education and clinical research, participated in over 80 clinical trials in 2 decades. He has, studied 50 bioequivalence studies from 2000 in 2007-8. He is the examiner for UG and PG courses for over 20 universities in India, 1 university in Nepal and 1 in Mauritius. He has published around 60 plus publications in high impact journals and 3 book chapters. He has been a trainer for GCP and regulatory guidelines in clinical research and delivered more than 100 sessions. So I don't think that there is someone who is, best to deliver this session except Dr. Ashok Shenoy sir. So I will hand over this mic to Dr. Ashok Shenoy to deliver this lecture. Thank you, sir. Thank you very much. Over to you.
Yeah, thank you. Thank you, Dr. Ajit, so much. I'll share the screen.
Please go ahead, sir.
Visible? The screen is visible?
Yes, sir. It is visible.
Okay. Yeah, I'll just cut off video to save on bandwidth.
Sure.
Good morning, dear participants. At the outset, I would like to thank the organizers of this event for giving me an opportunity to interact with you. If you are interested in participating in clinical trials, you must be trained with New Drugs and Clinical Trial Rules 2019. If you are an ethics committee member, then your registration will not be accepted unless in the training certificate it is mentioned clearly that you have been trained in New Drugs and Clinical Trial Rules 2019. So needless to say, this training is must and mandatory for those who want to pursue their career in clinical research. So I'll give you the salient features of NDCT 2019, and let us see what is important from investigators' point of view and ethics committee point of view, because I believe the audience here is mostly the investigators or the prospective investigators and prospective ethics committee members. Hence, let me concentrate on their roles and responsibility with respect to New Drugs and Clinical Trial Rules 2019. Disclaimer. So these rules came into force in 2019, and they are called New Drugs and Clinical Trials Rules 2019. Please note the nomenclature. There are plural in some places, singular in some places. Whenever you mention the title of the rules, you should mention it very clearly as it is. Once again, I repeat, New Drugs, plural, and Clinical Trials, plural, Rules, plural, 2019. It has 264 pages. It is gazetted, and you know the Indian Gazette is a PDF document. And if you get fed up by scrolling, the first 146 pages are in Hindi. From 147th page, the English version starts. So you can go to English version directly by clicking on page number 147. There are 13 chapters, 107 rules, 8 schedules. There are 27 forms in the 8th schedule, and it is gazetted and published on 19th March 2019, both in Hindi and English. Please note, the New Drugs and Clinical Trials Rules 2019 is applicable only for new drugs and investigational new drugs for human use. The veterinary use regulations continue to be a part of the Part 10A of the Drugs and Cosmetic Rules 1945. There is no amendment to that. With this, the Schedule Y gets obsolete. The Schedule Y is no more applicable. In other words, Schedule Y is replaced with New Drugs and Clinical Trial Rules 2019. I would like to define few terminologies so that you get more clarity on that. Orphan drug is a drug intended to treat a condition which affects not more than 5 lakh persons in India. We have always heard about orphan drugs and orphan conditions, orphan diseases, but the quantification of this was not done previously. So these rules mention very clearly that if a disease affects not more than 5 lakh persons in India, the disease is called orphan disease, and the drugs used in those treatment are called orphan drugs. What is an interventional study? Now, those who are ethics committee members, it is, they have to differentiate between interventional study and observational study. So what is an interventional study? That is defined in these rules once again. A type of clinical study which in which the participants are assigned to groups that receive one or more intervention or treatment so that researchers can evaluate the effects of the interventions on biomedical or health-related outcomes. So assignments are determined by study protocol. The investigator is going to assign an intervention or a study treatment. And the participants may receive diagnostic, therapeutic, or other types of intervention. So you are going to intervene. I mean, you're going to provide an intervention. It is not like as a part of routine clinical care, some intervention is done and you are sitting and observing and you are recording the findings. That is an observational study. Here, clinical trials are interventional studies. RCTs, as they're called, randomized clinical trials, are always interventional studies. These rules clearly define the purposes of ethics committee. The purposes of ethics committee has been dichotomized very clearly. Ethics committee to oversee clinical trials and bioavailability, bioequivalence studies, and ethics committee to oversee biomedical and health research. So you can call that EC as EC for CT, in other words, ethics committee for clinical trials, and EC for biomedical and health research. What comes under clinical trials? Any sponsored interventional study, the data of which is going to be sent or submitted to any regulatory agencies in the world, maybe FDA, DCGI, or any regulatory agencies, then it comes under clinical trials. Biomedical and health research comprises of any other research that is done from academic point of view. Most of us are working in a medical college or a pharmacy college, and many of you will be working with in the hospitals and involve yourself in human research. In such situations, maybe an undergraduate research or a PG dissertation or maybe a PhD thesis or research for the purpose of promotion from assistant to associate, associate to professor. You know, the NMC and these councils have made it very mandatory to have publications for various promotion purposes. So a faculty in a medical or a pharmacy college would like to do research and to build his own credentials, to build his CV, he would like to have publications. All that research comes under biomedical and health research. Now, a question might arise in your mind whether you should have two ethics committees in an institution, one for clinical trials and one for biomedical and health research. The answer is both yes or no. Yes, because if the workload is very high, if it is a well-established college with a heavy workload, probably the ethics committee may not be able to handle both. In such situations, it is better to have two ethics committees, one for clinical trial and one for biomedical and health research. Whereas a new college, upcoming college, where the workload is not that high, probably single ethics committee can handle both type of research. However, that single ethics committee should be registered under both these bodies. Ethics committee for clinical trials should be registered under rule number 8, which says it should be registered under DCGI, that is CDSCO, Central Drug Standard Control Organization. And ethics committee for biomedical and health research should be registered under rule number 16, which says, rule number 17, which says it should be registered under Department of Health Research or DHR. So these have been very clear. So if at all you are an ethics committee member, or especially if you are member secretary, please make sure your ethics committee is registered under DCGI. If you are overseeing clinical trials, please make sure that your ethics committee is registered under DHR if you are overseeing biomedical and health research. I hope this is clear. With this, let us move on to further clarify the difference between ethics committee for clinical trials and ethics committee for biomedical and health research. Ethics committee for clinical trial should be registered with the CDSCO, that is DCGI, and ethics committee for biomedical and health research should be registered with the DHR. Validity of registration is 5 years for ethics committee for CT, and initially for 2 years and subsequently for 5 years for biomedical and health research. It is relatively new and they're just setting it up. That is why they give provisionally for 2 years, and once registered and 2 years completed, subsequently for re-registration, it will be for 5 years. There is a rule in the NDCT that if the ethics committee is not in the same city as the site, the distance between the two should not be more than 50 kilometers. Whereas for biomedical and health research, there is no such specifications. And ethics committee for clinical trials functions as per the Indian GCP. It is not yet revised. I know it is quite old. It is 2001, more than 2 decades over, but still we are supposed to follow Indian GCP and as per NDCT 2019. Ethics Committee for Biomedical and Health Research follows or works as per ICMR guidelines. SA reporting has to be done by IEC for CT to DCGI because it oversees a regulatory trial. No such reporting with respect to biomedical and health research. Please note, a single ethics committee can oversee both clinical trials and biomedical and health research, provided it is registered as per the requirements, that is, both CDSCO as well as DHR. There are 3 schedules with respect to ethics committee functioning. 3rd schedule, which mentions about conduct of clinical trials. 7th schedule, which says the formula to determine the quantum of compensation in cases of research-related injury or death. And 8 schedules, which gives the various forms of application and issuance of permissions. Please download this document and go through this. I'll just give you an overview of what it contains, what are the contents. Throughout the document, it keeps mentioning Central Licensing Authority. Please note, Drugs Controller General of India, DCGI, appointed by the central government in the Ministry of Health and Family Welfare, is the CLA for us, Central Licensing Authority, and he is the final authority for approving the conduct of clinical trials in India. First of all, the title of the document is New Drugs and Clinical Trial Rules. So we should know what is a new drug. There are 6 clauses to define a new drug, and it is very clear. This is a regulatory document. It is a rule. That means if you do not comply with it, you are liable for punishment, penalty. Hence, each wording becomes important. And if I change, then probably the interpretation or the meaning might change. That is why I have retained as such, but they are quite long. So whatever is written in blue, please concentrate on that. What is important, I have highlighted in blue. So a new drug is defined as a drug that has not been used in the country to a significant extent. It may be there in the market for decades together, but if it is not used for a significant extent, then it is considered a new drug. A drug approved by the DCGI for certain claims and proposed to be marketed with modified or new claims, including indication, route of administration, dosage, and dosage form, is considered a new drug. I always take an example of paracetamol to explain this concept. Paracetamol is available as tablets and injectable. It is available as 500 mg and 650 mg tablets. It is available for oral route, intravenous route, intramuscular route. Now you want to check efficacy of 750 mg of paracetamol. That is a change in the dosage. It becomes a new drug. You cannot say paracetamol is such extensively used drug and I'm just changing from 650 to 750. I usually give 500 plus half tablet 750. So I want to make it a single tablet rather than 1.5. So, it can be done just like that. No, it becomes a new drug because there is a change in the dose. Now you want to change in the route of administration. You want to try subcutaneous. Then even though it is given by oral route, intramuscular, intravenous, and even rectal route, still if you want to try subcutaneous route, it becomes a new drug, even though the drug is there in the market for such a long time. New indication. We have seen this during COVID time. All the repurposing of the drugs, all it comes under new indication. COVID was a different indication, emergency use authorization. It comes under a different umbrella. Otherwise, if you feel, for example, let us consider 2 drugs with the same pathogenetic mechanisms. For example, I can give ulcerative colitis and rheumatoid arthritis. Both are autoimmune diseases. And if you go by the pathogenesis, a drug used in ulcerative colitis can be used in rheumatoid arthritis. There are many drugs which are common in both these conditions. Let us say a drug used in rheumatoid arthritis, approved in rheumatoid arthritis, is in the market. Now, I'm not saying if you use this drug in ulcerative colitis, it is unethical because it is available in the market and the pathogenesis is same. So this is called as off-label use of the drug, but this is not correct. It has to be approved in ulcerative colitis if you want to treat a patient. But if the patient is not benefited by other drugs and you want to use, of course, it is off-label use. If you want to label it and if you want to do a clinical trial of that particular drug in ulcerative colitis, you cannot argue saying that it is available in the market, it is used in rheumatoid arthritis, pathogenesis is same, so I want to use in my patients. So this is called a newer indication or repurposing of the drug. All these comes under new drug. A fixed dose combination of 2 or more drugs approved separately for certain claims and proposed to be combined for the first time in a fixed ratio. Drug A available in the market, drug B available in the market. You are not changing the dose, you're not changing the route of administration, you're just combining them in a fixed dose combination. Still, it becomes a new drug. A modified or sustained release form of a drug or a novel drug delivery system, vaccine, recombinant DNA product, living modified organism, monoclonal antibody, stem cell derived product, gene therapeutic product. Look at these two clauses. These two clauses, the drugs are new forever. The drug belonging to these classes are new drugs forever. Why? After a drug is released into the market, after the marketing authorization holder gets the permission to market, PSURs have to be submitted initially every 6 months for first 2 years, then annually for the next 2 years. That means 6 PSURs have to go. After 4 years, it is no more a new drug, then these PSURs need not be submitted. But if a drug belongs to this category, then it is a new drug forever. That means the PSURs have to keep going. So this is one way to improve the quality of the medication, and it is a welcome move actually. So otherwise, if you see the chronology, the periodicity, a drug is considered new for a period of 4 years from the date of the permission. So these are the 6 clauses of new drug. Next is serious adverse event. I want you to appreciate the difference between serious adverse event and the severity. Severity goes as per mild, moderate, and severe. Let us say I got involved in a clinical trial. My hemoglobin started dropping. Initially, it was 10. After getting involved in clinical trial, it became 9, mild anemia. It became 7, moderate anemia. And it became, let us say, 5, severe anemia. This is mild, moderate, and severe. After taking a particular drug, I developed fever. My temperature is 99, mild fever. My temperature is 101, probably moderate fever, and 104, severe fever. So this is grading of the severity, mild, moderate, and severe. Serious adverse event is something different. It is not equivalent to severity. Severity and seriousness are different entities. Seriousness goes by definition. Any untoward medical occurrence during the clinical trial resulting in death, hospitalization of the trial subject where the trial subject is outpatient, or prolongation of the hospital where the trial was inpatient. Persistent or significant disability or incapacity. Don't get confused with this disability or incapacity. Let us say I got involved in a clinical trial. I developed headache. I developed so much of headache that I didn't get up the whole day. I missed my breakfast. I missed my lunch. I missed my evening tea. Night, I could get up and have some meals a little bit. And that night, somehow I could get some sleep. This is not disability. Yes, my whole day is gone, but this is not disability. Disability is something like loss of a vital function, loss of eyesight, loss of limbs, loss of hearing sensation. This is disability. congenital anomaly or birth defect. Please note, pregnancy is not a serious adverse event. So during clinical trial, if the lady becomes pregnant, it is not considered serious adverse event. However, it is a event of special interest. That lady is followed up, and the child after born is followed up for any congenital anomaly. And if the child is born with congenital anomaly, then that is considered serious adverse event. This is a prolonged process. Probably by now you will realize why these companies stress so much on contraception during clinical trials. Or life-threatening event. Please once again have clarity on life-threatening event. Life-threatening event is not something like, to give an example, let us say the potassium is 7. As per textbook, if it is more than 6, then serum potassium levels, the patient can have an arrhythmia and it can be fatal. Let us say the potassium is 7 and the patient is fine. I mean, he is into your clinic and he looks fine. Now, if you assume if the potassium was 1 unit more, or if it was some 7.5, or if it was 8, he would have been dead. That is not life-threatening event. So let us say blood sugar of the patient is 50, and he is having some sort of uneasiness and giddiness, and he is uncomfortable. Now you cannot say if it had gone to 40 or 30 or something like that, he would have had permanent brain damage and the consequences of hypoglycemia. So, or he would have been dead. So that is not life-threatening. To call life-threatening, his life should be at threat at that point of time. With the same example, let us say serum potassium. Serum potassium was 7. He had arrhythmias. Intervention had to be done and he was saved. That is life-threatening. So whenever you decide on seriousness, this has to be considered very clearly and legitimately. Because if a patient suffers from a serious adverse event abbreviated as SAE, then compensation and it's a matter of money and finances. So you should be very clear. You should have a lot of clarity before branding something as serious adverse event and the causality assessment thereafter. Central Drug Standard Control Organization is a regulatory body. It's the arm of the Ministry of Health and Family Welfare, Government of India. And our counterparts are US FDA in the US, Health Canada in Canada, Therapeutic Goods Administration in Australia, and European Medicines Agency in the European Union. Till recently, it was called as EMEA, but since 2018, I think, it is called as EMA. Abbreviation is EMA only, EMA. So what is the relationship between DCGI and the CDSCO? DCGI is an official of the CDSCO, and he is a final regulatory authority for approval of clinical trials in India. Now let us move on to the new Drugs and Clinical Trial Rules 2019. The chapters and the rules are continuous. The chapters come in between. Rules are from 1 to 107. They are very continuous. It is nothing like chapter number 1, rule number 1, chapter number 2, rule number 1. It is not like that. For example, chapter number 1 has got 2 rules. Chapter number 2 has got 3 more rules. So 5 over. Then chapter number 3, the rule number 6 onwards starts. And chapter number 3 deals with ethics committee for clinical trial, bioavailability and bioequivalence studies. So let me stress a bit on this. Rule number 6 says, whoever intends to conduct clinical trial or BAB study shall be required to have approval of the ethics committee for clinical trial registered under rule number 8. And that rule number 8 says ethics committee should be registered under DCGI. So if you are an investigator and if you come with the clinical trial, sponsored clinical trial, regulatory clinical trial, and if you are planning to seek approval of the ethics committee, you should find out whether the particular ethics committee is registered or not. If you go to the CDSCO website, you will have the list of ethics committee registered and re-registered, because it's already many ethics committees that have undergone re-registration also. So find out whether your ethics committee is registered or not. Then only if it is registered, you submit your protocol. If an ethics committee is not registered, then they are not supposed to function. They should cease functioning and seek registration immediately. Rule number 7 says there should be a minimum of 7 members from medical, non-medical, scientific and non-scientific areas, at least one layperson, one woman member, one legal expert, and one independent member from any of these fields: social scientist, representative of NGO, philosopher, ethicist, theologian. Okay, all these it's similar with ICMR guidelines. If you have already read or known ICMR guidelines, it is somewhat the same. Rule number 7 also mentions that ethics committee shall consist of at least 50% of the members who are not affiliated with the institute or organization. Because of this, in 2019, ethics committee saw a revolutionary reconstitution because internals are more, externals are less for various reasons. That is why every ethics committee, almost majority of the ethics committees, they saw a re-registration wherein 50% of the members were outsiders. Chairperson is an outsiders because he should not be under the influence of any of the higher ups in the institution. Member secretary has to be internals because outsiders cannot come and run the show. Only the internal people will know how the institution runs and whereabouts and the working. Outsiders cannot come and run the show. So member secretary has to be internal. Every member of the ethics committee is required to undergo training and developmental programs as may be specified by the DCGI from time to time. Maybe GCP training or a training like this, wherein you're trained in NDCT 2019. This is to safeguard the right, safety, and well-being of trial subjects. In the previous regulations, it was not clear if you don't attend the training program, or if you're not willing to attend training program, what is the consequence? But here, these rules state very clearly that if you are not willing to get trained, you are disqualified to hold the post of the member of the ethics committee. That means you should quit ethics committee. Rule number 7 also mentions that the medical scientists or the clinicians have at least PG qualification. That means you should be a MD or MDS to be PI in a clinical trial. MBBS or BDS graduate cannot become the PI for any clinical trial. And he should have adequate experience in the respective fields and the requisite knowledge. Just like you cannot become assistant professor directly in medical college, you should join as a tutor or an SR. Then after 1 year, assistant professor. Then after 4 years, associate professor. Then after 3 years, professor. But here, that timeline is not very strict. The sponsor decides based on the experience and based on your trainings whether you can be the PI. Otherwise, you join as a co-investigator, gain adequate experience, and become PI later. Specific patient groups may be also included in the ethics committee. For example, if there is an HIV trial, then HIV patients, if they are willing, if they are fine with it, they can be called as a patient group representative. And there is no conflict of interest that has to be declared. I mean, every ethics committee member has to declare that he does not have conflict of interest. Please note, having conflict of interest is not bad, but not declaring it is unethical. That is violation of GCP. So this has to be declared in writing to the chairperson. There should be a form to declare conflict of interest, yes or no. Then you have to sign there. And if you have a conflict of interest, do not participate in the deliberation. Ideally, you're supposed to get up. You should go out of the room. You should not even hear the deliberations because your mere presence may interfere with the decision. And this has to be recorded in the minutes of the meeting that Dr. So-and-so left the room. He didn't participate in the deliberations. These are some rules which talk about the functioning of the ethics committee. So these are the same as ICMR rules. I think I'll skip this for the time being. Quorum is same as ICMR. For quorum, there should be 5 people: medical scientist, clinician, legal expert, social scientist, or representative of NGO, or ethicist, theologian, or a similar person, and a lay person. If these people are absent, if one of them is absent, let us say a legal expert, let us say he got a call from the judge last moment and 1 hour before he says, I'm sorry, I cannot attend the meeting. All others are on the way and all others come. Legal expert is not there. What will happen to the ethics committee meeting? Ethics committee meeting gets nullified. You cannot have the meeting because without this quorum, the sponsor will not accept that approval letter. What is the solution? If this happens in a place like Mangalore or some small cities, Mysore, etc., I mean, that is fine, Manipal, etc. That is fine. I mean, of course, uh, people can come again because it doesn't take a very long time to travel. But if it's a metro city, something like Mumbai or Bengaluru, and if your college is or the ethics committee meeting is around 1.5, 2 hours away, one way, you spend so much of time come there and just to have some remuneration and cup of tea or something like that, that is not the purpose for which you have applied leave and wasted that much of time. Hence, the institutions, they keep one more backup, what are called as alternate members. So institution maintains a list of alternate medical scientist, alternate clinician, alternate legal expert, alternate layperson, or these alternate people. If the main person, the main member cannot come, then these alternate people are called for to attend the meeting. So this is allowed as per NDCT 2019. You can have subcommittees, maybe SAE subcommittees or various subcommittees if the workload is high. And please note, subject experts have no voting rights. Any change in the membership has to be intimated to the DCGI within 30 working days. Records have to be maintained for 5 years after the completion of the trial. And this is stricter compared to document maintenance in other parts of the world. That time in 2019, they said Ethics Committee for Biomedical and Health Research has to be registered under a body which we will notify, and duly in September. They said within 6 months we will notify, and they did that. In September, it was notified, and DHR is the authority for that. So this is an information to be submitted by an applicant for grant of registration of Ethics Committee. This is for Ethics Committee member secretary. He has to compile all this data. So not an easy job, very demanding job, Ethics Committee member secretary. And this is the format of approval of clinical trial protocol by Ethics Committee. Many a times I see Ethics Committee approval letters. Dear Dr. So-and-so, the documents submitted by you are examined by the Ethics Committee and they are approved. Then thanking you, member secretary. No, this is not the way to approve a clinical trial or send approval letters. This is the format. You have to say very clearly which documents were reviewed. You should say the name of the ethics committee, where the meeting was done, when the meeting was done, time also if possible, which documents were reviewed, because the company may not submit all the documents. They may submit maybe English informed consent form initially and Kannada informed consent form and regional informed consent forms. For example, in Bangalore, we may need Malayalam. In Bengaluru, you may require Tamil. And maybe in the northern part of Karnataka, you may require Marathi informed consent forms. So that may not be ready. That doesn't mean that you wait till those informed consent forms are available. So English and maybe Kannada is ready, but the other regionals, if they're not ready, that doesn't mean that the approval should be withheld. Hence, you write what documents were approved and list them out. And the following members of the ethics committee were present at the meeting held on. So you should mention all the ethics committee members who are present, ethics committee members who are excused. Then say, we approve the trial to be conducted in its presented form. Then if at all you have some clause like submit the progress of the report once in a year or a SAE report or whatever you want to say, okay, you write that. Then you are sincerely member secretary. Even if you type this in Times New Roman font 12 with single spacing also, minimum 3 pages it will occupy. So it has to be as per this format. There is no shortcuts and abbreviations. The NDCT rules, they go on and on. This is chapter number 5. 28, the rule number 28 is very dear to me, academic clinical trial. This is applicable for all of us, majority of us rather, because we work in an academic institution. No person or institution or organization shall conduct clinical trial of a new drug or investigational new drug without permission from DCGI and Ethics Committee. So both permissions are required. Many ethics committees say that without DCGI permission, don't approach us. This is not necessary. Please note, Ethics Committee is an independent body. Ethics Committee, it does not require any other approvals. It can approve any trial for that matter. But however, the trial cannot be initiated without the DCGI permission. At the same time, DCGI also requires certain ethics committee approval letters. So this is something like a chicken and egg situation. So dear ethics committee members, please note you are independent. You don't need any other's approval to approve any clinical trial protocol that has come to you. You are autonomous, you are independent. Every clinical trial site shall be overseen by an ethics committee. DCGI has to take the decision within 90 working days. Now, there is an amendment to this, which I'll tell you briefly at the end. If rejected, then the sponsor has a clause to appeal in front of central government. They need not keep quiet, and if the sponsor feels that probably they had certain things to clarify, they can go to central government, and timelines are set for everything. If the drug is discovered in India or R&D of the drugs are done in India or proposed to be manufactured or marketed in India, then probably as a part of Make in India program, the decision is taken in a fast track technique within 30 working days. However, if the drug is already approved outside India, the same 90 days apply. DCGI puts a lot of conditions for permission. The DCGI and Ethics Committee permission is required. Registration with the CTRI is mandatory. Dear students, there is a lot of discussion with respect to whether CTRI registration is mandatory or not. My advice to you is please register all your studies, because what you're going to lose by registering. It is a new learning experience, and please note, it is not an approval. It is a registering body, that's all. You have to register. They will allot you a number of registration, and this may be useful when it's the time to publish, because many journals are asking for CTRI registration number now. So the dictum is, whenever you are in the doubt, register. Of course, observational studies need not be registered. It is not mandatory. Clinical trials, it is mandatory, and you have to register prospectively. Retrospective registration has been cancelled. For you, all your academic work, whether observational or interventional, whatever you do, register, because you're not going to lose anything by registration. Follow protocol and GCP. Status of enrollment to the DCGI at least quarterly, 6 monthly report to the DCGI in the SUGAM portal. And if the trial is terminated, DCGI has to be informed within 30 days. Validity of clinical trials is for 2 years, and it is extendable because hardly clinical trials get over in 2 years. Post-trial access is something where, for the first time, a big country like India is vocal about it. Otherwise, even the big boss, the US FDA, is not very vocal, not very explicit about the post-trial access. A few countries, small countries, have been quite clear about post-trial access. So what is post-trial access? Once the trial is over, making an investigational new drug available to the trial subject, through which the said drug has been found beneficial to a trial subject, and for a period as considered necessary by investigator and ethics committee. The role of ethics committee is of paramount importance here. Let us say same ulcerative colitis. Some drug, 1 year is the trial period, found beneficial. The patient really had a good time. He didn't have pain abdomen. He didn't bleed. He had a good quality of life. The trial is over 1 year, and the investigator says, trial is over. Now go back to your whatever drugs you want, or I'll put you on the same old azathioprine, steroids, etc. If he develops a relapse, now he can blame the investigator. You gave me a good quality of life for 1 year. That drug was very good. I was found it was beneficial for me. Now you are taking off the trial. You're pushing me back to the same old status. So now, if that investigational drug has to be given by the patient, given to the patient, it is an unapproved drug. So it cannot be given just like that. At the same time, if it is beneficial for the patient, if the patient is demanding it, then it can be given from humanitarian and ethical point of view. So here the DCGI, I mean these rules put some clause. If there is no alternative therapy exists and it is found beneficial to the patient with the legal heir's consent and declaration in writing that the sponsor shall have no liability for post-trial use of investigational new drug. That means
So,
if the
sir,
patient dies.
sorry to interrupt. We are exceeding
Yeah.
the time.
Oh,
5
okay.
Yeah.
Okay. Yeah.
Thank
Yeah. So if uh, if the patient dies during this post-trial access, then there is no compensation that can be given. If that is okay with the patient, then it can be given. Post-trial access can be given. Chapter number 6 deals with the compensation. So compensation amount is clearly stated. It is the quantification is done. ICMR guidelines, they say that the compensation has to be given, but it doesn't say how much it has to be given. These rules say very clearly what is the quantum of compensation that has to be given. Chapter number 7 deals with bioavailability and bioequivalence study center. You can go through this. Chapter number 8 onwards, it is something like manufacture new drugs, import, export, etc. Chapter number 9 deals about import of new drugs. 10 also import or manufacture of new drugs for sales and distribution. Chapter number 11 also the same. Chapter number 12 amendments to drugs and cosmetic rules. Miscellaneous 13. First, there are 8 schedules. I told you the first schedules, general principles and practice for clinical trials. Second schedule, requirements and guidelines for permission to import or manufacturing of new drugs for sale or to undertake clinical trial. Third schedule deals about conduct of clinical trials. Nicely, it is given there how to conduct a clinical trial. Please go through this. Fourth schedule talks about the requirements and guidelines for conducting BAB studies. Fifth schedule talks about post-market assessment. Sixth talks about fees payable for license, permission, and registration certificate. Something to be noted here, all the government institution, the fees is 0. If it's a private, it goes something like 1 lakh, 2 lakhs, 5 lakhs. Seventh schedule talks about the formula to determine the quantum of compensation in cases of clinical trial related injury or death. This is important if you come across a CE. And 8th schedule talks about application of registration renewal of ethics committees. Yeah, these are some amendments that have been done. Small, last 2-3 slides. So the central licensing authority has been designated as registration authority because for non-regulatory trials, this is not a regulation. And it says analytical part for BABA studies. Previously, it was only clinical, it was mentioned. Small changes, stem cell derived product, it is replaced with cell or stem cell derived product. This was in January 2022. October 2022 had a major amendment. I'll summarize in a minute. I told you 90 days is the time with the DCGI for saying whether the clinical trial is approved or not. Now, amendment in October, it says clearly that if there is no communication that is received from the CLA within the said period of 45 working days, this is for registration of ethics committees. And if you don't hear anything within 45 working days, then it is considered registered. So you don't have to wait and you have to submit CT02 form. And for the grant of permission to conduct clinical trial, if you don't hear anything from the DCGI within the 90 working days, then it is considered permitted. And you have to submit form CT06A and you're done with it. So no need, I mean, it is sure that within 90 working days, the trial is approved. Either DCGI will get back to you, or if you don't hear anything from the DCGI, then it is considered approved. The same thing is applicable for BABA studies also. These are the various forms available. I thought I'll just show you this. This is available in the NDCT rules. Have a look at this. Yeah. Next was the compensation. Please have a look at the compensation clauses, the various formulae for compensation, and how to decide on the quantum of compensation, because DCGI wants the opinion of the investigator, ethics committee, and an expert committee. All the three are taken into consideration. So it is necessary that you train yourself in deciding the quantum of compensation in case if there is a SAE. So once again, I thank the organizers of this training program for giving me an opportunity. And if there are any questions, I'll be more than happy to answer the same.
Thank you so much, Dr. Ashok Sheno, sir, for your wonderful presentation. It's really heartwarming about new drugs and clinical trials. We get a lot of knowledge about it. Thank you so much for your conveying message about it. Now I heartily welcome our panel, Dr. Rakesh Somani, sir, to please share the session.
Yes, I'm there.
Please start your share this session, sir.
Yeah. So I don't have a formal presentation as of now. Okay. But let me express my thanks to Dr. Ashok Shenoy, who gave a very fantastic overview about the new, policies or features in the new drugs clinical trials to 2019 rules. Dr. Shenoy, you have cleared a lot of, our doubts. Couple of things which you mentioned and which I have also noted down. You said that Schedule Y is absolute, right? In this particular new policy. So what are the other alternative provided for this? If you can highlight more on that part.
Sir, the Schedule Y spoke about conduct of clinical trials in
Right.
India. Now, NDCT talks about the same. So it is elaborate. So Schedule Y, so the moral of the story is don't refer Schedule Y if you want to know the regulations for conduct of clinical trials in India. You have to refer
complete
to NDCT.
this. Yeah, this new rule is completely the alternative to the Schedule Y, right?
Schedule Y.
Now,
And not to the Drugs and Cosmetic Acts and Drugs and Cosmetic Rules.
That you have clearly mentioned. That is very important feature, not to the DNC. Per se, I think the Pharmacy Council of India has recently initiated to change certain provisions of Pharmacy Act also. Probably there is a need to change certain provision of DNC Act also in near future. We look forward for that.
Already the draft stage. It's already in the draft stage.
Correct. So because this new rule is very precise, particularly as far as the activities of the ethics committee, the functioning of the ethics committee, it is very elaborately defined about it. Also, one small question, or when I was going through a presentation, here the definition of orphan drug in your earlier slide, you mentioned that around 5 lakh population if it is suffering from that particular disease. So my question is there any statistical data or any kind of studies being done to evaluate this 5 lakh number? Do you have any thought on that?
It's nowhere it is mentioned which are exactly statistics. There are certain diseases that are classified under orphan diseases and the orphan drugs, and as you know, the government gives incentives for the pharmaceutical companies to manufacture, maybe some tax exemption or something like that. But still,
because these numbers seems to be a little bit
very true.
It's a questionable kind of thing, because ultimately even if one person suppose is suffering from something, as a medical professional, we need to look into that and try to treat it. I mean, that is what is
we cannot leave that patient. Very true. Very
correct.
True.
Absolutely. So I was wondering that any studies have been done for this 5 lakh. Okay, and this time frame of 90 days for clinical trials, if DCGI doesn't respond in 90 days, that is something unique, something good, taken by undertaken by this. Your comments on a little bit more comments on this, I would be expecting, because 90 days is a very crucial period. We could bring a vaccine in the market in a period of span of say 1 year or so, and so every day in such kind of thing matters a lot. So could you throw some more light on this 90 days period? If
Sir, usually after the submission of the application for clinical trials, there is a pre-submission meeting and there's a post-submission meeting also. The CLA may call for the pre-submission and post-submission meetings. And previously, because I started getting involved in clinical trials in the year 2002, and I still remember many trials we lost because the companies waited for DCG approval for 6 months, 1 year, and every time when we asked what is the status under DCGA consideration, etc. So now that the companies know within 3 months, everything will be clear, and it's exactly not 3 months because it is 90 working days. So they have put 90 working days, and I
that's
believe
important.
it is 5 days a week. And anything to do with the investigator, SAE reporting or something like that, all these are calendar days. So from the investigator side, it is calendar days. And within 90 working days, at least, we know the fate of the submitted protocol.
True.
So that is a welcome move. And
Very
sometimes it happened that after 90 days also, nothing happened. I mean, we didn't hear. So the investigators and the companies were confused what to do next. So the amendment has come in October 2022, stating very clearly, if you don't hear from us, you submit that form, CT06A, and you deem it as approved. So such a welcome move, and it is really getting streamlined. And with reference to ethics committees, I have received plenty of calls stating that, this has to be submitted, that has to be submitted. And the ethics committee members training does not mention NDCD 2019. What I'm trying to tell you is the DCJ's office has really become very rigorous and proactive, which we cannot undermine. We cannot take it in a lighter sense.
Absolutely. I mean, it is certainly a welcome move. Now, the last point, which as a discussion, if that, Dr. Singh, I have some 2 more minutes, right?
Sure, sir. Please go ahead. No problem.
Yeah. So what happened? More the rules, more the regulations, more stringent or more clearly it is defined. Many times there is delay or kind of what you call it as lethargy in the system also that has been observed. So I mean, here if you go through, I mean, if anyone go through the document, lots of things have been very clearly defined. So there is a clarity on one hand, but when more clarity is there, probably there are a lot of questions also, hurdles also. That is what is observed in every law or every rule and act. So it shouldn't happen that these particular rules have made a job of a researcher or a clinical trial person very difficult or something like that. Your comment on this, Dr. Shenoy?
Yeah, sir. For the period from 2000 probably to 2008 or so, Indian regulations were not that stringent. That is why there was a hike of clinical trials in India, and there was mushrooming of, I would use the term mushrooming of ethics committees also. Some 4 or 5 people will get together and they'll form an independent ethics committee, and there was a mushrooming of clinical research training institutes, ethics committees, and the investigators also from private setup, something like a private clinic, private nursing home, wherein this is an area with lot of money. The grants are quite high, and there is always a chance for a human being to slip from his ethical standards when so much of money is involved. So many unethical practices were observed. It came in papers also. Even the Times even showed Indians were used as the guinea pigs. Definitely, the parliament took it seriously. DCGA was put a whole lot of questions, and immediately we know what happened in 2008 or something like that. The previous year approval was 100 plus. The next year after this phenomenon, the approvals became 4, single digit. And there was a necessity to make the rules stringent, and today the Indian rules are most stringent. So the companies do come, the compensation which was not there before, now it is very streamlined, it is very clear. And even we get letters from the DCGI, please submit the bills of the free medical management that you are given, because I'm sure the audience here, what is free medical management and what is compensation. They are different. Free medical management is different, compensation is different. We have to submit the bills. We have to send by email the scanned copies of the bills used to treat an adverse event as a part of free medical management. They are so proactive. So escaping from all this is almost impossible. And the patients are also aware that they are eligible for compensation. Informed consent form is audio-visual, and there is auditing by ethics committee as well as the regulatory, and ethics committees have become more powerful. So if at all there are any ethics committee members in this audience, please note, you have become more powerful, and that means you have to spend more time, you have to get more trained, and your duties have expanded. Previously, the monitoring of the trials was not so much, in the sense, okay, if you go fine, otherwise it is okay. But now today, you have to go to the investigator site, you have to monitor, you have to do an auditing, you have to generate a report, and that report has to go to the sponsor. The sponsor looks for such reports. So all these things show very clearly that Indian regulations are more stringent, and I would say it is for the right, safety, and well-being of the trial participants, definitely, because I've observed for 2 decades the growth of clinical trials and clinical research in India, at least in my center, which is a quite busy center for clinical research. Definitely, it has improved the right and safety and well-being of the trial subjects. It is a welcome move. Stringent regulations definitely are required for everybody, for that matter.
Yeah, Dr. Shenoy, I mean, a lot of doubts you have cleared, and there are a couple of questions in the chat box also. Before I go to the questions or ask someone to take up the questions, you rightly said that IEC must be, I mean, it is a strong now, but training to IEC is very, very important, because if I'm not clear in my concepts, and then I will not be able to execute it properly. So I think we all should look into these aspects also. The organization like Climate Research Solutions, Dr. Singh, can also take up a lot of training session for the IEC members, because more you get trained, more you get the things clarified, and probably execution will happen. Then only the provisions in the rules can be appropriately executed. So please note on that aspects, and I over to you, Dr. Singh, for the further questions. Thank you so much.
Thank you, sir. Thank you.
There is one question. Any difference between IRB and IEC? No. Institutional Review Board and Institutional Ethics Committee, as per GCP guidelines, they call IRB. As per Indian GCP guidelines, we call IEC. Both are the same. And is that now schedule wise only for veterinary products? Only that part, only that is applicable for veterinary products, because we do not do clinical trials in veterinary science. We extrapolate probably the human information, and clinical trials are not done in, especially in the higher animals. Veterinary is something like higher animals, isn't it? So clinical trials as such are not done in them. Only the preclinical studies are done in as per the requirements. Rodents, non-rodents, you know that.
Yes.
I hope it is clear.
I think perfectly fine, sir. Yes.
Yes, sir. There are no more questions. So I thank Dr. Ashok Shenoy, sir, and Dr. Rakesh Somani, sir, for this awesome session for us. And let's move towards our next session. The topic of this session is safety narrative writing. For this session, I'm honored to invite Dr. Akshima Sahu, Pharmacovigilance expert, Climate Research Solutions. And I'm honored to give introduction about Akshima Sahu ma'am. Dr. Akshima, PGD, BDS PGD CR. She is currently serving as team leader with one of the top life sciences companies in India. She is a registered dentist and seasoned medical writer. Following her bachelor's degree, Akshima worked as a dentist, where she organized several dental outreach campuses in rural and remote areas. In 2011, she pursued a postgraduate diploma in clinical research and pharmacovigilance. In 2014, Akshima ventured into the field of medical writing and has experience of more than 8 years with various job roles. She trained her team in medical ethics and obtaining unbiased consent from trial participants. So I am honored to invite Dr. Akshima to please speak up this session, and I am heartily welcoming our panel Dr. S.N. Sriharsha Sir, Principal and Professor, Hillside College of Pharmacy, Bengaluru, and Dr. Giriraj Kulkarni, Principal and Professor, Gokaraju Rangaraju College of Pharmacy, Hyderabad. With this note, I am handovering the mic to Dr. Akshima ma'am to please start your presentation and enlighten us.
Thank you, Dr. Sakina, for the such extensive introduction, and I welcome everybody in the session for one of the most and very important topics, which is generally encountered in medical writing. Usually, the one very important aspect when a medical writer starts his journey, and there are various aspects, especially speaking when we talk of writing a safety narrative. So let's carry on with the session, and let me know if you have any doubts in between, and also we'll be clarifying all the doubts at the end of the session as well. So I'm sharing my screen. Let me know when you're able to see my screen.
Yes, ma'am, sure. The screen is visible, ma'am.
Thank you. Thank you for the confirmation. So let's start about safety narratives. So we'll be broadly covering the introduction, a brief introduction, the types of narratives, the therapeutic areas involved, the regulatory requirements and ICH E3 guidelines, regulatory review perspective narrative writing process, and challenges in narrative writing process, and very oftenly speaking, and the burning topic these days about the automation part, the automation of narrative, and also about the few tips writing a good narrative. So first, what is a patient? What is a narrative? Narrative, as the name only speaks, it is the story. It is the overall journey of any particular aspect. So patient narratives are an important part of clinical study reports and pharmacovigilance activities. Patient narratives is written for informed consent process, deaths, SAEs, and other events of clinical interest. These narratives are submitted along with clinical study reports. So broadly, we need to understand that patient narratives are not only involved during the clinical trials, they are also involved during the post-clinical trial studies. It's one very important document in while submitting the clinical study report, CSR. CSR, it's an extensive document, but it contains a lot of information from the safety. It has safety narratives also attached to it. And even during the marketing studies, which is called the pharmacovigilance, safety narratives also form a part of it. So one cannot avoid writing a narrative starting from, the journey of a clinical trial to, the life cycle of the drug, and even during the clinical trial process. So I'm very fortunate that as a medical writer, I have undergone a rigorous training on writing these narratives, and I have been a part of writing the safety narratives during my tenure with one of the companies in India. And also during the clinical trial process, I was a part of writing the informed consent narratives. And also, while I was working as a pharmacovigilance professional, I had written the ICSR safety narratives as well. So what are the types of narratives? The type of narratives depends on whether the trial is ongoing, the interim trials, or if it is completed. Informed consent narratives, patient and safety narratives, which are called the pharmacovigilance narratives. Narratives may be written in text or tabular format depending upon the requirement. So as I was discussing during my previous slide, the journey of a narrative starts when the clinical trial process is ongoing. So there, medical writer writes the informed consent narratives. So what is an informed consent narrative? We'll be discussing that also in detail. Then after the drug is circulated, once that clinical trial is completed or interim analysis is going on, then safety narratives are written. So most of the big pharmaceutical giants, they basically give this responsibility to some vendor, and you would see that most of the vendors, you will see there are various departments. For example, when I was working with Gynaps, then we used to write safety narratives for many pharmaceutical companies, and there used to be a set format or template for writing those narratives. And even during the pharmacovigilance process, when we are doing the case processing or entering that particular case in the database, there is a section of the safety narrative which is written for any ICSR which qualifies that the criteria. Then that particular ICSR, when it is entered in the database, the safety narrative pertaining to that ICSR is entered in the system. So we'll be discussing that in detail in the ongoing slides or in the upcoming slides. So as I was discussing, what is an informed consent narrative? So it is an important part of the informed consent process where every bit of conversation is being recorded or written live. And so what is that conversation? Obviously, there is a consent which is taken from the trial participant. So if I am a trial participant and, for example, for example, Dr. Sakina is taking my consent. So she must be, it's impossible that she would be, as a human, it is not possible that she would be able to jot down all the important points, what I said during the consent process. She will be explaining the entire protocol to me in my term, in the layman term, or in the regional language. But it is not humanly possible to remember each and every conversation, what happened. I may ask certain questions to her if I'll be a trial participant, and then she would be answering those questions. So this conversation or this particular consent process is recorded audio-visually. We know that now the only way we can take the consent is through audio-visual. We know that it has gone virtual also, but when it is a audio-visual consent process, or even if it is virtual, there are certain very important points which are there, while taking the consent. So what will happen is that as a consenter, she would go back to that audiovisual mode, and then retrospectively, she will check that what were the important points of discussion. So she would write or copy those points, and then those discussion points or the important points. For example, I asked that, will I get some compensation if I die during the clinical trial? Then she would answer, you can get compensation only if your adverse event was found related to the drug. Okay, so that's an important part which she's telling me. So such questions would be narrated by Dr. Sakina in her own language into that informed consent narrative. But definitely there is a template which is decided mutually in that site. When I was working in a vaccine trial, I remember that parents used to ask questions whether their children would have, what kind of compensation or if the child is facing allergy, then what to do. Every patient or every parent or every caretaker would have their set of questions. So there is a set format or template of writing the informed consent narratives also. But yes, those important parts of conversation are recorded in that informed consent narrative. And basically, coming to the next point, the complete narrative can be divided into 3 parts: the identificatory part, the main body, and the reviewer details. So what basically it contains? So when I will have to start writing, or when the example we have taken here, when Dr. Sakina will start writing that consent narrative, then first she will mention the protocol number, then she will mention the site number. She will also have to write subject ID, initials, the date of birth, age in years, PI details, the ICF date, and language, in which language, for example, she took my consent in Hindi, then she will have to mention that. And the main body will primarily contain patient complaints, medical history, patient's eligibility, explanation about the study, explanation regarding informed consent process, time at which subject signed and dated the ICF, time at which PI signed and dated the ICF. So as a consenter, it is very important that even if as a consenter, she will have to sign the form, but it is mandatory to get the sign of a principal investigator or, the co-investigator in that main body. No consent narrative can go without the sign of the principal investigator. So it is one very important aspect of writing the informed consent narrative. So, and what are the reviewer details? The name of the people present during the informed consent process. There could be a legal person who might be present. There could, that there are various conditions on which a patient can, or a participant can accompany caretaker or relatives. So the consenter has to write all those details. What is prepared by a CRC, the name, date, the signature, reviewed by investigator, the name, date, and the signature, everything is written. CRC is basically the clinical research coordinator. Generally, the clinical research coordinators only take the consent when the clinical trial is going on. So all these details are present in the informed consent narrative. Informed consent narrative is very, very important, because if some patient, or it is one of the important essential clinical trial document of that particular patient. And with that, mostly I have, when I was working as a clinical trial consenter, the other with the clinical trial consent narrative, other things like the Aadhaar card of the patient or other details stating that he's an Indian citizen, that those details are also attached. For children, for example, there is no Aadhaar card, or children who are infants, the details of the patients are attached with the informed consent narrative. That will prove that the consent has been taken from the citizen of India, or whatever the details are there are. He's legitimate to be a participant of that particular clinical trial. So moving on to another aspect of narratives, they are called as pharmacovigilance narratives. So what are pharmacovigilance narratives? So a patient narrative is written for each patient who experienced at least one of the qualifying events as per the criteria defined and agreed upon with the regulatory authority. Thus, a patient narrative may have more than one qualifying events and is submitted as an appendix to the CSR. So they are called the safety narratives. That narratives which are attached to the clinical study report, they are called as the safety narratives, and narratives which go to the ICSR, which when the case is processed in the pharmacovigilance or in the database, they are called as the pharmacovigilance narratives. However, for spontaneous reporting, a safety narrative is written for each event event or a couple of events that occur together. So this is there. So this is just wanted to show you that how that narrative is being added. That's just an example that where they are added, and you can see event summary. So when this is the particular section where in this format, a pharmacovigilance narrative is generally written. You can see subject X was age. So see, you can make out generally the template is fixed. We just have to fill in the details. Nevertheless, but the role of the medical writer comes when he or she has to narrate the event, which is here in the following slide. So I hope it is visible to everyone, how basically they will have to tell the hospital details, the lab parameters, everything has to be narrated well, and why, what event happened and how it happened. So basically, a narrative should include the assessment of causality, relationship to the study by the investigator and the sponsor. In the end, it is generally written that the so-and-so event was found possibly related to the drug, or the event was not related to the drug. So that kind of interpretation also has to be given from the perspective of the sponsor and also from the perspective of the investigator, and it may match or it may not match. So everything has to be written in the narrative. So talking about the therapeutic areas, so patient narratives are written all across all therapeutic areas. Basically, for every condition, patient narratives are written, and for all phases of clinical trial, from phase 1 to phase 4, depending on the drug under evaluation, molecule and its mechanism of action. So we know that it is written, we've been discussing the same. And framing the template for the narrative is a critical step that requires extensive knowledge of relevant therapeutic area. So generally, what happens is when we are writing a safety narrative is a narrative which is written during the phase of the clinical trial, or if some interim analysis has to go. So generally, the medical writers which are selected for writing those safety narratives are generally the subject matter experts of that particular area. And what happens is, for example, pharmaceutical giants, for example, Cipla is conducting a study, and they have given the responsibility to some vendor to write the safety narratives. So generally, how this process starts is there is an extensive discussion on the finalization of the template, because that template will be written, that template would be followed from beginning till the end of writing those safety narratives, whether it is an AE. So generally what happens is that the template for AE narrative is different, the template for a SA narrative is different. Essay narratives are definitely of high importance. So you would find that there are more details. I think somebody goes to go on mute here. Yeah, thank you. So generally, you would find that the template of an essay narrative in the clinical trial process is more exhaustive or more expansive than an AE. And why so? Because yes, essay is more crucial, and it is definitely more under the scrutiny of regulatory bodies. Hence, they may ask a more exhaustive detail for a serious adverse event. Rest is another thing. I mean, that essay could be related or could not be related to the drug. That's a separate story. But generally, the template of an essay is more expansive. So it is important for a medical writer to have a thorough understanding of the therapeutic area for working on a narrative project, as it helps in having a better understanding of the safety profile of the drug class and its derivatives. Definitely, it would be an ask, and if the team of medical writers is not that expert, or maybe the manager or the head of that particular whoever would be leading that project has to have the detailed therapeutic knowledge of that particular therapeutic area. And I know that I remember that when I used to write the safety narratives for dermatitis, so exhaustive training was given to us as junior medical writers, that we were made to study all the conditions in detail. and the my manager who was leading that project was the expert in that field. So that kind of environment is there when you are working as a safety writer narrative, which would ultimately be fixed to the clinical study report. So patient narratives are submitted to regulatory authorities along with CSR, which I was just telling you, and they are included either as a text in section 12.3.2 of CSR if the number of narratives is less than 6 to 10, or provided in section 14.3.3 as an appendix. So remember these sections, section 12.3.2, if the number of narrative is less, that is the number is 6 to 10, or if the number is large, then those CSR narratives are added as added in the as an appendix in section 14.3.3. So the narratives can also be appended in CSR addendum, and they may also be submitted as FDA or to other health regulatory response if the regulatory authority identifies and requests narratives for any event or events of interest due to safety concerns. So regulatory bodies may ask those narratives in silos. I mean, in particular also. I mean, not necessarily they may ask along with the CSR, and if they ask so, generally these narratives are submitted in alone to these regulatory bodies. And patient narratives are also submitted for DSMB, that is Data Safety Monitoring Board review, if there are any safety concerns for the study drug which need evaluation by an independent committee on an ongoing basis. So these bodies can ask for these narratives separately also. It is not necessarily that they will ask it with the clinical study report only, if needed so, because I was just telling you in my last slide that SAE narratives are generally read very, very carefully by these regulatory bodies. Generally, these narratives are not very lengthy narratives. They are they are quite short. I mean, they are generally of 1 page from top to bottom. If there are some tables also, they would not be that detailed, but even if it is generally a very, very small document, but the importance of this document is very, very high, because analysis of any SAE or AE is done through those safety narratives only. And regulatory bodies, I mean, they are quite strict when it comes of analyzing the SAEs associated with the drug. And they will carefully study those events, because what is a narrative? Narrative is the entire detail, or you can call a story of that particular case or the patient. So they would want to read that what actually happened. So while writing the narrative or while fixing the template of the narrative, it has to be done very, very carefully between the two parties, generally between the vendor and the sponsor. And once that template is fixed, one cannot generally deviate from that template. Small alterations does happen, but generally the initial few phases when the pilot writing is going on and the narrative, the template fixing is going on, it is very important that it is fixed in a way that it can detail every inch of that particular particular event. So what is the perspective of regulatory bodies upon the review of these narratives? So a medical writer has to keep several factors in mind while drafting a narrative. As narratives get reviewed by regulatory authorities, it is important for a medical writer to understand their role and expectations while drafting a narrative. I was just discussing the same. While we are fixing the template, we have to see that if I am sitting on the other side of the table, if I am under in the shoes of a regulatory body, then what all they would want to see? They would want to see all the lab details. They would want to see what happened, what was the course of the event. So I have to fix the template, keeping myself under the shoes of a regulatory body. Only then, that will satisfy the condition if the regulatory bodies are looking for the SAEs separately, or even if it is attached with CSR, it has to clarify all those details. So, what are the objectives during their review of during the review of regulatory bodies? It is to identify and closely examine AEs, SAEs that require further monitoring. I was discussing the same. To identify and monitor frequency, severity, and seriousness of unexpected AEs. To identify the factors which affect and predict for the occurrence of adverse drug reactions during patient-related factors such as age, gender, ethnicity, race, target illness, abnormalities of renal or hepatic function, comorbid disease, genetic characteristics such as metabolic status, environment and drug-related factors such as dose, plasma concentration, duration of exposure, and concomitant medication. So to evaluate whether the data are suitable for support of the safety analysis, to identify the limitations of the drug on basis of the data, and to evaluate and analyze drug-drug interaction if applicable. So regulatory bodies will take into consideration, or they would be viewing those narrative with keeping in to see all these particular factors which I just narrated, which I just spoke. So all these things they will look into while assessing or analyzing the AE or SAE narratives. So what is narrative and writing from a writer's perspective? I mean, what a medical writer has to consider while writing a narrative. A medical writer prepares a narrative template using various data sources like CRF pages, analysis data sets, pharmacovigilance database, clinical database listings, peer and scientific review by CRO project lead to check the document is accurate, complete, and consistent with requirement. Clinical review of draft narrative by sponsor or stakeholders, redrafting narrative after review comments if writer does not agree with clinical review comments. When the review comments are unclear, a conflict with evidence, these should be discussed with sponsors or other stakeholders and duly noted and filed. So review is one very important aspect while during the journey of the narrative process, and every comment, whatever comment is being given to that primary writer, that needs to be addressed. And if he or she is not agreeing to that comment, that comment has to be annotated, that comment has to be addressed. Why, if I am a writer, I will have to write why I agree and I have addressed. And also I have to give the reason to the comments to which I don't agree, but I have to give it with a proper explanation. I just can't say that I don't agree because I don't like it. It has to be with a proper scientific explanation, or it has to be related to the source document on which basis I am writing, because I'm considering CRF, I'm considering other things while writing that narrative. I'm taking into account various source documents while writing that narrative. So if, for example, my reviewer has given the comment that please do not mention the lab details, but I will have to I will justify the QC person that I will have to write these details because these lab parameters are important with respect to analyzing this therapeutic condition. Hence, that comment would be given by the primary writer, can be given by the primary writer. And if I missed some lab details or I have not mentioned the data correctly, then also I will have to rectify as a writer all the comments which a QC reviewer would be giving me. So quality check or quality control review is based on the final patient data, and due to large number of narratives required for individual study and small size of each document relative to the CSR, it is recommended that single QC review be performed towards the end of the process rather than QC review of the first draft and final deliverable. Yes, it is very important. Generally, the QC is done at the end, but very frankly speaking, I have experienced I was working for a vendor. The QC was done simultaneously. QC, I was in the QC team, and I know that as soon as, for example, 30 set of narratives were written, those narratives were given to me for the quality check. Quality check means checking that narrative from top to bottom, like everything from data check, data fact check from the template perspective. Also, a QC reviewer has to check that has that medical writer missed important details, like for example, some renal studies going on, and that medical writer has not mentioned that when assay, for example, some assays has happened on day 10th of giving the drug. So, and for example, as a medical writer, I've missed writing the lab details of the kidney function test. So QC reviewer can point out that why have you missed the altered medical or even if it is not altered, it is important that when that assay is or AE has happened, what were the lab parameters during that time? So that kind of small intricacies or small details need to be checked or need to be kept in mind as a writer, as a medical writer. So medical writing is just not about writing. It is I remember my colleague Sarabjit was mentioning yesterday that it is an art, it is amalgamation of the art and science. So one has to be artistic in writing as well. Finalization of template, it is your art, that how you are presenting the things, but at the same time you have to be scientifically oriented while presenting those details. And in the end, approval by the sponsor after the final review. Once the final review is done, then it is up and running for the submission. So what are the challenges in narrative writing? First is the large number of resources required for any given project. Definitely, if some phase 3 study is going on, I am sure there are X or nth number of A's which would have happened, and definitely huge amount of essays might have also happened if it's a large study. That's a separate story, that whether it is related or not related to the drug. But yes, the number of safety narratives is quite voluminous. Maintaining consistency in narratives due to multiple resources working on the same project. There are multiple writers. One writer cannot write all the safety narrative or the pharmacovigilance narratives or anything, any narrative. No one person can write. So definitely consistency is one major concern. That is the reason a lot of emphasis is given when we are finalizing the template. So requirement of quick TAT or turnaround time and timely deliverable with high quality. Yes, it is very important. I remember I used to work till 2 o'clock in the night at times because that was too short, and but that particular CSR had to go for the submission, and so even if some interim analysis is going on during that study, suddenly there you would see a shoot in or an upstride in the number of A's or essays, then the burden will come onto the team of medical writers that they will have to write those X number of safety narratives in that stipulated time. And not having standardized data sources, patient profiles for most of the programs. It is a problem because there is, yes, CRFs are there. They are general, CRFs are also there with that set template, but generally, the hospital details, everything is at times bizarre. I mean, it is very, very difficult. Generally, the printed copies come in a very blurred way. It is so difficult at times even to read that data. So it is an exhaustive job, I would say, in collating those source documents, understanding what happened when, and then trying to write that what one we cannot miss, as I was giving that example. For example, some renal studies going on, and I cannot miss writing all the important parameters with respect to kidney function test or whatever during that particular essay, post essay, pre essay. All these small things have to be kept in mind. And different types of data sources, that which I was speaking, because one study can go some studies are generally multi-centric, and also there are different hospitals empaneled. So a patient might go, for example, some studies going in Delhi, but then for that particular site, that site has empaneled various hospitals under that. So every hospital has a different style of giving the data sources. So that is a problem. High variability in template finalization before and after sample narratives due to differences in opinions of reviewers, and change in reviewers during immediate later stages of the project, delay in review cycles, and not receiving timely clinical team consensus on the address comments and pending queries. So these all are the problems or the challenges which come while writing the narratives, safety narratives especially. So coming to one very important section, automation of narratives. Automation as it's one of the most burning topics these days. But to be very honest with you, automation of narratives has happened long back. I mean, I know in year 2014 when I was writing safety narratives, I have undergone through and have encountered basically the automated narratives, and I used to feel so that was the first time I felt that if everything a machine can do, then where is my need? I mean, I used to only do the data fact check at times, and I used to feel a little discouragement that where would be my role here. But surprisingly, that project could not sustain more, because I was just discussing in my previous slides that machine cannot do everything. You will even have to make the machine learn things. So slowly we used to enhance the tool. For example, I gave that example that some essays has happened. So here the human analysis will come, that I will have to record the kidney function text or any small things from the small source documents which are pertinent with respect to that event and needs to be added. So ultimately, most of the narratives had a lot of issues during that time. But now it's a it's a decade back that that automation narratives which I had seen and I had reviewed. But now obviously automation has also grown. Everything is pacing up and everything is becoming more sophisticated. So automation of narratives is quite a possibility. But yes, the role of human being, or that medical analysis or that medical eye or that writer's eye would always be needed, maybe even for the quality check or writing those narratives. Adding those finesse, or adding that finesse or adding those details which are mandatory to for the regulatory bodies to see or to check what could be the possibility of that event, or whether it is related or not related to the drug. So in the current digital era, as we know, that revolution in technology has brought modernization in healthcare industry, and we are not untouched. Writers are even not untouched for that. So automation in narrative writing is current trend. I would say it's not yes, it is written current, but yes, I've been experiencing it since a decade back. And obviously with ChatGPT or these sophisticated tools, it will become more refined. And the tool which helps in creating automated narrative extracts relevant information from all the identified patient desired format or template. It can extract, yes, but it may not extract also. So a human being would always be required. So the tool is very helpful for studies with large number of narratives. Yes, when we have resource crunch, or we have, as I was discussing, that generally the turnaround time for such narratives is quite short. So in that case, automation should be of quite help. But we should not be scared of automation at the end of the day. And I was reading one very interesting quote few days back, that we are not to get scared by the automation or the machine. We need to get scared by the people who are learning automation. So basically, maybe your peers or colleagues who are up-leveling themselves in automation, they are basically our competitors, and machine is that, or automation machine is not basically a competitor. So learning automation is a healthy thing, and automation is always a boon. But yes, we should not be scared. One cannot replace God's creation, I would say, or a human being, because that is irreplaceable. So we would always be needed. We were needed, we are needed, and we would be needed, especially in the field of medical writing. So what are the key factors that one should take into account when one is writing the safety or the patient narratives? Consistency, high quality with minimal or no error, pre-formatted and editable narratives generation, which gives flexibility to modify based on the need. This automation definitely does. Decreased turnaround time for overall narratives, ability to generate graphs and bar diagrams, and maintain audit trail and version control at each. It is very important. If whatever changes you are doing in your narrative, it has to be version control. And generally, the older copy becomes obsolete, but yes, it is generally not removed from the system. And there are, I have written few tips because these are self-created tips by myself, because when I was working as a safety writer narrative, these are very important points I think all medical writers should know. And frankly, I landed up as a fresher, and there were very small details. And, generally, it happens if somebody knows these small details, one is considered very smart. So I think these details I just wanted to share with you, and I'll quickly share. So always use hard space and hard hyphens for days, dates, and lab values to avoid splitting on the next page or next line, except at the beginning of the sentence. And how we do it? Control Shift hyphen and hard space, and Control Shift plus space. So if you will do like this, you will say that date is not breaking. It will be consistent. It is one very important thing because in a safety narrative, when you are writing a safety narratives, this is considered as and your QC reviewer will give you comments if you are breaking the days, dates, or lab values. They have to be in synchronization or in a one page, or they have to be mentioned in one page. So be consistent while using patient or subject throughout the narrative as per agreement with clinical team. Tagging the narrative template against the information available in data sources, which helps the QC reviewer to trace the information easily. You will have to mention from which source document you have pulled this information. It will help the QC reviewer. Always use generic names for concomitant medications or AE medications. Exception is for combination drugs. Generic drug names should be provided along with trade name in bracket. For example, lisinopril hydrochlorothiazide, it is the brand name is mentioned in bracket. So basically, the idea is to write either the generic name or we basically have to avoid writing the trade name unless and until it is a combination drug or generic name of the drug itself. But this is the way generally it is shown, or it is written in the narrative. Always use preferred terms for the event and medical history or ongoing history. PT is to be mentioned. The verbatim term should only be used if there are some PTs with two different conditions. Example, PT pain in extremity should be reported as pain in hands or pain in legs. So preference should be given to writing the preference term. This is very, very important point. I think if you know while you are going for an interview or somebody is asking you, if you are mentioning that PT is to be preferred, then you would get an edge that this person knows such small details. And self-QC by the author is very important. I always used to do it first time right approach. So if you are putting less burden on the rest of the team members, never write anything, or as a medical writer, we should never take that, okay, I am writing QC, somebody else has to do. It's okay. I mean, but then the approach should be first time right. So in conclusion, the purpose of writing patient narrative is to provide a concise summary of any event, including identified, specified adverse events occurring in a patient, to conclude a causal relationship between the investigational drug and the AE. So basically writing that story of that event, where we can conclude whether this event was related to that investigation product or not. And this helps in quick and specified review of vast patient data. So with this, I come to an end to this presentation. Thank you for listening actively to this session. And the house is open for all the queries, and any doubts. Thank you.
Thank you so much, Dr. Akshima Sahu ma'am, for your wonderful presentation. We got to know a lot of notes of medical writing. Thank you so much for enlightening us about safety narrative writing. Thank you so much for your awesome presentation. So now I am inviting Dr. S.N. Sriharsha sir, Principal and Professor of Hillside College of Pharmacy, Bengaluru, to please chair the session.
Yeah, good morning. Thank you, Dr. Sakina. First of all, I'd like to thank Dr. Ajit Singh, the Climate Research Solutions, for organizing this wonderful and informative and interactive seminar on the medical writing, clinical research, and pharmacovigilance. And also, I'd like to congratulate Dr. Akshima Sahu for the wonderful outline and informative lecture on the safety and narrative writing. So, I noted some of the points. Definitely, safety and narrative writing is a brief summary of specific events experienced by the patients during the course of clinical trials. So it's definitely it's in a complex process. Okay, it requires the high skill to do the medical writing. So there are a lot of challenges you told about the medical in the narrative writing. That is the large number of resources required for the any given project, and maintaining the consistency in the narratives due to the multiple resources working on the same projects. I'm just coming to the main question, that is maintaining the consistency in the narrative due to the multiple resources. So that will lead to the unclear safety messaging, and also it introduces the questions and potential discrepancies in the drug safety. So how we can overcome the inconsistency in the narrative writing so that it will make you more safer in the writing? What is your comment about this, ma'am?
Thank you, doctor, for that question. And yes, it is very, very important that how to maintain consistency. And as I told in my session also, that fixing the template, I think template fixation is one very important aspect when we are talking about consistency. Basically, we have to, and one very important aspect that medical writer has to take into account is that if I am a regulator, I'm in the shoes of a regulatory body, what I would want to see if in that particular AE or SAE. So for the example which I gave is, for example, I'm doing a renal trial. I'm on the kidney. So there is a kidney disease, acute kidney disease. So when I'm administering an investigational product or a drug, I will have to closely monitor that if, for example, some SAE happened on 15th day of post-administration of that investigation product. So I will have to closely monitor the lab parameters. What was the lab parameter on day 0? On day 10th, on day 15th, and even the post, giving the treatment or addressing that event, how were the lab parameters? What was the doctor's opinion? So that kind of thing and that kind of training to the medical writer team needs to be given, and this generally is fixed in the template. So fixing a template is a very rigorous task, and it is a very close and intricate discussion between the sponsor and the vendor, and generally these small details are added while fixing the template. So fixing the template is one important aspect when we are talking about the consistency.
But in the case of template also, we are finding high variability in the template finalization also. That will lead to the deviations. Again, that will lead to the inconsistency. How do you overcome that part, madam?
So I think, variation in template, this is not, which is there if you are giving the responsibility to a single vendor. Then there is a team of QC reviewers also. They will check that that particular narrative is written as per that template or not. And I will also mention that if something is lacking while seeing the possibility of that event to that investigational drug, that particular SAE might be asked by the regulatory body from that sponsor separately also. if something is missing. So, but talking about the consistency specifically, I would say that writing as working as a safety writer narrative, I have not observed such kind of feedback unless and until, the quality, there are quality issues since inception, because QC is also three-layered. First there is an internal QC, then there is an in-house QC, then there are the QC members who are being appointed by the sponsors. So consistency, I would say that template fixation, that is addressed, and even if small loopholes are there, they are addressed during the final stages of the quality check.
Okay. And also you mentioned about the automation in narrative writing also. Okay. And you mentioned that people learning the automation. Okay. So in that, what are the challenges you have faced in the actually narrative writing? Is it the automation is the solution for that, or how actually you can give more importance for the people learning the automation like that?
Yeah. So that's a very important question, sir. And automation is becoming more of a scare than a help these days. But I would say that, as I was mentioning during my session also, that automation cannot address anything. Nothing can surpass human mind. I mean, currently I'm working on a project where I'm analyzing the huge data set and extracting the market strategy of that drug. And I am also working on an in-house tool, which is regulatory getting updated to get that information. But unfortunately, whatever crude data is coming from the backend, it is not possible. It is currently not possible to automate from the scratch. I mean, what patient is telling you, how doctor is writing the prescription, that cannot be automated. Similarly, when we are writing the safety narrative, we are also seeing the doctor's prescription. We are seeing the lab parameters. Some lab people will just write in a very bad or bizarre handwriting that an automation tool cannot identify. There a human eye is needed. Automation to be successful completely in years to come, I think then automation will have to be employed from hospitals to the CROs to from top to bottom. But it's a huge investment, it's a huge task. And even if it is done, we cannot resolve the fact that a human eye would be needed to assess that event. But learning automation, I would say, is good. It is healthy, but we should not be scared because even writing a safety narrative, as you mentioned, it's a rigorous task. It's a complicated process. Even if the templates are fixed, then also mistakes are there. So a tool cannot check it. Only a human mind or a human being can check those details.
Yeah, wonderful. Only last question. With your experiences in the narrative writing, what changes you expect to be done in the narrative writing so that it should be more consistent and more successful for the future?
Yes, sir. So I would say that although narrative is a very, very small document, I mean, considering the complete CSR, it's a very short and narrow, very sweet kind of document. And but I would say that the way it can be more refined is that certain acronyms or small terms can be added. We are generally expanding terms. Obviously, we are not expanding it further, but narrowing down or, the template fixation has to be done in a way that certain terms to be a library of things can be given to the regulatory bodies. And, it can be made more crisper rather than more expansive because it has there is still a traditional way of going and writing the narrative, but just jotting down or bulleting the important points can help writing a shorter narrative. But then that's my perspective. It's might not be the perspective of a regulatory body. They would want to see that traditional way only, because I know it's still going on, that traditional way of writing narratives.
Whether this narrative writing will support the PSU or the actually periodical safety update report, does it support?
PSURs, yes. Why not? Because we are adding the individual case safety report narratives in the database. Any database, any vendor may choose, or a sponsor may choose. Even if I'm adding in Argus, that narrative would be pulled out, and those narratives would be summed in or understood in, in even the PSUR, they are important narratives are added.
Thank you, madam. Thank you for the wonderful clarification. Over to Dr. Giriraj Kulkarni.
Thank you so much, Akshima ma'am, and thank you so much, Dr. Hassan Sriharsha sir, for raising such awesome questions. And I'm honorable to invite Dr. Giriraja Kulkarni sir, Principal and Professor of Gokaraju Rangaraju College of Pharmacy, Hyderabad, to please take over about chairmanship of session.
Dr. Lakshmi, for this beautiful presentation. Almost you have made it very clear. There should not be any question arising out of it. But one or two small things I have. For example, you told about the reviewing of this narrative. Is there any timeline for this?
Yes, sir. So review also has a timeline, and the turnaround, it depends upon the turnaround time for that particular CSR, or if the regulatory body has asked for that solitary SAE narrative or an AE narrative, definitely there is a timeline. So for QC, generally, I have worked as a QC reviewer also, and I know that I used to run after the primary writers that please, if you can complete it, I will get more time to QC it. But generally, the a writer will take its own sweet time. It is on the shoulder of the QC reviewer to match that TAT or to match that timeline which is given by the sponsor.
Okay. Second thing, what I wanted to ask is, have you ever thought of nowadays we have AI and Chat GPT? What do you think may be the role of this Chat GPT in writing the narrative? Is it possible? Can we utilize Chat GPT for this?
I think, sir, Chat GPT would be able to write a safety narrative in a more sophisticated way. I mean, during the template fixation state, I would say there can be a role of Chat GPT. But once the template fixation is there, I don't see much role of Chat GPT. Chat GPT is good for can be good for scientific writing. It can help in other, like in content writing. But when we are talking about regulatory writing, especially, Chat GPT, I don't see much of much use. Only in the stages of template fixation, maybe Chat GPT can give sophisticated terms or sophisticated words, which can fast pace the process. But I would say that nothing can compete a human mind, because we cannot compete with God's creation. I mean, I would say that.
That's true. Thank you, madam.
Thank you, sir.
Thank you.
Thank you so much, Akshima ma'am, and thank you so much, Dr. Giriraja Kulkarni sir, for raising points and asking questions. It was an awesome discussion. Thank you so much, everyone. So without any delay, let's move towards our another session, which is the topic of the session is clinical trial transparency. Clinical trial transparency. And I'm honorable to invite our speaker, Ms. Sarabjit Sidhu, MSc, PGD, CRM, MBA, Manager of Clinical Trial Transparency, GSK Pharma, currently serving as Manager, Clinical Trial Transparency, GSK Pharma, worked at Kinase as Principal Medical Writer. Sarab has expertise in different domains of clinical research, site management, monitoring, and auditing, medical writing, and clinical trial transparency. She also shared her knowledge while taking sessions at different educational institutes in her free time. She has published a book with title Clinical Research and Medical Writing Career Guide. The book is available on Amazon. So, and with this, I'm also welcoming our panel guest, Dr. Muhammad Salahuddin, Principal Al-Amin College of Pharmacy, Bengaluru, Dr. P.K. Lakshmi, HOD Pharmacy Practice, G. Pulla Reddy College of Pharmacy, Hyderabad. We welcome you. And with this note, I am handovering mic to Ms. Sarabjit Sidhu. Please start your presentation and honor us with your knowledge.
Yeah, thanks, Kina. Thank you. Just let me share my screen.
Yes, ma'am.
Can you please let me know if you can see the slides?
Yes, ma'am, I will confirm.
Okay. So thanks everyone. So we will be talking around what is clinical trial transparency. It is little new in the clinical research field because it is evolving. So the plan of the content is I have kept it very simple. First is introduction to clinical trial disclosure, or you can say transparency or a public disclosure. These are different names, those are used. And we will also speak around what information is made public and what are the key regulatory requirements from US, what are the key regulatory requirements in European Union, and of course, what is the requirement in India. And I will also speak around layperson summaries because this is a new requirement. This is gaining a lot of attention these days. And we will also try to understand what actually is the difference between a protocol writing and you can say the protocol or result summary disclosure. And at the end, I wanted to stress on the fact is that disclosure is not optional. It is a necessity now. So what is clinical trial transparency? As the word transparency means that now there is a demand, quite a demand that the company should be sharing the information around the trials with the common public. So there should be some registry where they should be registering. So everyone knows that who is doing what trial, right? So that is the literal meaning of that transparency. We also call it a public disclosure, that we are disclosing information for clinical trials on a public platform. So these registries where we are disclosing these trials, they are mostly governed or regulated by the regulatory bodies like clinicaltrial.gov is governed by FDA, NIH, right? And European Union, they have their own registry. In fact, in a European Union, clinical trial transparency is part of your CTA application. They have gone one step ahead. So they have not kept it separate. They are saying that it is the part of a clinical trial application. So it is a broad term which covers a sharing of clinical trial data across international registries as well as sponsored channels. Sponsored channels, why I'm saying so, that all the big pharmas, they have made their own registers. Like Pfizer, they have their own register, J&J, they have their own register. And when they disclose information on ct.gov or EU website, they also disclose on their registers.
Madam, your PPTs are not shared.
Hello?
Can you share?
PPTs are not yet visible, ma'am.
So that's, I requested you to confirm, Skin.
Yes, ma'am. I thought you did not share yet.
Okay.
Sorry, ma'am, for that.
Okay, no problem. So because I was speaking, I was not knowing that it is not visible,
And make it
so,
slide share.
I think now it's visible. If I recheck.
Yes, ma'am, it is visible.
Make it slide share so that full view will be.
I'll do that. I'll do that. Let me put it on a slideshow. So, and if during the session also, if you lose, Dr. Skin, I'll request you just do let me know.
I will do that, ma'am. Sorry for that. I will do that.
No problem at all, right? So, so this is because there were not slides out there, so I can little repeat. So what is the content for today is that we will be talking around what clinical trial disclosure is, what information is made public, what are the key regulatory requirements in US, EU, and India, and what is the layperson summaries, because this is little trend now, or what is the difference between general writing and disclosure writing, and disclosure is a necessity. So So this is, I talked around is that what transparency means. There is a quite push from a regulatory bodies and general public also that whatever clinical trials that pharma companies they are doing, there should be information out there for a patient. So that is the reason this whole movement started. And then this is a broad term that covers sharing clinical trial data across international registries. These international registries are your clinicaltrial.gov, EU CITES, right, as well as sponsored channel. When I do see sponsored channel, that means all the pharmas, they have their own registers because they wanted to say that we are committed to transparency. So like Pfizer, they have their own register, J&J, they have their own register. So they also disclose information on these registers. So the information on these international registries is accessible to common public. So anyone from any common person can go to that register and can learn about, okay, which company on which therapeutic area they are doing the trial. So these are also known as a public disclosure. This is a new domain. It's like not more than 2 decades. Like it is just gained, started coming into a picture in like 2008 or 9. So now in the last 5 years, the rules and the guidelines, they have become more stringent, and there are new guidelines are also coming in a transparency domain. And this is the also reason all the pharmas, they have started setting up a different function. Earlier, what happened is most of the clinical trial transparency work either was handled by a regulatory affairs team or a medical writers team. So now they started, setting a separate function, which is known as a clinical trial transparency. So what is the advantage of clinical trial transparency is that it gives you improved patient care and it helps in research advances and overall better healthcare system. So what information we disclose, I have at the end, I will also show you on the registry, like you can also use that for your advantage. What information we are disclosing, that for at the 2 stages, if I wanted to categorize it, 2 stages we will be sharing information. One is when you start your study, right? And once your study is completed. So when you start your study, so you have to register your protocol before enrolling mostly the requirement is before enrolling your first patient. So you have to register your protocol before clinical trial start. And once your trial is over, you have collected all the data, you have collected all the, analyzed all the data, then also you have to disclose the result. Like what are the findings of these trials. So results are disclosed mostly either one year after study is completed. And in case of trial is terminated or it may suspended, so then whatever data you have to make it public. And information about protocol and the result is shared in a simple non-scientific language on international and regional registries as per applicable law. So if it is a global trial, maybe we are also disclosing on clinicaltrial.gov, European Union, as well as on Indian registry. And these days, this is must by law and ethics. So now why we should register a clinical trial? Because there is a lot of education going on in this domain. Because earlier people were hesitating to register their trial. So first is human subject protection, because one of our GCP pillar is right, safety, well-being of the subject. So the very fundamental principle, it helps us to increase the transparency of the trial. So you are not keeping your research with yourself. Right, you are open out there that, okay, this is our pipeline, this is what we are doing our trials. Of course, all the sponsors, they have the rights to protect their confidentiality, right? So maybe they don't want the competitors to know about that, but still they have to disclose that what phase, what clinical trial they are doing. It allows the potential participant to find the studies, right? It help the the participant who are interested to enroll themselves in a clinical research, so they can join in. Because on the registry, anyone supposed today, I want to search Like what are the cancer studies those are going in my area? Maybe I can put AIMS hospital, I can apply that filter, that will give me all the results, right? And it assists in ethical protection of the research. Another is the research integrity. When I say research integrity, then it reduces the duplicacy of the research. So if it is already known that someone already doing this kind of the trial, then there is no need to repeat that trial. And not only that, in the past, what happened is that a sponsor used to do is that if the results are positive, they used to publish it in terms of scientific manuscript and anyway, they used to share with the world. But if the results are negative, they used to keep it themselves. So others, they have found the same thing after doing that experiment only. So if, just imagine if they would have shared their results, even the negative result earlier on, the other research community would have learned from that. So that also help in a research integrity. It help in avoid unnecessary duplication, as I said. It helps us also to create an evidence-based medicine because it allows for more complete identification of a relevant result studies. Suppose I wanted to find out like what are the relevant research studies are going on, that I can identify, simply go to these registries and can find that information. Not only that, you will be amazed to know that even you are investigator, you find that this particular trial is very good for my research, right? And because on a public register, information is limited. You can also further request that pharma company to share their data. The transparency is to that extent. You can make an official request and then you can get, you have to, of course, give your rationale. You can get that data also. And when we are posting result on clinicaltrial.gov, we are also posting protocol and SANC. So just imagine the level of our transparency. So it assists researchers in a meta-analysis, as said. And legally obligation, it is a, from US, India, all the regulatory authorities, it is a legal requirement. And you also won't be able to publish your research if you don't have NCT number, if you don't have a registration number. So you have to oblige for this. Now, I will also talk about what are the major registries so that after this session, maybe you can do your own research. First is, I will also show you at the end, clinicaltrial.gov. I have made a note that I will show it to you live. So clinicaltrial.gov, it is like one of the biggest registry. It is developed by NIH, National Institute of Health. So in collaboration with FDA. So it is the largest clinical trial database. Mostly sponsors, especially big pharma, they are saying that our choice of registry is ct.gov. No matter in which part of the world they are doing research, but they are saying our choice of registry is ct.gov. And they are disclosing all the interventional studies on clinicaltrial.gov, protocol as well as results also. And that one unique thing for a clinicaltrial.gov is, which is not with other registries I have seen, is that they also review. The moment you submit the record, say I have submitted the protocol summary, they will review it. And if they found that there's an inconsistency or there's something, they will send me QA comment also. And I have to address those comments. And same goes with the results. And these observations that they give, they are quite logical. So they have their review process. Other registries, I have not seen to that extent that they have a review process. There is one registry which I came to know, Israeli registry. They review on a periodic basis, like after 2 years or after 3 years, based on their convenience, and they give us the comment, but not as stringent as ct.gov. Even a German registry, they also give the quality comments. EduCities, earlier it was known as a Udacity, but this year they have launched their new clinical trial information system, which is known as CTIS, and it is run by European Union, and it is also clinical trial register. The difference between these two is, if I wanted to say, is that look at this, clinicaltrial.gov is handled by NIH, right? Not directly by FDA. So your clinical trial application and your disclosure are two different things. But here in a European Union, disclosure is a part of your clinical trial application, which means when you are giving your application to EU for to conduct a clinical trial, you have to disclose your protocol at that time. So you have less time here. Here you can enroll before the enrollment of first subject, but here you have to give at the time of dossier submission. So in India, we have also CTRI. I will highly recommend you, go on these website and do your own research. And India, again, it is also developed by ICMR and National Institute of Medical Statistics. They are handling it. It is basically online database, right? And all the research that we are doing in India, you have to register here. This is applicable to interventional studies, but there are sponsors, those also register voluntarily their observational studies. Now, I will also talk around that what are the elements we disclose, right? Sometimes people get, quite scared by these regulatory requirements, but these are very simple. Now I can show you. This is a little background, like when it started. See, in 2007, it was first we hear about it. In 2008, these basic result. Earlier, they said you have to register only protocol. Then they said you have to publish your result also. So then this is our international journal also emphasized on the importance of transparency. Then they again came up with new amendments and proposals. Then finally in 2017, the final rule became effective. They made it quite stringent, right? So now if I talk about US regulation only, so what trials those are, if I say that what are the trials, those are mandatory, those are legally you need to disclose on clinicaltrial.gov. These are all interventional studies, and from phase 2, phase 4. Phase 1 is excluded, right? So, and if the trial is in US FDA, if the IND is registered with US FDA, and all the trials, those are initiated after 2007. So because before 2007, there was no such guideline. And when we have to register within 21 days of enrollment of a first subject. But generally, although we have 21 days of time, but generally what sponsors, as per their internal SOPs, they are doing before the enrollment of first subject. So this is also this type, although here I have written this US FDA, but this is broadly applicable to all the regulatory registries. They are asking for all the interventional studies from mostly phase 1 also. For EU is asking for phase 1 also. US is the only one they are asking for phase 2 to phase 4, right? Then what information we really disclose there, once you have your protocol should be approved with you before you wanted to register your trial. So you have to describe study type, what is phase, what is design, what are the outcome measures, like what is the goal of your study, all primary and secondary. Legal obligation is that you have to disclose primary as well as a secondary outcome measure. Then you have to explain the recruitment information, that what is the admission criteria, who can participate, location where you are doing this research, and little administrative information like key dates, when you are going to start your trial, when there is an end date of a trial, like this, and contact information. And for a result, that was about protocol. For result, what information you publish? Of course, trial information, general administrative information, what is identification number, what is sponsor detail. Then you explain participant flow. What I mean by participant flow is the in very basic term, how many participants participated, how many randomized, how many continued, how many discontinued, and why they discontinued. All that information you have to give it here. Basically, you have to tell that we started with 200, but we ended up with 170. Then you also have to give them a math of rest of 30, right? So that is participant flow. And baseline character is very generic information on a demographic, like what age group, sex, gender, race, and ethnicity of that populations, and where you actually conducted the clinical trial. Now, this is important. Outcome measures. Outcome measure is what was your primary goal and what was your secondary goal, right? So all the data of that endpoint is revealed along with statistical analysis. I will also show you the example, so don't worry. It is very interesting also, if you are a researcher or a student, this can upskill your learning. Just if you have any confusion, anything you wanted to know, go to clinicaltrial.gov, go to CTRI and learn about things. And we also present safety data. When safety data comes to adverse event data, all cause mortality, like how many deaths happened in the trial, what was anticipated, serious adverse event, other adverse event, and of course, administrative information, the dates and all that. What are the consequences of non-compliance? Like if you choose that, no, I don't want, this is additional work for me and I don't want to post on the register, what can be the consequence for you? First of all, internationally, no application is possible without prior registration where trial is mandated, right? So that is one thing. Plus, ethics committees, they are also emphasizing a lot on the posting on a public registry. That is also one of the reasons sometimes your trial may be observational, but you have to post it. Why? Because ethics committee asks you to post out there. And this International Committee of Medical Journal Editors, they can reject your publication. So they made it mandatory. Most of the journals, they have made it mandatory that if you don't have a registration number, we are not going to accept your research paper. And this is the also US also said that that we will be finding the sponsors also, but till date they have not fined people. They have just said this, but I'm expecting soon they will be started finding the pharmas. But we are getting letters. If you are not posting your result, they will send you the letters. Why you have violation letter, that why you have not posted your result, your protocol is out there, or why you have not. But pharmas, they don't dare to do this because the consequences are very dangerous. And of course, this can be one of the major findings in audits and inspection, right? Now, when if we are talking about the regulatory requirement in European Union, the content, what is disclosed on all the registry is same, but only difference is, I just wanted to talk around what is the key difference. Key difference here is this. In US, their legal obligation is that you have to post from phase 2 to phase 4, but here they're saying from phase 1 to phase 4. So this is new system they have launched this year. This is mandatory. They launched last year. From this year, we have to use it mandatory. And I have given a link also for a register. You can visit, right? And now I just wanted to also touch upon around a layperson summaries. I'm getting here a little ambitious because the layperson summary is a new thing in a clinical trial transparency, and we are also building capabilities, and it is gaining a lot of attention these days, especially from patient advisory groups, right? So because this is basically, if I'm talking around this, it is a patient-centric document, just like your informed consent form. It is also patient-centric document because especially patient advisory groups, they were saying that research participants, they are the heroes of clinical trial. I mean, without them, no trial is possible. So they have all the rights to know the results of clinical trials. Right. Not only that, they said that they should be knowing in their own language. Because if I am sharing in a quite scientific language, that is not making any sense to a normal person. So what we have to do is a layperson summary that we have to communicate to a research to non-specialist audience. One of the, when we this is the guideline, this is launched by first is European Union. So when they first initiated their, released their guidance document, they given a kind of a guidance, like just imagine you are talking to your neighbor or your family member who is from non-scientific background, and you have to explain this medical stuff to them. You have to come to their level. So like I say, these are very basic examples I have written here just to make you understand. We follow certain principle like health literacy principle. Instead of using hypertension, that is our term, right? So instead of using that, we will be using like high blood pressure in a writing document. And as per numeracy principle says that don't leave any calculation to a patient, to a normal person. Like 52 weeks, if I'm writing, then one has to calculate how much time it is. But at the same time, if I'm saying 1 year to a normal person, they can relate it very easily, right? But 52 weeks to a scientific person, that is very natural, very easy. But numeracy principle says that we have to explain in their language. So I will be using 1 year instead of 52 weeks, or say, instead of saying percentage, I have to say 48 out of 100, like this. Instead of saying 2 ml of blood, I will be saying like half teaspoon or something like that, right? We have to tell them in their language. And also we have to see the reader sensitivity. We can't say that people suffering from this disease, that is not sensitive, that is not respectable to the patient, right? So instead of saying that, we should be saying living with, right? Something like that. So that sensitivity we have to generate. Now, because in our all sessions, people are our audience is very interested to know like how we can use artificial intelligence or tools out there, those are available. I'll say that here is the scope, layperson summaries. So they, in pharma that already has started, they are creating a dictionaries that how you can break down the scientific words into the simple words. Now, even here you can, as a person, as a medical writer, I can still think around, okay, hypertension, I have to write in this way. But just imagine when we have to explain the mechanism of action on this receptor, on that cell, right? So when we have to explain that complex stuff in everyday's language, that becomes very hard. So that is also one way that with the help of artificial tools, they are developing dictionaries. So as a writer, I'm having dictionary, I can, just find the relevant word. Like a placebo, if I can't write a placebo in a layperson summaries, I have to break it down. Maybe like a copycat drug, or I can say the drug which looks like a drug but doesn't have any drug in it, something like that. But instead of thinking about that simple language every time, if I'm having a dictionary with me, I'm just putting a placebo in that, and a simpler language is in front of me, that is going to be very helpful, right? So here is the scope of AI. Now coming to India regulatory requirement, I have given little basic information is that it is a free online public record. You should go today only and check. I think most of you already know it as well. This was launched in 2017 as 7 as a voluntary measure. But in 2009, it was made a mandatory requirement. So again, what is disclosed? All interventional trial, irrespective of phase. It's only, I have found interesting, only clinicaltrial.gov is, they're little flexible. They are saying that from phase 2 to phase 4, but most of the regulatory, they are asking for all the interventional trials. And in a CTRA database, you can register your protocol. And CTI also requesting or suggesting is that observational studies are not required to register. So the universal logic is that all the registries, their requirement is to register the interventional studies, not a observational studies. This is how it looks like. Please go and visit, and then you can here type a trial, study design, trial phase, sponsor, recruitment status. These are the keywords that you can use to refine your research, right? And I always tell students when we take session in the courses that, make it a point to visit these registries, because it is loads of free information is available there, right? You can take advantage of that. Not only that, suppose that maybe you are having an interview on some research site, and I will highly recommend you to come here and get information about that research site from CTRI. From CTRI, you can pull a data that what are all the trials those are happening on that site. That will help you to prepare for your interview. So how it is different from a medical writing, and how a medical writer and disclosure writer together they can work. So medical writers are mostly, they are working on like a protocol, clinical study report. And now, as I told you, that because the work is increasing a lot in a transparency domain, the pharmas, they have started setting up a separate teams, which is known as a disclosure or transparency team. One thing is that they need to adapt with fast changing guidelines in a clinical trial disclosure space. I mean, since the time it has came into picture, 2007, 2009, every other year we are having a new guideline. So we have to adapt according to fast changing guidelines. We should aim that our document are disclosure ready. Why I'm saying that we should aim that disclosure ready? Because I told you at the time of result, we also post protocol, we also post statistical analysis report. We have to redact the important information. If there is some sensitive information which is confidential for a company, we have to mask it. So if tomorrow is my submission, I can't do that today. So we have to think and make a strategy in advance. And all the source documents should define primary and secondary endpoints very clearly, because the legal obligation is that we have to disclose the primary as well as secondary outcome. So this also should include disclosure medical writers in a review team, because mostly when we are developing these scientific documents, most of the teams are involved, but still there are only few innovative pharmas, they are involving disclosure writers also, but they should be part, because they can tell you the registry perspective. Now, these are like little steps I have written here is that what it actually requires. Disclosure is like a very ambitious field. You can't work single person. You need a biostatistician, patent, medical writer, medically responsible person. So all these stakeholders, they get involved in the disclosure. It need a continuous brainstorming, because just imagine your CSR is thousands of pages. And here in a disclosure, you are condensing that information to like 10 or 15 pages. So that need a lot of work, right? And of course, because we are getting a lot of new updates, we have to continuously update our SOPs, work instructions. We have to educate our study teams also. Sometimes they said that, oh, no, this is very sensitive information, we can't disclose it. So that is continuous effort to educate them. No, it is a legal requirement. We have to disclose it. So working on programming, and this is, as I said, one field, we are already using a lot of tools in a disclosure. So there is a scope. We are also taking us these coding language, Pythons, they are generating some programs by which we are taking advantage. So this is like discover, design, then execute and analyze and keep on optimize. So this is one of the area where we have to improve continuously. So because till last year, we were settled. This year we have new guideline. Now we have to update everything. We have to educate our teams also. We have to learn for ourselves also. So with this, I can let me share the real examples with you. I have collected one or two for you. Please let me know if you can see the browser. If you can see the browser.
Yes, ma'am. I'm able to see.
Yes. So this is clinicaltrial.com. You can come here and then type in the Google clinicaltrial.com. And if you see on this side, look at the number of the research studies this database has. If you are feeling clueless or something that or maybe you have some block, maybe, and you want to get the information on some topic, I'll highly suggest come here. You will get a authentic information. So this is it has a data from 221 countries. So if I just simply type here, lung cancer, whatever you can type, even you can go to advanced search also, that you can explore at your own. I simply press the search button. Look at the number of the trials I am able to see. It's more than 10,000, right? So not only that, you can also able to see if I wanted to see I wanted to apply some filters that recruiting only, maybe the trial, those are completed. If I am interested to know that, let me see why all the terminated trials, why they terminated or suspended. You can apply filters from here, right? So I just wanted to see this trial is completed. Let's hit this. You will be able to find all the information, like when it is first posted, when it is last posted, what is NCT number. These are the number, like, every registry assigned this trial. Like CTRI also assigned this number, clinicaltrial.gov, they also assign number, Udacity, they also assign number. Without this number, your international journals, they are not accepting your publication. So keep that in mind. So here you will always be able to find like which hospital is doing this research, who is the sponsor, right? And the amazing thing is that, see, study detail is there. Just look at it. Results are not there. Means they have not yet posted. It is to be posted. So this is how they disclose the protocol. You will be able to see the condition, short description, right? How many participants are participating, what is the trial design, right? When it is starting, when it is closing, and what is the cohort. This looks like an observational study, still they have posted, right? What is the primary outcome measure? And here is your study population. So this is how you will be able to navigate this, what I say, that registry, and you can also use it to your advantage. I also wanted to show you one result. So I can come here and can apply filter, is that with results, right? So with result only we have to see, and I can apply. So lung cancer with results, let me hit this. So these many studies has posted results. So so this is again a research done by some university, not by some one pharma company. So look at if I see the result, it will give you the study design, condition, intervention, how many participants were there. This is participant flow that I talked about in my slide, like how they present, right? You will be simply able to see that how many patients started, how many completed, how many not completed, why the reason for non-completion. See, withdrawn by subject 3, death 3, right? So Now, again, similarly, you will be able to see the baseline characteristics, all the male-female ratio, race. Here you will be able to see the race, reason of enrollment. So only participating country here was the US. We are able to see it here. Then you will also able to see the primary outcome measure. So number of patients that completed SST treatment. So you will be able to see that what result they have achieved. Here is that result, like this. And similarly, you will be also able to see the safety information. Let me take you down. So here is you will be able to see the information on adverse events. All mortality, how many died? 3 people died out of 30, right? So even you will be able to see the data on serious adverse events. So this is how you can navigate this, website. And if you want to find some information on something, I'll suggest you can come here, Now, one more thing I wanted to show you is that layperson summaries, right? We talked about layperson summaries, so that you can also pull from Google a lot. I wanted to tell you that how it is written. This is one I have downloaded, written by Pfizer. It is available on a Google only. So in a layperson summary, because our audience is a patient, right? So then you talk in very simple language. You start with a thank you note, thank you to our participants, right? And then why was this study done? You explain them. See that if you start the first sentence, how simply it is written. Like this atopic dermatitis is a common skin condition that can cause many symptoms, some such as itching, redness, rash. So it is explained in very simple language, basically everyday's language. So this also explains like what will happen during this study, what treatment they will be getting. They also use some of the diagrams and all that, right? So here they have explained the design in a detail, like on day 15, what will happen? Sample will be sample of treated skin will be collected and used for testing. What will happen? Patient then entered in an open label, right? That design they have explained in a simpler language. And you will also see that they have written here double-blind treatment, right? So then you will be finding in the next line, they are explaining what
your slide is not moving.
It is not moving, ma'am. It is
Yeah.
stable at one position.
Okay, so will you able to tell me which part of the document you are seeing?
All cause mortality part, ma'am, and serious adverse effect part.
Okay, so I have to stop share, and thank you for letting me know, and have to share the screen again. That means, right? So now I hope.
Yes, ma'am, it is visible. Your screen is visible right now.
Okay, thank you so much. So I can tell you little again. So this is a layperson summary that I pulled from net. This is prepared by Pfizer. Then how they start is that they start with the thank you note to our patients, because they are the one, with the help of them we are able to complete the trial. So then they explain is that why was study done. So if you looking at this, the medical terminology, they have explained in a very simple language that it is a skin condition that can cause many symptoms such as itching, redness, rash, and that is scaling or oozing. But if it was a protocol, then we need not to explain this. But since this is the layperson something, we have to explain, right? And see, instead of using investigator, they are using term researchers, or they can use doctors, because a common person can understand that. Right? Investigator is a little difficult term. So they also explain like what happened during study, right? How this study was done. They also explain like this included adult patient with this disease. So what I was telling you at that time, if you see this, look at this. The patient entered the double blind treatment period. So here they have written double blind, but you will be see in a second line only they will
Sarabji, time
be
is
explaining
exceeding.
what double blind meaning. Okay, no problem. I lost.
5 more minutes, that's
I need
okay.
just 2 minute more. That's it, right? Not more than that. So double blind means is that patient and doctor did not know. So in this way, these layperson summaries are written. Idea behind to tell you that how they are simplified. And design also, they are explained in more diagram, infographics, so that a common person can understand. What else I can show you? So see how they have written in a question answer session is that what are the result of this study. So here you will be able to see some bar diagram or something like that. So look at this. They have explained in a very easy to understand way, like 4.5 point improvement, 2.1 point improvement. This was second group, this was first group. So this is how layperson summaries are written. So you see, most common medical problem, generally we say adverse event, but here you will be saying medical problems or side effect. A normal person can understand medical problem. If we are saying adverse event, serious adverse event, they won't be able to understand. Right. So this is how it is written. I won't take much time. I think Dr. Ajit has given me heads up. So we can move to our question answer sessions.
Yes. Thanks a lot, Ms. Sarabjit Sidhu ma'am, for your fantastic presentation on clinical trial transparency. We have got to learn a lot about clinical trial and the facts and details of it. And thank you so much for showing us live example too. I appreciate you for that. And moving towards our panel, we are very grateful to have Dr. Muhammad Salahuddin sir, Principal of Al Amin College of Pharmacy, Bengaluru. I request sir to please go ahead.
Very thank you, Sarab Sidhuji, for a wonderful presentation that has given a complete insight on the clinical transparency. Probably you took all of us to the websites also, where I hope people should visit and take knowledge more about how uh, the world is changing in the field of medicine, and people should. The transparency is very common. I have seen when I was in the field of the pharma, that transparency was not much common. Medicines trials where pharma industries used to take over at that time, I'm talking about a decade back, transparency was not much. The results are being done based on the personal contacts, and that's how the medicines used to be there in the field without any database, without any transparency. But now we have a very good clear-cut transparency in the field of the medicine. I have a question for you, as you have highlighted that the National Institute of Health or the European Union or our own ICMR's clinical trial registry. Amongst these three, as per you, which you feel that the regulations are stringent? Which one is the stringent one? Where it says that the protocols are very strict, the rules and regulations are very strict, need to be followed, whether it is Indians or US or the Europeans. Which one you feel that yes, this the protocol system of the so and so is very strict? Which you, according to, is the strict regulations? Second thing, is it mandatory that if an Indian carrying out the clinical trial has to register only in the the clinical trials registry of the CTRI of the India, or it can register in the US or in the European also?
Okay, so first, answering your question is that which one is the most strict guideline? Legally, all local or international. See, we are talking about US and EU, they are international. Local, India also has their own, Israel have own, Kenya, Germany, every country has their local registry also, right? Legally, they are saying it is required, right? But when it comes to stringent, I'll say that till couple of year last, a couple of years back, US guidelines were quite strict because they are reviewing also what you are posting. They are reviewing also. They are giving you comment also. But with the new guideline, European Union, they have progressed a lot because they are the one first who are bringing layperson summary requirement also. So both EU and US, they are becoming quite strict in terms of transparency, and they are sending sponsors notification also. Suppose you have registered your protocol and they have seen that a trial is completed and window time is over and you have not posted your results. They are sending you letters, notification, violation letters, right? So they are quite strict. And when it comes to Indian or local guidelines, they are not as strict, I'll say that, because as of now, we are not posting results. They are asking us just to register our protocol, which is quite simple. And also ICMR is handling it. It is not directly under DCGI, right?
How about copyright protection of the trials that are being carried? The transparency
Copyright
is very high. As the
Hmm.
transparency, I'm giving every data being presented. How the copyright is protected?
So that is known as a CCI and PI, right? Company confidential information and personal information, patient information and company. So as US guideline, they have a flexibility that phase 1, where is the highest confidentiality a sponsor can claim is a phase 1, right? That is like new to us, that is so confidential, that is phase 1. Then they are having flexibility, you can't post, it is not mandatory. Another thing is that these disclosures, they are not asking us to put a lot of information, they are asking us to put minimum information. So we can also disclose the information in a clever way, in a smart way, I'll say, that where I'm not talking about my dose, frequency, and administration. What is confidential? My drug and my product information is more confidential, right? So that I can disclose in a way that is a protected. That is one aspect. Another aspect is European Union, they also allow you deferral. Deferral is that you can take a permission that this is confidential, this is due to XYZ reason, I am disclosing late. You have to disclose, but you can buy some time, maybe of 2 years or 3 years. That is also, there is another area under the transparency, which is known as a redaction and anonymization. Because when we will be sharing our protocol and clinical study report, then you have all the data. And patient information is also there. So there are teams, they are redacting or anonymizing that important information before it go on a public register. So these are the tools we are protecting the information.
Thank you, madam. Over
Welcome.
to my co-panelist, Lakshmi ma'am.
I have a small question.
Hi, Preetam, I think, yeah. I can.
Yeah, Preetam. Hi, Sarabjit.
Hi, Preetam, how are you?
I'm good. So my question is maybe little in a different way. So we are talking about clinical trial transparency, right?
Mhm.
So we know about US and EU. There are a lot of transparency. But what is the idea? What is this transparency? How the transparency word is there in India? Because India is very hazy in terms of clinical trial systems. We have a lot of information, news of that. So in current scenario, what is the take of the word transparency? Even if we have a lot of regulations, a lot of thing in a very, what are the take on transparency in the land of India?
So India, if I look at the Indian guidelines, they have not said anything on a transparency as such, honestly. Only thing is that they have made it mandatory. First, when they launched CTRA registry, it was a voluntary. It was voluntary, that it is up to you. You want to register, good, okay. If you are not registering, then it's also fine. But now it's a mandatory, right? Mandatory means that now you can know that it is a legally required. Otherwise, as of now, they are not doing much in India. Another thing is that when you're part of a global trial, because the trials are mostly happening in the global, so you are also falling in the scope of international registers, that US and EU. So definitely you will be posting there also, along with our local or a national registry.
Okay. So, and I have seen, because I have not gone through the CTIS side of India, but I've gone through this EU's clinical trial site, and our most favorite friend, which is clinical trial govs. I have seen a lot of in lot of major trials. Maybe I've seen a lot of major trials where India is not included. So uh, is that a correct one, or because we are doing trials to take a lot of to get the data from lot of different kind of populations. It's not only Caucasian, or it's not only European, but India is already mentioned. But I've cross-checked with the internal areas wherein I've not found any sites in India, or India is somewhere not mentioned in some of the major trials. So what is your input on
So see, if that is that, you have to go back to your at the time of designing, right? So if my India is not a participating country, that it won't be get listed out there, right? So even if it is a major trial, if the research is planned to be conducted in Europe or in the US, then like the one example I was showing you on a lung cancer, the participating country was only US, right? So that information was only there. And then if you wanted to see where India is participating, I can suggest you go and check few COVID study where there are few sites from India, right? But again, if the participating country are from US territory only, then you will be finding that country. If the participating country are from European Union, then those country will be only listed.
Thank you so much. Another last question. I have already posed that question on that thing. So we are writing protocol writing, right? Protocol writing is written. So when does it start? When does it start of the thought of then we have to write a protocol? We have to write our details about the protocol. So, but my question was, when does protocol writing starts, and based on which parameters or data we initiate the protocol writing?
So you are asking about protocol writing or protocol summary for a disclosure?
Protocol summary. So when it is
protocol summary, see, as I explained in my one of the slide, is that once your protocol is approved, then you start writing about your protocols. Until it is unapproved, you don't start. Once it is approved, then you start with your protocols.
So that 90 days and all those things, submission is basically on the protocol summary. You have to keep on submitting the protocol information, right, to the regulatory body.
See. There are two different things. One is our clinical trial application, where we submit the dossier to our regulatory body, DCGI, FDA, that please give us a permission to conduct a clinical trial. Whereas the registry disclosure I am saying is a posting irrespective of that. We need not to submit some application to the regulators. We are simply logging into a registry and pushing our information there. So we had a lot of time. So that is going on parallelly when we can do this. And for European Union, because it is a part of a clinical trial application, there is one performa. You have to fill that only.
Yeah, there is updates have come on clinical trial application in European Union a few days back, right? Yeah. Thank you so much, Sarji.
Yeah, of course. Lakshmi ma'am, you were saying
Yeah. Good afternoon, participants, and good afternoon, Dr. Ajit, and thanks for this opportunity. Thanks to Clement, and congratulations, madam. So once again, opportunity to see Mrs. Sarabji. And it was a very excellent presentation, with a lot of clarity. So in fact, I think you have told that clinicaltrial.gov. So that is the website. Whenever I teach my students also, I always tell that whenever clinical trial protocol is has to be written, for their protocol or fifth-year projects, or to learn also, it is better to go to clinicaltrial.gov and see some similar studies and then learn about how to write the outcomes and how to write objectives and how to write aim, inclusion, exclusion criteria and all. So it's overall a very good session. I have the two questions before you, like this layperson summaries, which is only meant for EU CT, or is it necessary or mandatory for even the Indian, that is CTRI, and even for the clinical trial US one? That is the first question.
That law is being launched by European Union only, ma'am.
So not for India or US?
No.
Okay.
It's a requirement.
Yeah, please continue.
Yeah, it is a requirement from European Union, but there are few local registry like Israeli. They have started developing, but they have not came up with this regulation. But as of now, layperson summary requirement is from European Union only, no other international or regional register.
Okay, so the other question, maybe on behalf of the participants also, we talk about so much about medical transparency and most of the data needs to be disclosed. So is there any data which need not be disclosed or should not be disclosed, or it is not required to be disclosed by the pharmaceutical industry or from the academic projects or for this thing? Because CTRI talks about even the BAB studies and any of the part of the PG thesis also should be disclosed. So what about this?
Answer your question, if I refer my guidelines, right, either from US and EU or any registry, they are saying that all the data collected on primary and secondary outcome measures, you should be disclosing, plus all the safety data. Safety data, they are going to an extent, maybe it was not a part of your plan, maybe you have done just to check your operations, you have to disclose. So guidance from them is that any data that you have collected, you should be disclosing. So they are emphasizing more on a disclosure, not to keep it to yourself. So even they're saying that if you have terminated, suppose I have disclosed 5 outcome measure at the beginning, then when I am posting result, I have data for 2 only. Then I also have to tell them why I am unable to collect the data for rest of 3 outcome measures. So they are asking, this much of information, like why you are unable to collect the data for those 3 outcomes.
Okay, thank you, ma'am, and congratulations for a wonderful lecture. So all the best for all your future lectures, and we are eager to wait for that. Thank you once again. Thank you, Clement.
Yeah.
Thank you, madam. Thank you very much, and thank you, sir,
sorry, Dr.
for joining. कोई बात नहीं, अभी मेरा lecture जो है, I think I have to postpone, or I am taking another lecture in the evening, then another session in the evening, then I have to cover this and that. Anyways, this is just some information I wanted to give on result biased in clinical research that I'll give. Okay, so thank you everyone. Shall we go to the next session right away, guys, or in the evening only you want to cover? You want me to cover in the evening only? I'll take the opinion because this is a lunchtime and 2 o'clock sharp we have to start with Dr. Krishna Undela. So in the evening, if you guys want, I can cover 20 minutes in the evening extra after my regular session on protocol writing or grant proposal writing. I can take extra 15 to 20 minutes on this. Continue. Okay, great. Our audience is so enthusiastic and this is really good. Right, guys, so thank you very much. Sarabji, you're going to get a call from Siddesh. Hello.
Yes, Dr. Ajit.
You are going to get a call from Dr. Siddesh.
Yeah, I'm talking to him only. Yeah.
Okay, fine, thank you. So, yes, guys, you're welcome. And so this is the debate session I actually, included in this, and this is one of the most important session I felt that it should be there for all the, academicians and for the, PG students, especially for the PharmD students and for the even medical MD, DM students as well. Because when I went to the clinical research, I'm working with the hospital, I'm working with ICMR as scientist C right now. So I see a lot of, lot of discrepancies and lot of things with the students and their mindset that, sir, our results are not coming positive. Our results are not coming significant. There's, let me give you the background. So when you are doing a research, whether it is a, academic project or your PG project, your DM project, or maybe your PhD project as well. Okay. So before starting the project, you go to the literature search, you do the, good literature search and come up with the question. Definitely, the nice question you think that is the need of the hour, and that is the research gap. Based on the research gap only, you come up with the aims and objectives and the right question. And you select the methodology, the research design, right? You conduct the entire clinical trial, entire observational study, whatever is your research design. So if you are a student, might be a PharmD student, then within 6 months you try to do in certain number of population, retrospectively or prospectively. If you are MD or DM student, then you have to cover it, you have to complete your research in a year or a year and a half. And if you are a PhD student as well, you try to cover it as soon as possible within 3 years or 4 years. And definitely, I'm not criticizing at that point, because all the mentors, all the guides, and all the students, they come up with the questions and aims and objectives with their own, what we say, instances and instincts, that it is going to be one of the wonderful project, it is going to be one of the wonderful result. And we do that research, we collect the data, whether it's in the hospital settings, or sometimes in the animal settings also, sometimes in the laboratories also, if we are doing with biomarkers or anything. But when we do the analysis at the end, when we do the analysis, what we get is that we are not getting the p-value less than 0.05. We're not getting the significant results. What we hypothesized, okay, our hypothesis was like, okay, what we are thinking based on our aims and objective, based on our very critical and a very restricted inclusion and exclusion criteria, still you are not getting your p-value significant. Still your results are not showing any kind of significant results. Means your protocol or your project or your research, what you are doing, you think is failed, right? You think it's failed, it will not get published, it will not get accepted by the mentors, by the peers, by the people. They will just say that you have wasted some time, you have wasted your 6 months, you have wasted your 1 year, or whatever the time you took into the research. But guys, let me tell you, this leads, this mentality leads our students, a lot of students, I'm telling you, a lot of students. And I feel that is a very, very, what we say, the critical statement to state here, that it leads to the data modification, data malfunctions, data fabrication. And people think that if we come up with at least one p-value less than 0.05, if we come up with some significant result, our paper will be accepted easily, because that will show the new findings, that will show something significant findings, and that will be accepted. But did you think about the consequences? I've seen many people, really many people, who always think like, oh, there is a little, there's a just little difference. My p-value is 0.6. 0.06, my p-value is 0.07. I think, there is some mistake. Let's correct the data. That's not data correction, that's data fabrication. And what are the consequences after that? Did you ever think of like what could be the consequences? Suppose if you are doing a drug clinical trial or in observational studies, suppose you are comparing two drugs. You have a very good, perfectly fine, ticagrelor or clopidogrel in, for the MI patients, for the heart attack patients, suppose. Well-established, well-researched clopidogrel or well-researched ticagrelor is there available for the MI patients, for the, CVS patients, cardiovascular patients, CVD patients. After heart attack, we give clopidogrel or ticagrelor along with aspirin as an, antiplatelet agents. And it's mandatory to give daily, right? Suppose there is a new drug in the market and you are checking efficacy of that drug along with clopidogrel or ticagrelor. And you want to see the results and results are not favorable, right? So being a sponsor, what sponsors think that's different. But being a researcher, you also think like, oh, I have spent a lot of time, lot of efforts, lot of money, and lot of resources. Why it's not coming significant? Right, in your comparative studies as well. So what do you think like, okay, let's fabricate the data or let's alter the things or, let's alter the analysis. I'll tell you one by one, there are, multiple ways of altering the things. Okay, multiple ways of altering the things. And how people do that and how they lead to the wrong science, how they lead to the incorrect, publications, how they, lead to the, incorrect results and harming the life of the people, harming the life of those, all the patients. And they are endangering, seriously, let me tell you that they are, endangering the science as well in our, research. And that's why I think till, few years back, Indian research was really, at the back step. And when I really, came to this, scenario, I've, seeing a lot of researchers, a lot of, many of the students and many of the people, they come up with that mindset only that my research is not coming significant or my research is not coming up with the positive results. So, as I was giving you the example, if suppose someone is thinking that, okay, let's alter the, results, let's alter the Excel sheet, let's alter, a few things, few observations or few values in our results, and we'll get the significant result, and that paper must be published in JAMA or in, NEJM or British Medical Journal or, in any open science journal. Right, it will definitely give you the credits. But what are the consequences? The new drug will definitely, based on your research and based on certain other research, that will be approved or, that will come to the market. Or if it's in the market, then people will start believing on that. Or the doctors, the clinicians, they will start writing that drug again. Again means they will start writing that drug. They will be, taking that more frequently in the use. And once it is with the patients, after certain time, after some time, maybe after a month, 2 months, 3 months, 4 months, or a year, it will show the actual results, because you cannot alter the mechanism, you cannot alter the characteristics of the drug, you cannot alter the technical part of the drug or manufacturing of the drug. You are just altering your research data, right? So it will show the side effects, it will show the adverse event, or it can lead to mortality or permanent disabilities in the patients. So you are indirectly harming the patient, you are indirectly harming the society, you are indirectly harming the health, healthcare system. Right, let me tell you how people do that and how you should not do it. People used to do it from the starting only. They start altering in the inclusion and exclusion criteria only. when they feel like in sub-analysis groups or after a certain time they they do the preliminary analysis and they do not get the results, they start reducing the inclusion criteria. The restricted inclusion criteria, what they start is they start losing that knot. They start alterations in the protocol, and when they do that, that means they are altering the protocol. They are deviating from the protocol. They are deviating from the main path, what they have decided to achieve the goal. What they have decided to achieve the aims and objectives of the research, and it again leads to that, right? So at the first stage only, and once people are done with the what we say the complete research. If it is short-term research, you don't have time to do it at the first stage. In the long term, in the long-term research, they can do the alterations or modifications in the protocol in initial stage only. But if it is a short-term research like a 1 year or 2 years or 6 months, you don't have the chance to do that. But when you collect the results, what students or they think after the analysis only, either they directly write the p-value. They don't look into the data also. They just write the p-value and they just try to publish it in some if the not the good journals, they just publish in some journals, some maybe predatory journals as well. If not the good journal, predatory journals, they just charge the money and they will publish anything. They usually publish anything. There are guys, let me tell you, there are around 52,000 journals. 52,000 journals around the globe. There are 52,000 journals, and in 52,000 journals, there are 39,000 predatory journals. 39,000 predatory journals in all the streams. I'm not talking about medical, but all the streams: science, art, marine science, or all mathematics, computer science, everything. There are 52,000 journals right now in 2022. And out of 52,000 journals, there are 39 predatory journals. Predatory journals means these journals just charge the money. They are not following any kind of indexation, and they are not at all legit. They are in the Bell's predatory list, which is very well valid. Do not follow the review process. Do not follow the ICMG guidelines, which is committee especially made for the editors to look after the journals, if they are following the reviewing process, if they are following the journal guidelines, if they are following the certain things which should be followed, and get the papers published, right? So that's how can you imagine 13,000 and 39,000 means only 20 to 25 journals, 20 to 25% journals are legit. Otherwise, you can publish anything. You just pay the money, you publish. They will publish online, offline, anything, but that will influence the society. You will get the paper, but the harmness of the paper will come later, right? And it will be very, very bad for the science, bad for the research, bad for the publications as well. Now, I'll tell you how we can avoid and what are the advantages of avoiding such situations. That's very important to say. See, if you are getting a negative result, not the positive result, if you think your your research was a failure, if you think that your clinical trial was a fail, you did not achieve the objective. You did not achieve the objective. You did not achieve the aim of the research. Statistically, definitely, you look out statistically and clinically both, if you are thinking of, right? If you think that we did not achieve the goal, we did not achieve the aims and objectives of our research, fantastic. I'll say amazing. First of all, there could be certain mistakes. Okay, there could be certain mistakes. That mistakes could be at your planning as well. From the planning to the end, I'll tell you where could be your mistakes, where you did some mistakes, or where there is a certain chances of scrutiny. First of all, question. When you were framing the question, you might have not went through the proper literature research. you did not find the proper gap of the science or gap in the research gap, what we say is, or you could not define the proper hypothesis. Check for that. Check for literature search, check for research gap, check for hypothesis. And then the second part comes about the methodology. Your inclusion exclusion criteria might not be correct. Okay, your inclusion criteria might not be correct. Either you left some population which must be included in the in your research, or you have included such populations which should not be there in the research. Understand? See, suppose I want to do the research. I want to try and test some new drug in heart failure with preserved ejection fraction. So it is very simple. If I'm talking heart failure with preserved ejection fraction, where my ejection fraction of the patient should be more than 50, then it is very important. If I'm not getting the patients, because the patients are really quite less, other than the heart failure with the reduced ejection fraction or heart failure with mid-range ejection fraction, there are less patients for heart failure with preserved ejection fraction. So if you're not getting the getting your sample size fulfilled, it is not giving you any kind of authority to take the patients of maybe whatever is your inclusion criteria, like heart failure with preserved ejection fraction, but with other problems, with other like MI, or with cancer, or with some infectious disease, or with some diabetes or hypertension, which should not be there, because the comorbidity or the other problems could also influence the drug efficacy, could also influence the diagnosis, could also influence the path of the mechanism, and that could lead to the unsuccess or the negative results of your clinical trial. So first of all, look at the trial design, look at the study design, if it is an appropriate or not. If you want a case control study, why you are doing the cross-sectional study, right? If you have to do the cohort study, why you are doing the case control study? There are certain people who do not have the appropriate knowledge. So I understand that I have seen that people do not have the 100% knowledge of the research. But if they really want to do the research, if they really want to do certain kind of studies, then they should have at least basic knowledge of inclusion exclusion criteria, sample size, and how to choose the research design, right? If you are a student, then go and read about it, go and search about it, compare your study designs, exactly find out what must be your study design when you are choosing a study, when you are choosing the course of your research, right? Inclusion exclusion criteria, research design, and how you are putting your intervention, if it's an intervention or observational. And then if you are done with that, very important part is analysis. You might be choosing the wrong test as well when you are doing the analysis in your result, right? Suppose there are different types of data. There is numerical data, okay, there is categorical data, there is continuous data. In categorical also there are ranked data, there is unranked data or nominal data, multiple types of data. And people always mistaken with types of data, and based on types of data, all the statistical tests are being chosen, right? First of all, understand your data, what type of data it is, what type of test exactly you need, right? There are different types of tests. There are parametric tests, there are non-parametric tests, there are central tendency, and the normal distribution of data, not normal distribution of data. First, you have to understand your data, and based on your data only, you have to choose your test. And if you're not choosing the correct statistical test, you might get the wrong results. Okay, you might get the wrong results, and these entire thing will lead to your unsuccessful study results, or the failure of the study results. So if you have that courtesy to do the research, always have the courtesy to learn also the proper research methodology, proper statistics, proper choosing of the data and doing it appropriately. Okay, I'll not take much time because there are certain other things also. We have to go for lunches, everyone, and then we have to join again at 2. So now I was talking about the advantages of getting negative results. If you are perfectly fine, if you have done everything correctly, you have chosen the correct question, your methodology is correct, you have chosen the correct study design, and your results are also correctly analyzed, right? And still you are getting the p-value less than 0.05, or you feel your results are not correct. Got my point, guys? Then there is no need to feel bad. See, you got some new investigations with you. You got some new learnings with you, and those learnings are that, okay, if we are doing everything correct with this population, with this study design, with this intervention, with this sample size, this will not work. Okay, that path of research will not work, and this negative result could be published proudly in high impact journals as well. If you think these negative results could not be published, these negative results could be published better than the positive results. Why? Now I'm telling you why it is important to publish the negative results and why it should be published. Because it will convey the message that after doing everything right, this path was not correct. You will not achieve anything. So you should not waste your time, your money, your resources, your efforts, anything. This is not the correct pathway. Choose the next pathway. Try something else. Try alteration in this. You understand? Try alteration in this. Whatever you are doing is not correct. Choose some alterations. Choose some alternative paths to get and to do some more research, to extrapolate this, but not exactly what we have done. Some people, what they do, they try to repeat it. So when we are publishing a negative results, that means it is not recommended to repeat if everything was intact. Definitely, if everything was intact, term and conditions, if everything was intact, research design, research question, research methodology, statistical analysis, everything was correct, that means it was a negative result and this path will not work in the future. So the further your peers, your researchers, your colleagues, your students, anyone in the field will not repeat that and will save their time, will save their efforts, will save their resources. Nowadays getting funds is very tough for the research, for the clinical research, for the medical research, for any kind of research. So if you are not publishing negative results, someone else might may try after some time, or the same time somewhere else in the world, somewhere else in your country as well, or maybe after a certain time in your center only, and they will also get the failure again. Means they will be wasting the time, they will be wasting the resources, they will be wasting the funds and the the local hospitality, the research facilities. So always try to publish negative results and do not go for the result biased. Result biased could be done at multiple stages, as I told you, from the topic selection to the analysis. Topic selection, gap, research gap identification, hypothesis creation, research design, sample size. Many people do not know how to get the exact sample size, right? So you have to do it correctly. Okay, and let me show you something. So where you can publish the negative results, just 5 more minutes. Okay, 5 more minutes I'll take, and then you will understand what exactly it means and how exactly you can publish, and you will get an idea of this. So here, look at this. Look at this. International Journal of Negative Results. If you can see, International Journal of Negative Results. They proudly published only negative results. See, BMC is one of the big publishing house. The Springer Nature, part of the Springer Nature is BMC. Okay, BMC, Springer Nature also publishes the journals and Journal of Negative Results in Biomedicine. Okay, if you have the negative results, submit it. If you look at the publications, if you look at the papers they have published, see, in vitro aggregating beta lactamase poly-Q chimerase do not induce toxic effect in an in vitro conohabititis elegans model. So they are publishing do not induce, means they hypothesized that it will induce, but it is not inducing. So they have published proudly, they have published this paper in Biomedicine, International Journal of Negative Results in Biomedicine. See, look at this. Polymorphism RS5479H4 on human chromosome 1q43 is not associated with primary open angle glaucoma in a Saudi cohort. Look at the results, guys. See, this is the pure negative results, and it is published in a high impact journal. Okay, so there are specific journals as well for the negative results publication. So don't think about creating any kind of bias, creating any kind of thing in your results, right? And then there are a lot of things. Does the placebo effect modulate drug bioavailability? Randomized crossover studies of 3 drugs. See, they have published all. See, absence of the dermatan sulfate chain of decorin does not affect mouse development. See, they are proudly publishing the negative results. See, look at this. This is another Journal of Negative Results. Look at this. This is also one of the Journal of Negative Results. Okay, one sec. This is opening. Yeah, Journal of Negative Results. See, this is the Journal of Negative Results. This is also Journal of Pharmaceutical Negative Results. Journal of Pharmaceutical Negative Results. There are journals of negative results in all the fields, guys. Okay. So as this is an open debate session, I would like to give you the background now. I think we have Dr. Kanav Khera sir with us to put on some light on this. So he will put on some more light on this for the next 5-7 minutes. And then if someone wants to participate from the audience, this is an open debate session. If someone wants to, ask any questions, or someone wants to, put an example or share something, they can share. Over to you, sir.
Okay, thank you, Dr. Ajit, and good afternoon all. So I think, Dr. Ajit, this is a very, very important topic which we had took today for this debate with respect to the negative results. Because I think as an academician and researcher, I think all the journey towards negative results and the students getting afraid, it's all starts, what I will say is unpreparedness among the researchers. So especially when the researchers are in their early stages of doing research, like when they are in their 3rd year or 4th year, when they will be having the project, especially in B.Pharm or in PharmD when they are in 5th year. So the thing is, a properly planned and executed research will help them overcome this type of scaredness which they have, like in case if they get negative results, because they are not reading the journals properly. They are not getting enough time to read the journals and other researchers' opinions. So what I will think is, if the researchers are given a chance early in their academic stage to read a lot of journals, especially now, I think the world is going towards top list of journal, as you are telling, there are so many pediatric journals out there. And I was looking at some of the retractions which has happened during COVID time. And I think all the records had been broken of paper retraction, especially which are linked with COVID. So there is nothing to be getting afraid of it, because the retraction is now increasing. This is all because everyone is in a very big hurry to publish their results.
Yes.
And because of the competition in place, because of the performance pressures and all these things. And that is actually what is pushing researchers towards, when they are publishing unplanned results, they are publishing, and bias definitely in their result publication. A lot of bias are there where they do all the sort of malpractices to get the results quickly without following the standards, norms which are in place. So what I will think is, if a student prepare his research properly, that is, his literature search is proper, he's reading the top-notch journal, like we have Web of Science and Scopus indexing. So I think the students at an early stage should be given an awareness about these type of indexing, where a student can actually find the right journals which he should refer for, where the like research papers, whichever, actually he should not be attracted towards pediatric journals. So that is the first step in the research. And when you were telling about inclusion exclusion criteria, because when we go through the research protocols in our ethics committee of the students, and I think inclusion and exclusion is one area where the researchers are not putting much effort.
Yeah.
So whatever they think, like they will just put out, like, but they will not have any justifications like why they are excluding one group or including one group. So that is where, and also the methodology when they are designing, I think the methodology is the heart of any research design, because the pilot testing of the research project, how much sample size they should get. By, for example, if most of the projects in PharmD are of 1 year, or in B-Pharm it is of around 6 months to 12 months. So the researchers should understand, especially when they are involving human research, that human research are not that easy, because there are ethical approvals, protocol making will take time, ethical approval will take time. And by the time they get all these approvals in place, and also CTRI nowadays is becoming mandatory. So a lot of critical time is lost, and the students will panic. They will not be able to recruit sufficient number of sample size, which they were planning. And because of that, which you are telling, the students will not get their sufficient p-values for the research, and they will finally get and manipulate the data to get the positive results. So I think if they plan early, and if the guides and the academicians supports their students, like, so I think these type of bias can be reduced, especially publication bias can be reduced at a very, very early stage. Yeah. And I think the trainings and skill development among the researchers and awareness about, like, nowadays we have plagiarism checkers are in place, and we have reference managers in place. So I think all these tools, the updated tools will help us in helping the students getting their results published. One of the aspect, as you are telling, most of the students, when they are reading articles, they are attracted towards positive results. So they will be very, very afraid of getting negative. But the students should understand, especially Indian students, because most of the top-notch journals which are publishing results, if you talk about some 20 years back, 10 years back, most of these publications are coming from Western world. But we have to understand that India as a country, we have a separate different population, which is totally different from the West. We have a different genetic makeup, we have different social habits, we have different lifestyle. So our results, it's not mandatory that it should match with the Western results. So in case if we are getting negative, that is fine with us. But only thing is, we should accept those findings. And I think many of the top-notch journals nowadays are also asking for the raw data. So in order to better understand and better visualize the whole scenario in place, and whether the right picture has been projected in the research papers or not. So I think in all the academic institute, we should promote the exact findings. And I think that can start with the thesis publications. Like when the students are publishing thesis in their academic levels, I think we can start promoting students to publish even the negative findings.
Yes.
So I think things will change.
Yes, sir.
But I think the retraction of paper is a very serious thing which are happening nowadays because of the research bias. And I think the students should be made aware what are the consequences of retractions and publishing like wrong results. So it can be very, very dangerous for the kids. So they should understand at an early stage of their careers. So I think negative results is all fine in the upcoming days.
Sir, yes, definitely. And if you remember in 2017-18, there were 20 papers retracted from Nature, and those all 20 papers were some of one or two of the top-notch Indian institutions.
Yes, exactly.
Institutions, just because of one picture plagiarization and just because of some errors in the data.
Yes, exactly,
From the
Dr.
nature,
Rajit.
from the nature we have retracted.
Yes, exactly. Because I think there are now guidelines in place. I think the ghost authorships and like gift authorships are actually increasing nowadays. I think because of the competitions and the pressure out there among the academic institute and among the research institutes.
Absolutely.
So, but I think as a researcher, we have to understand that we should not rush towards publication and we should not compete for just publication. I think India as a country, we should compete for quality publication instead of numbers. if we can all focus on quality publication, I think the things can improve a lot. So that's what my input is. So
yes, sir. Thank you very much. So anyone wants to put something on, want to add something, or want to share the experience, or any questions from audience, guys, you can write or you can directly talk to us, as this forum is open for everyone. Okay. Okay. If not, then we have conveyed the message was exactly we wanted to. And sir has also, additionally put the comments on it, and he has rightly said that we should avoid these things at different stages. So yes, thank you very much, guys. And we will immediately, start the sessions after 10 minutes only. So let's take a rest for 10 minutes. I hope you guys had your lunch. And if not, then take it aram se. You can have at your home, because you don't have to speak. But let's start the session at 2 p.m. So our next participants, next our speakers and the panelists should not wait, right, guys? So let's start in 10 minutes. Take care, guys. Let's take a rest for 10 minutes. Thank you. Hi. So, okay, by the time can we take one presentation was left for last 3 days. See here.
Hello.
Yeah, Anya.
Yes, sir. I'm joining from my laptop first.
No problem. Please join.
Yes, sir.
This meeting is being recorded.
Please present, Anya. No problem. Restrict your presentation to 7 minutes. Yeah, please go ahead. Share your screen and go ahead.
Yes, sir. Actually, I'm not able to share my screen.
Once go back and again share. It might work.
Yes, ma'am.
Yes, ma'am.
Actually, I'm not able to present my presentation here. So, can anyone else display that?
Anya, you have all the permissions to present from your side.
Yes, sir. But I don't know why I'm not able to. I'm so
Try.
sorry for taking lots of time. Sir, actually, screen share option is not coming in my Zoom meeting. So can I orally present my presentation? Hello?
Anya, wait for some time. Then I'll tell you how to do that after this, because we are going to start the sessions in 5 minutes.
Okay. Okay, sir. Okay.
Great.
Wi-Fi password கொடுத்தாங்களா? தெரியுதுங்க. நமக்கு. Hello.
Hello, sir. I think sir has joined. Okay.
Yeah, Dr. Ajit, good afternoon.
Hi, Dr. Krishna. Hi. Hi, good afternoon. Okay, so we'll just inform our participants to start and to join once again, because we have just finished our morning session just 5 minutes back.
Yes, I can understand. Yeah. We can wait for a few minutes, no issue.
No problem. 2-3 minutes will wait, sir.
Yeah, sure.
Actually, some sessions were quite amazing, so we couldn't stop our speakers, and
Yes,
really
yes. Good. No, good that they are all actively participating even on Sunday and Saturday.
Absolutely. Hi, Murugan sir. Namaste.
Namaste.
See you tomorrow physically.
Sir, good afternoon, sir.
Sure. So, how are you?
I'm good, sir. I'm good. Perfectly fine.
Okay.
Murugan sir, good afternoon.
Good afternoon, sir. So the session is going to start probably after a few minutes.
Just 2-3 minutes, sir. Give us 2-3 minutes. I'm just telling my team to, remind everyone so they can join.
Sure. Yeah, sure. ಇದೊಂದು ಪಾಸ್ವರ್ಡ್ ಇದು ಮೀಟಿಂಗ್ ಐಡಿ ಇದು ಪಾಸ್ವರ್ಡ್ ಇದು ಫುಲ್ 895 ಫುಲ್ 5
Dr. Sakina, I think we should not wait much. Let's start.
Sure, we can start now. So, I offer a warm welcome to everyone after morning session. Come and start our afternoon session. So, without waiting and doing any delay, let's start. Everyone, all the participants and all the panel and all the speakers, everyone to MCPCON conference. So let's move towards our first session. Topic of the session is signal detection. So for this session, I am honorable to invite Krishna Undela. Dr. Krishna Undela has done M.Pharma and has done PhD. And then PhD National Institute of Pharmaceutical Education and Research, Guwahati. And our panel are Dr. V. Murugan, Professor Academic Director, Nargan College of Pharmacy, Bengaluru, and Dr. Karthikeyan, Professor and HOD Pharmacy Practice School, Pharmaceutical Sciences, Chennai. So let me give an introduction about Dr. Krishna Undela, working as Associate Professor, Department of Pharmacy Practice, National Institute of Pharmaceutical Education.
Your voice is not clear.
Executive Fellowship from the Department of Education, Australian Government, to undertake 3 months of training on advanced pharmacoepidemiology and quality use of medicines at the University of South Australia, Adelaide. He received travel grant from the International Society for Pharmacoepidemiology to attend ISPE conferences 9 times and from ISPOR 3 times. In the last 10 years, he delivered over 150 lectures at international conferences. He is currently a member of many societies in pharmacoepidemiology and pharmacovigilance. Dr. Krishna is a coordinator of the Adverse Reaction Monitoring Center, Atal Incubation Center and BioNest at NICAR, Guwahati. Dear sir, please start your presentation and enlighten us with your knowledge. You can go ahead, sir. Thank you so much.
Thank you, Dr. Sakina, for the kind introduction. And before going to the presentation, thanks to the organizers for giving me the opportunity to speak during this 3 days conference, a very important conference on different aspects of pharmacy. And thanks to the moderators for joining for the session, and hopefully we can have a very good interaction after the session. So without wasting much time, maybe I can share my screen so that we can go for the presentation. Can you please confirm you can able to see?
Yes, sir. If the screen is visible.
Yeah, thank you. So the topic we are going to be discussed now in the afternoon session is a signal detection in pharmacovigilance. One of the very important topic as well as advanced topic, I can say, under the pharmacovigilance. And whatever we do under the pharmacovigilance, finally, we are going to be end up with the signal detection so that we can try to understand the association between any drug and the reaction. So that's what we are going to be do, especially the signal detection. And where we are going to be follow some quantitative and qualitative methods for identifying or detecting the signals between a drug and the reaction. And on a regular basis, we used to see so many signals every day, every time, like, we can have a traffic signals when we are going on the road, and when you have a no parking signal, and especially in Australia, you can see a signal called, there is and maybe you can find some animals on the road. So be slow when you are going on the road. So this kind of signals we used to see day in and day out every time, especially the mobile signals we are going to be get it just to going to alert us that we have a message, right? Either you have a message or you have maybe a mail or maybe any kind of signal you are going to be get it in the phone. That means it's going to be alert you so that you can able to check your mobile and then see what kind of message you got it. So similarly in pharmacovigilance also we have a signals. It is going to be give an alert on a drug and the reaction. That's what we are going to be get it in the pharmacovigilance, the signal. And there are different definitions for signal detection, which are given by the different agencies. So the first one, if you see the one which is given by the WHO, World Health Organization, and what they mention about the signal in pharmacovigilance is, it is a reported information on a possible causal relationship between an adverse event and the drug, and the relationship is being unknown or incompletely documented previously. So if you find something new information, a new possible causal association between a drug and reaction, and if you want to try to identify further either the real association is there or not, we can call it as a signal detection. And usually we require more than one report to identify the signals, and depending upon the seriousness of the event and as well as the quality of information available with us on a particular drug and reaction. And usually people feels that the signal can be a new thing, a new one we need to identify. The signal can be of anything. It may be a labeled or unlabeled reaction. It may be an expected or unexpected reaction. Anything can be a signal. In the sense, by identifying the signal, you're going to get more information about the drug and reaction. Thereby, you can take a specific decision on that particular reaction. That's what we are going to be do, especially in the signal detection. So coming to the definition, which is given by the EMA, European Medicines Agency, they have given their own definition for the signal in pharmacovigilance. So what they say is the safety signal is an information on a new or known adverse event. As I mentioned, the signal can be of a new reaction. It can be of a known adverse reaction that may be caused by a medicine and requires further investigation. So it is not that we identify the signal and that's the end of the story. We are going to be taking a decision, but it is going to be an initial stage of signal detection where we need to have further understanding, further investigation. Then only we can take the decision. So safety signals can be detected from a wide range of sources, such as spontaneous reports, clinical studies, as well as a scientific literature. That's what EMA mentioned. So coming to the definition of UMC, one more important organization, internationally renowned organization for pharmacovigilance, Uppsala Monitoring Center, and they define the signal as it is one of the essentially a hypothesis of a risk with a medicine with the data and arguments that support it, which are derived from a data from one or more or many possible sources. So that's what the complicated definition given by UMC for the signal detection. But overall, whatever the definition it may be from WHO, it may be from EMA, it may be from UMC, whatever the definition, some of the important components we need to understand in the definition of signal detection. The meaning is the signal means it is an information coming from different sources, and it's going to be identify the potential harm for a medicine caused by a medicine, and that requires further evaluation. So these are some of the important terminologies. Maybe we can take it up from all these definitions and try to understand what exactly the meaning of the signal detection. So it's basically the information which is coming from different sources on a potential harm caused by a medicine, and that requires the further evaluation and investigation. And the signal detection is not a single step process. We are going to have various components in the process of signal detection. So basically, we are going to divide them into three different components. The first one is signal generation, where we are going to have two important steps, signal detection and signal selection. And the second component is a strengthening. Once you generate the signal, we need to strengthen the signal identified. And under that, we have two important steps, strengthening and assessment. And the final component is going to be the follow-up. And under the follow-up, we are going to have the action and follow-up. So these are the three important components we are going to have under the signal detection process. And these are the 6 important steps we are going to have under the signal detection process. So we are going to discuss one by one. And what will happen exactly here is over a period of time, when you're going on a time, the knowledge we are having on that particular adverse event, we are going to have more, and it's going to be finally creates an S-shaped curve. That's what we are going to have basically under the signal detection. Over a period of time, we are going to get more and more understanding on that particular reaction. And based on that understanding, we are going to take up the final decision. So there are some favoring situation. It is not that every drug and every reaction can be a signal. And there are some favoring situation we need to take into the consideration. So that for those pair, in the sense the drug and reaction, we can go for further evaluation and identify the signals. So what are those favoring situations where we can try to identify the signals means where there is a high frequency of exposure to a drug. So whenever any drug releases into the market, if you expect that the drug is going to be taken by a good number of people in India or maybe in the world, so we need to go for the signal detection basically for that medication. And if any adverse effect with one or more following features where the adverse reaction is having the high frequency where number of people are getting the reaction, and suggestive time dose relationship where exactly we're going to get the information on, the reaction is occurring within a short period of after giving the medication, and where there's a possible pharmacopathological mechanism behind the adverse effect. So those are some of the favorite situations in terms of the adverse effect. And if you also identify some of the cluster or clinical events or maybe the syndrome with one or more of the following, wherever you are having the very low natural frequency of a particular reaction or maybe the drug, and the characteristics of unusual signs and symptoms, if you're getting some unusual signs and symptoms, you may suspect that may be because of medication, and we can go for further evaluation and then see, is it a signal or not? And occurring in group of similar patients. For example, all pediatrics are getting a kind of reaction after taking certain medication. So that's going to be a favoring situation where we can try to understand the signals. and frequently known to be drug-induced. For example, we know that some of the dermatological reactions like anaphylaxis, agranulocytosis, Stevens-Johnson syndrome, toxic abdominal necrosis. So these are some of the reactions where they're going to be caused mainly by the medications. And whenever those reactions occurs, we can suspect that that may be because of medication only, and then we can go for the further evaluation. And this is some of the important favoring situation we need to keep in mind before going for further process of the signal detection. And coming to the methods of signal detection, like we have 4 important quantitative methods. As I mentioned initially, there are 2 types of method we are going to have under the signal detection. One is quantitative, where we are going to be play with the numbers, and another one is going to be the qualitative, where we are going to be check each and every report and try to understand the qualitative aspects of the reaction. Okay, so coming to the quantitative, there are various methods are available for signal detection, and among all those, there are 4 important methods we are going to be discussed today. And in that, 2 methods mainly comes under the disproportionality analysis or measures, and under that we have one called a proportional reporting ratio. We can also call it as PRR along with a chi-square value. And another one we are having is a reporting odds ratio, ROR, along with a confidence interval, 95% confidence interval. So these 2 measures, basically, we are going to be keep under the disproportionality measures. One of the important quantitative signal detection analysis we are going to be performed, maybe under any kind of pharmacovigilance databases. So apart from that, we have 2 more advanced methods. We are going to be keep them under the Bayesian measures. And in that, the first one we have is a information component, IC, that is mainly using by the UMC, Uppsala Monitoring Center, for identifying the signals. And that comes under the Bayesian confidence propagation neural network method. So the method what we are using here for the information component is a BCP-NN. And there is one more important method we are going to have under the Bayesian measures is a empirical Bayesian geometric mean, EBGM, we can call it as in a short form. And that is the one commonly using by the US FDA for identifying the signals. And the method what they're following is a Dumont's multi-item gamma poison shrinker method, MGPS method. That is a method they're following basically under the EBGM. So this slide is very important for you. Maybe you can take a snapshot of it. So the 4 important signal detection methods, quantitative methods we are using for identify the signals are PRR, proportional reporting ratio, ROR, reporting odds ratio. And under the Bayesian, we have 2 important methods, information component commonly using by the UMC, Uppsala Monitoring Center. And the other one is EBGM, empirical Bayesian geometric mean, that is commonly using by US FDA for identifying the signals. So we can take up this example. It will be very interesting. You can easily understand how exactly we are going to be calculate the signals in pharmacovigilance by using a very simple 2x2 table. It's not complicated, a simple 2x2 table. By using that, by using some formulas, we can try to identify the signals between a drug and reaction. So the example I have taken here, it is to identify the signal between acetagliptin and anti-diabetic medication, and the reaction is going to be the pancreatitis. Either there is a signal identified between acetagliptin and pancreatitis. So what exactly we need to do here is we need to search in a database. For example, UMC, Uppsala Monitoring Center, they're having a huge database of reactions which are coming from all over the world. And currently they're having more than 33 million adverse drug reaction in their database called VGBase. Hope you hear the VGBase. And that is having more than 33 million adverse drug reactions. So by searching in the database, we are going to be get these numbers. By using these numbers, we are going to be identify the signals. So how exactly we are going to be perform that is, for example, we are having here 23 million odd reactions overall in a database. Okay. Imagine that we have 23 million reactions in a database. And out of those 23 million, we got to know that 30,278 are on sitagliptin. Okay. So out of this many reactions in a database, 30,278 are on sitagliptin. And out of this 30,278 for sitagliptin, 2,252 are on pancreatitis. So there are so many reactions reported for sitagliptin. Out of all this, 2,252 are basically on pancreatitis reaction. Okay, and remaining all for the all other reactions for sitagliptin. And similarly, we are going to be have out of 23 million, as we know, 30,000 are for the sitagliptin, and remaining all reactions what we are having here. So this one minus the 30,000, if you do, we are going to get this number. So these are all the reactions for all other medications. In the sense, except sitagliptin, these many reactions are reported for all other medications in the database. Okay. And out of all these reactions, 46,923 are on pancreatitis, and remaining all are on other reactions. Okay. So out of all these reactions for all other medications, 46,923 are on pancreatitis, and remaining all are for all other reactions. Okay. And then we are going to be give the coding basically here. So this one is going to be A, B, and this is going to be A plus B. Total number of reactions for drug of interest, that is sitagliptin, and C, D, and this one is C plus D. These are the total number of reactions for all other medications. And this one is going to be A plus C, a combination of these two. That's the total number of pancreatitis reactions. And we are going to have all other reactions, B and D. Combination, we are going to have the B and D. Okay, B plus D. And this is the total number of reactions available in the database. And we are going to give the coding of A plus B plus C plus D. Okay. So if you know the pharmacoepidemiology and the calculation of relative risk and odds ratio, in a similar fashion, we are calculating the odds ratio relative risk in pharmacoepidemiology. In a similar fashion, we are calculating the signals in a pharmacovigilance. So just a simple 2x2 table, and we are giving the coding. And from this, we are going to be identify the signals by using two important methods. One is PRR, as I mentioned, proportional reporting ratio. And very simple formula we are having here for calculating the PRR is A by A plus B divided by C by C plus D. Hope you know the formula. It is the same formula as a relative risk in pharmacoepidemiology. And the formula is same for PRR in pharmacovigilance. Okay, the formula is same. Only the terminology we are using something different here. Instead of relative risk, we are mentioning it as a proportional reporting ratio for pharmacovigilance for identifying the signals. And other one we are having is a ROR. ROR is a reporting odds ratio. And here once again we have a very simple formula: A by B divided by C by D. We can also write it like AD by BC. A cross multiplication, we are going to have AD by BC. And it's once again similar formula of the odds ratio, A by B divided by C by D. In pharmacoepidemiology, we can call it as odds ratio. In pharmacovigilance, we can call it as a reporting odds ratio. Okay. So by using this 2x2 table and these two simple formulas, we are going to be identify the signals between a drug and reaction. And if you use all these numbers and calculate the PRR, for example, here we got 37.35. Okay. So the PRR value, what we got here is a 37.35. And along with that, we are going to get the chi-square value if you use any statistical software. And by keeping all these numbers, along with the PRR, you are also going to get the chi-square value. And this is very important for us to understand the signal. And similarly, if you calculate the ROR by using the formula A by B divided by C by D, we got the ROR value of 40.28. And along with that, we're having the 94% confidence interval. And this one is a lower limit, and this one is a upper limit. Okay. So these are the very two simple calculations we perform for signal detection: proportional reporting ratio and reporting odds ratio. Okay. And maybe you can note down these values so that you can try to understand when we can say the signal is identified between the drug and reaction. So based on these numbers, can we say or not the signal is identified between the drug and reaction? Okay. Just remember these values: 37 here and 40 here. Okay. For ROR. And the other value we are having is a IC. As I mentioned, IC is the information component commonly using by the UMC, Uppsala Monitoring Center, for identifying the signals. And this is a platform of Visualize. If someone is not seen till now, the Visualize platform, where we are going to be perform all the calculations by using the data available in the Vigibase. And it is only accessed by the UMC or maybe the National Centers of Pharmacovigilance Programs. So what we need to do simply here is we can enter the name of the medication, sitagliptin, and the name of the reaction, pancreatitis. And for these two combination of reaction and the drug, these are the number of reactions available in the database, and N observed, N expected. And for that, they're going to calculate the IC value. So IC value here, it is a 5.1. And for that, we have something called IC025. So IC025 is a lower limit of the IC value. lower limit of IC value. And based on this, we are going to be understand the signals. Based on the IC025, we are going to be understand the signals. And remember the IC025 value here, that is 5.1. Okay, whatever we got IC value here, the IC025 value here in UMC database, it is a 5.1 for sitagliptin and pancreatitis. Okay, so now these are the very standard statements we need to remember. When can we say the signal is identified between the drug and reaction? Based on all these numbers, what are the numbers we have for PRR, proportional reporting ratio, ROR, reporting odds ratio, and information component? Now we have the values. Based on these values, can we say that the signal is identified between sitagliptin and pancreatitis? Okay. So when can we say that? Means there are standard statements. In case of PRR, if you're taking the PRR to identify the signals, when we can say signal is identified means a signal is detected if the count of co-occurrences more than or equal to 3. The count of co-occurrences means number of reactions on this drug and reaction. Number of reactions on the pair, there's a drug and reaction, that should be at least more than 3. So I'll show you quickly. This is a number. So this is a number. It should be more than 3. Then only we can say the signal is identified between the drug and reaction. That's one of the points we need to consider for PRR calculation or assessment. Okay. So the count of co-occurrences should be more than or equal to 3. And the PRR value should be more than or equal to 2 with an associated chi-square value more than or equal to 4. So whenever all the 3 satisfies only, we can say the signal is identified between the drug and reaction. Okay. So in this case of sitagliptin pancreatitis, the reactions are more than 3. It is around 2000 reactions are there. And the PRR, of course, more than 2. I think we got around 37. And chi-square values in thousands. So it should be more than 4. And now we are having in thousands. So we can clearly say that the signal identified between the sitagliptin and pancreatitis by using the PRR, proportional reporting ratio. And the second one is ROR, that is a reporting odds ratio. And we can identify the signal by using this ROR in case the lower bound of the 95% confidence interval, two-sided confidence interval of ROR exceeds 1. In the sense, so this is the ROR value. This is the ROR value, 40.28. And for that, we have a confidence interval. And this is the lower limit, and this is the upper limit. And this lower limit value should be more than 1. We can say that signal is identified between the drug and reaction. It should be more than 1. Here in this case, we have 38. So whenever it is going much far from the cutoff value 1, we can say that much strong signal identified between the drug and reaction. So the 38 is very, very far from 1. In the sense, we can say that there is a strong signal identified between the sitagliptin and pancreatitis by using the ROR, reporting odds ratio. So thus we can get it from the ROR statement. And coming to the IC, information component, very similarly to ROR, here also, we can identify the signal whenever the lower bound of the 95% two-sided confidence interval of IC, that is IC025, exceeds 0. In this case, the standard is a 0. So whenever the IC025 value is more than 0, in the sense a positive value, more than 0 means positive value, we can say that the signal is identified between the drug and reaction. In the previous case, in the previous slide, we saw that the IC025 value is 5.1. It is much higher than 0 cutoff value. So we can say once again, there's a strong signal identified between the sitagliptin and pancreatitis based on the data available in the VG base. Okay. So that's what we can get it from IC. We need to check here the IC025 value, and that should be more than 0. We can say that signal identified between the drug and reaction. And finally, we have one more EBCM. I did not show the calculation here because it is not that easy to show the calculation because there are so many parameters that will be taken into the consideration for calculating this EBCM, and especially using by the US FDA. And but you can remember the standard of EBCM as well. When we can say signal by using the EBM means the signal is detected when the lower one-sided confidence interval limit of EBCM exceeds 2. So here the limit is 2 for EBCM. Whenever the lower confidence interval of EBGM exceeds more than or equal to 2, we can say that the signal identified between the drug and reaction. So this slide is very important. These are some of the standard statements we need to understand or we need to follow to identify the signals by using any one of these methods. So basically, it's a quantitative where we are using all the numbers, playing with the numbers, and calculate the PRR, ROR, IC, EBCM, all these things, and then try to identify either the signal identified between the drug and reaction. Okay, so this is one of our recent publication published in International Journal of Clinical Pharmacy, where we try to identify the signals for SGLT2 inhibitors, the novel class of anti-diabetic medications, and causing the pancreatitis. And what we found here is all the three novel SGLT2 inhibitors, canagliflozin, dapagliflozin, empagliflozin, are found to have a signal for the pancreatitis. They're found to have causing the pancreatitis significantly based on all the calculations we performed. That's a PRR and lower bound of ROR along with the IC value. So we also did some adjustment here by adjusting the confounding factors. We did that. And finally, we can see that especially by taking the IC025 value, you can see here 1.3 for canagliflozin, 1.2 for dapagliflozin, and 1.3 for empagliflozin. And all the three are more than the cutoff value for IC025. So what's the cutoff value for IC025? It is 0. So 0 is a cutoff value for IC025. And anything more than 0, we can say signal identified between the drug and reaction. And here it is more than 1, 1.3, 1.2, and 1.3 for canagliflozin, dapagliflozin, and empagliflozin. Even by seeing the PRR values as well as a lower bound of the ROR values, we can understand the signal identified between the SGLT2 inhibitors and pancreatitis. And we need to be careful when we are giving these medications to the patients, especially who are having the risk of getting the pancreatitis. Okay, so that's what the understanding we can have it from the basic signal detection calculation. So after doing this calculation by using all these numbers, we need to have some positive factors and negative factors. We need to take them into consideration. So that gives us information that can we go further by having the signals from the quantitative. When you go for quantitative signal detection analysis in a database, we are going to get thousands of signals you are going to be getting. But all the 1,000, we cannot take it further and go for the further assessment. From that, we need to come out with some selective signals. So how we are going to be select signals from this quantitative means there will be some positive factors, there will be some negative factors. Okay. So the positive factors means, for example, the ADR may be unknown previously. That may be one of the positive factor. We can take it to the consideration and go for the further assessment. And the strong statistical connection, like sitagliptin, pancreatitis, we saw that PRR value, ROR values, there 40, 38, 37 values are there. That means we can understand that there is a positive, strong signal identified between the sitagliptin and pancreatitis. So in that case, we can go further and do the further assessment. And reaction can be unexpected, and the reaction may be unlabeled reaction, till now not identified, not mentioned in the prescribed information leaflet or patient information leaflet. And it may be the specific characteristic, and it's going to be a definitive, objective one. The typical drug-related events, wherever the reaction is going to be expected for the medication, and the low background frequency is available. And if you find out any serious adverse drug reaction, and it is having the high potential relevance, in all these cases, whenever you find any signal, it is having any one of these points, the positive points, we can take it further and go for further assessment. As I mentioned, we need to go for the qualitative assessment. Then you can go for the decision making. Okay, these are some of the positive aspects we need to consider before going further for the signals what we identified based on the quantitative methods. And the signal strengthening, after identifying the signals, after selecting some signals, imagine that out of 1,000, we selected some 10 signals now based on all these positive factors. And now these 10 signals, we need to go and strengthen to get more and more information. We need to go and strengthen. And what exactly we're going to be do to strengthen these signals means individual case assessment. Individually, we're going to be assess all the cases pertaining to these reactions, and country comparison. If you identify any signal now in India, the first thing we need to do is either the same thing is identified in any other country or not, especially in US or maybe in UK or maybe in Australia. We need to understand that the same is identified previously in these countries or not. That's what is country comparison. And targeted comparisons, in the sense we can go on target. For example, if you identify for the sitagliptin, what about for other medications, other anti-diabetic medications, either it is identified or not? We can go for the targeted comparisons. And then best case, worst case scenario, like in case if it is identified based on the signals, what will be happening in the best case, what will be happening in the worst case. So we need to understand what's going to be happening in the future in the best case and worst case scenario. That's what the assessment we need to perform. And finally, we can go and conduct some more studies, because pharmacovigilance data alone may not be sufficient enough to understand or to take any decision. Okay. So for that reason, we need to go and do further studies, especially by using some of the pharmacoepidemiology methods. We need to go and do some studies. Then we can get more and more information. Then we can try to take the decision possible. So for that reason, we can go and conduct, for example, nested case control studies or case cohort studies. Any other studies we can go and conduct further to get more information. Then we can go for the decision making. So these are the things we usually are going to be do under the signal strengthening. For example, the presence of similar connection, like between the reactions. We can go and search for the other databases what we are having and other sources what we are having. Especially we can go and search a very good database or very good website from EMA, European Medicine Agency. That's a PRAC. PRAC is a very important one. That is a signal risk assessment committee, pharmacovigilance risk assessment committee we are going to be have under the EMA, European Medicine Agency. They're going to be published the minutes after every meeting, and they're going to be identify the signals from European database called Vigilance. And that's one of the important sources maybe where you can go and search to see that either these kind of signals are identified elsewhere, especially in US or Europe, or not. Then we'll get more and more information on the reaction. And the additional observation, we can go and search for the literature in any database. And spontaneous reports, we can go and read all the spontaneous reports what we're having. Registration files and recent all this information given by the manufacturing companies, like periodic safety update reports, PSUR, and periodic benefit risk evaluation report we are going to have, and drug safety utilization report we are going to have. And then we are going to have a risk minimization plan. So all these reports are submitted basically by the marketing authorization holders to the regulatory authorities. If you go and read those documents, you may get more information on that particular reaction and medication. So we need to understand the information available from all these documents for further strengthening the signal. And also we can get some experimental data. We can go and do some experiments, or maybe if already available, we can take some information about the pharmacological and immunological mechanism behind the reaction. So we can get some information basically from the experimental studies. So these are all the things combinedly can gives us more information on a particular drug and reaction. Thereby we can go and strengthen the particular signal. And the signal assessment, once you strengthen the signal, we are going to be assess a signal once again by using the quantitative and qualitative methods. And another quantitative, this time we can have this relative risk odds ratio, especially by performing some pharmaco-AP studies. And also we can have some exposure versus reactions. So these are all the quantitative information we can get it once again from the other study designs. And also we can go and assess qualitatively, like consistency of data, like how it is reported. For example, the reaction reported in 2014, and after that there are no reactions in 2015, 16, 17. Only in 18 there are few more reactions. That means the data is not consistent. The people are not reporting this reaction consistently, so that may not give enough information for us to identify the signals. And exposure response relationship, we need to understand more about the reaction, like site of reaction, timing of reaction, dose response relationship. That means when the dose is increasing, the reaction is increasing or not, that we need to get it. And reversibility, in the sense we have a de-challenge, re-challenge. So we need to get that information for the qualitative assessment. And also we can understand the biological plausibility of the hypothesis by having more information on the pharmacological mechanism as well as the pathological mechanisms. And these are all the things we are going to understand basically from the qualitative analysis. And we are going to get some more information from the experimental studies, as I mentioned. And from there, we can get the information on re-challenge. We can get some information on the laboratory test for that particular reaction. And maybe we can also get some drug levels by performing some blood test analysis. We can try to understand the drug levels in the blood. And analogy, like if you have any previous experience with the related medications. For example, sitagliptin we are evaluating here. and you are already having the information that teneligliptin is causing the pancreatitis. That means that's the existing information already available from the same class of medication. From the same class, there is another medication causing this reaction, means we can suspect that even sitagliptin can cause this reaction because the class is same, the structure is going to be almost same. So we can expect some reaction, similar reaction for this medication as well from the previous experience of a medication available in the same class. And one more example I can give you, like rosiglitazone, pioglitazone, because rosiglitazone already banned from the market because of the cardiovascular issues. Whenever pioglitazone is coming into the market, we can expect that this may also can cause some reactions related to the cardiovascular issues, right? We can expect something based on the previous experience, and that's what comes under the analogy. And even known to be frequently drug-induced, especially like dermatological reactions, what I mentioned, SJS, toxic epidermolysis, any other reactions which are commonly caused by the medications. And nature and quality of data, we are going to have the various characteristics from each and every report. We are going to have the objectivity, accuracy, validity of documentation. And also very important thing is we are going to be assess the causality. So that's the reason the causality assessment of each and every area is very much important, because that's going to be gives us more information in the signal assessment, signal detection and signal assessment. So once you do all these things, what we need to do is we need to take an action on that particular reaction. If you feel that that's very important, sitagliptin causing pancreatitis, the signal is very strengthened. Okay, we assessed all the qualitative parameters. So finally, we need to take some action on that, and we need to even follow up and then try to see that how it is effective. So for that reason, the action will be taken by the different levels. For example, the regulatory action can be taken by the regulatory authorities. So what maximum they can do whenever they identify any reaction is, for example, changing the data sheet. What is the data available currently on that medication? They can change that by including this reaction. And the label revision and monitored use, what are the label currently given for that medication can be changed. And also sometimes they can restrict the usage of that medication for the certain group of people. So they can do all those things. And finally, if they feel that the reaction is very important, for example, like thalidomide, it caused pocomylia, a very important reaction. In that case, they need to go for the withdrawal or restriction usage of the medication. Okay. So these are all some of the regulatory actions can be taken by the regulatory authorities after identifying the signals between a drug and reaction. So that is not sufficient enough. We need to even give some awareness to the public, especially to the healthcare professionals. And that can be done by communication. We can communicate the signal, we can communicate the information to the regulatory authorities, to the healthcare professionals, to the public, so that they can be aware that this drug can cause a reaction. Okay. So for example, the signal can be communicated to the national centers like PVPA in India. So whenever UMC identify any signal, they can communicate to the PVPA so that PVPA can be aware of it and take some necessary action. And they can communicate to the company, the respective company who is manufacturing that medication. They can communicate to the company so that they can take some action related to that reaction. And any other institutes, or maybe to the public, or maybe to the healthcare professionals. So they need to communicate the information to all the stakeholders so that action can be taken and it can be prevented in the future occurrence. And the publications can be done in the form of newsletter, in the form of article. We can publish the articles in the journals, and maybe like PVPA, UMC, they're coming out with the newsletters once in every 3 months or 4 months. With that, they're going to be spread the information on the signals. And that's not the end of the story. We need to go and conduct the further studies, maybe pharmacoepi studies, or maybe the randomized control trials. We can go and conduct further so that we can get more and more information on that particular reaction. So till now, what we did is a hypothesis generation. And by conducting few more studies, further studies, we can go for the hypothesis testing, and we can also conduct some follow-up studies. And this is what I mentioned, signal testing, like we are going to have the hypothesis generation from the spontaneous reports, and hypothesis testing by conducting more and more studies by using different pharmacovigilance methods. Like we have different methods under pharmacovigilance, for example, spontaneous reporting, targeted reporting, cohort-event monitoring. So these are the methods we are going to use commonly under the pharmacovigilance. We can go and conduct some studies by using any one of these methods so that we are going to get more and more information. And that comes under the hypothesis testing. Okay. And also we can go and conduct some pharmacoepi studies, or maybe the randomized control trials, or maybe the pharmacological or pathological studies. And all this mainly comes under the hypothesis testing. So what we are doing from the pharmacovigilance data, by analyzing the data available in a database, what exactly we are doing is, it is only hypothesis generation. We are generating the hypothesis. And to test the hypothesis, we need to go and conduct few more studies. So what basically the difference here it is, from the pharmacovigilance data, we are going to have only the numerator, in the sense, we are going to have the data whenever the patient is getting the reaction. Only that data is available in the pharmacovigilance databases. There may be so many other people who are taking the sitagliptin, but not got any pancreatitis. And the information about these patients who did not get the event may not be available in the pharmacovigilance database, because we are going to report the reaction only when patient gets a reaction. So they're going to have all that information who got the reaction. But what about, there are lakhs of people who are taking the medication but did not get the reaction? That information basically may not be available in the pharmacovigilance. To get that information, so that we can try to identify the incidence proportion, incidence rate, the percentage of occurrence, all these things we can understand basically from the, by conducting these studies. And that's why we can keep these studies under the hypothesis testing. Okay. And this is the procedure basically following by the UMC, Uppsala Monitoring Center, for identifying the signals. And there are 4 important steps that we're going to have. The first one is a first pass statistical screening. That's analysis they are doing. They're going to be prioritize the medicine adverse effect combinations by their strength of evidence. And what are the signals identified from this quantitative assessment? Now onwards, they'll go for the manual assessment. So initial manual assessment will be there, so that they're going to identify the signals which requires further understanding. And then for this reaction, they'll go and do the in-depth manual assessment, so that they're going to come to the conclusion, what are the signals which are important, where we need to go for the decision taking. Okay. And whenever they identify any signals, the final thing is going to be happen is a signal communication. They need to communicate the signal to the various stakeholders. And that's what they're going to do by distributing information to the national pharmacovigilance centers as well as to the public. Okay. So these are the 4 important steps the UMC is following, and many national pharmacovigilance, they're following the same process for identifying the signals among the data what they're having. And all that information from UMC, whenever they identify any signals, if you want to understand the signal between any drug and reaction, we have one website for signal library of the UMC, where we can go and search for the drug, or maybe search for the reaction. You're going to get all the signals identified for that particular medication or reaction. And that information you can get it from this particular web page. And one more important document, or maybe the resource you can follow to get more and more information on the signals. Not only UMC, not only India, not only Australia, throughout the world, any regulatory authority identify any signal between a drug and reaction, it will be published under this WHO Pharmaceuticals newsletter, published by UMC, that is freely available. So once in a quarter, they're going to publish this newsletter, where they're going to be explained throughout the world what are the signals identified between the drug and reaction. You're going to get the complete information. So they're going to give a description and final conclusion, so that we are going to get more and more understanding on the signals between a drug and reaction from this WHO Pharmaceuticals newsletter, a very important resource for understanding the signals. And you may be wondering, like, what exactly in India we are doing. As you know, that PVPI is existing from 2010, and they're collecting reactions from all these 600 odd ADR monitoring centers are there throughout India. They're collecting the reactions every month. And I think currently they're having more than 6 lakhs adverse reactions with them. So what exactly they're doing is they're also going to identify the signals. And after identifying the signals, you know that they are not the regulatory authorities to take any action on the medications. So what they're going to do is, whenever they identify any signals from this signal review panel committee meetings, they're going to have once in 6 months a committee meeting. So during that meeting, what are the signals identified between a drug and reaction, for example, remdesivir and sinus bradycardia, whenever they identify anything, they're going to be communicated to the CDSCO. As you know, CDSCO is a regulatory authority in India to take any action on the medications. So they're going to be communicate all the signals to CDSCO, and CDSCO is having one more expert panel, and they're going to be review that once again, the drug and reaction importance and all those things. And finally, the CDSCO is going to be taken action. For example, in this case, they suggested that CDCO suggested that this reaction for this medication should be included in the PAL, patient information leaflet, because that reaction is not available till now for the remdesivir in the PAL, and CDCO suggested that that need to be included in the PAL. Okay, so like that, whenever PVPI identifies any signals, they're going to be communicated to the CDCO, and finally CDCO responsible for taking any action on that particular signal. Okay, that's what is going to happen exactly in India about the signal detections. So finally, based on all this information, whatever we saw till now about the signal detection, some of the key points, the take-home messages, like the signal means it is an expectation of a reaction which is unexpected. So signal can be something like expect the unexpected. That's what is about the signal. And signal detection is not a single step. As we saw that there are 6 steps are involved basically under the signal detection, including both quantitative as well as qualitative steps, qualitative parameters. And as I mentioned initially, there are different methods are available for identifying the signals, quantitative, qualitative. Another quantitative, we have 4 important methods: PRR, ROR, IC, and EBGM. And whatever the data we require for this analysis, we are going to be getting from the variety of sources. Basically, from a pharmacovigilance database, we are going to get all that information. But apart from that, we can get that information from any other sources to get more information on a drug and reaction. And as I mentioned, there are some automated tools that are available for the signal detection calculation, and we can use them for identifying the signals. And very important point we need to keep in mind is only the quantitative one, what are the numbers I shown you, only that one is not sufficient enough to understand the signals or to strengthen the signals. We need to have both quantitative as well as a qualitative criteria to identify the signals, to take some action on the signals. An approach for emerging markets and generic pharma as well as manufactured products. That means the signal detection, people will be in the imagination that signal detection is only for the new medications. May not be like that. It may be for the medications which are coming into the market, which may be for the generic medications, which may be for the medications available in the market from long time. Even for aspirin, we can identify some signals if you find something new for that medication. Okay, so it can be for any medication, anytime you can identify the signals. Only thing is, it should be something novel. It should be causing some serious reactions. Then we can go for the signal detection assessment. Okay. And if you want to get more understanding on the signal detection, there are so many guidelines that are available, especially you can go for the EMA guidelines. And there are some PIPA guidelines that are available for signal management for the small and medium-sized pharmaceutical companies. You can go for this guidelines so that you are going to get more and more understanding. They have very clearly explained about all these methods by giving some examples. And also I can suggest you, you can go to this UMC website where there's an education and training pages there, where there are several programs that are offering currently, which are free to everyone. You can go and register and attend those courses. You're going to get more understanding, not only on signal detection, but basically on so many other pharmacovigilance aspects. So along with that, you can also get some understanding on signal detection, but you can get more information on overall pharmacovigilance from this freely available education and training material from UMC, Uppsala Montic Center. So that's about the very crisp understanding about signal detection. If you have any questions, maybe we can discuss further. Thank you very much.
Yeah, good afternoon, Krishna and Ale. The presentation was good.
Thank you, sir. Thank you very much.
Yeah, Dr. Karthikeyan also has come. I hope he also agree with my point. Anyway, I know very well you're from JSS Mysore. We are all selling the same book. Now, the session is open for the discussion. The preference goes to the participant. Anyway, now, afterwards, myself and Karthik, we may have some discussion, but now we request the participant to ask questions from the speaker, Mr. Krishna Undale. Please, whoever has doubt, you can go ahead. I hope Dr. Karthikeyan, is it okay?
Yes, sir. Yes, sir. Yes, sir.
Yeah.
So
Yeah,
one question was received in the chat box itself, I think.
Yeah.
So first
Signal
we can,
detection for
yeah.
medical device as
Dr. V.
material
Murugan,
vigilance, same
I
as
invite
PV
Dr.
visit
V.
flow. But
Yeah, 1 second, madam. Yeah, please go ahead, madam.
yeah, data not at WHO visit flow software used in material vigilance program of India. So the question here is, there are two different things. As you know that there's something called PVPI, there is something called MVPI. And the UMC, WHO UMC is currently responsible only for PVPI, only for pharmacovigilance, not yet for material vigilance. That's why you may not able to find any material vigilance related information in the UMC WHO. But that information may be available with the MEPI India. Once again, IPC, Indian Pharmacopoeia Commission, is a NCC for that MEPI. Maybe from there you can get more information on this MEPI. That information may not be available in the BG flow.
That's good. So any other questions from the audience? Kindly.
Good afternoon, sir.
Good afternoon.
Sir, I have a question. I'm a
Yeah,
family
please.
intern from Ramaya University, Bangalore, sir.
Yeah, please.
So I had also presented based on FAIRS only, sir, my presentation, oral presentation.
Okay.
So I
Okay.
had presented twice, sir. So the most commonly the question they ask me will be, this is basically a database work. So what novel do you find will be their question from their side? So it feels like it is just a database work, data that is readily being available. So what will be your opinion on this, sir?
Exactly. That question is valid, but only answer we can have is, even though it's a database related, okay, it's a openly available FERS data, openly available in the public, in a public database. But the novelty here it is, we are identifying the signals here. There's a huge amount of data available there. By doing the signal detection calculations, among the data, we are identifying the signals. So we are telling that to the world first time, okay, this drug may cause reaction. That's what is entirely meant for pharmacovigilance. Pharmacovigilance is only meant for giving some initial triggers. Okay, this drug can cause reaction. So that's what even we are doing by analyzing the FERS data. We are trying to identify that any new signals are there for any medication which is causing the reaction. But that is not the end of the story. Certainly, after that, we need to go and do more and more studies, pharmacopy studies, to get more information. That's common. But however, it is going to be one of the initial steps. We can say that this is an initial step so that we are getting some information that this drug can cause this reaction. That's what maybe basically we can say.
Okay, sir. Thank you.
The signal detection itself is an initial process, initial step of the entire pharmacovigilance.
Yes, sir. Yes, sir. Thank you, sir.
Yeah, sure. Any other questions? Yeah.
Krishna, I have one doubt. Yeah.
Yes, sir.
You have mentioned about IC025.
Yes, sir. Yes, sir.
Yeah, generally in anti-cancer studies, they used to have IC50, but in this case, you are talking about IC025. Any significance on this, Krishna?
Sir, I think that IC50, what we are using is something different in pharmacology, especially for understanding the toxicity there. And here it is a calculation, sir. IC here it is an information component that is basically developed by UMC, Uppsala Montic Center, for understanding the signals. And there they have taken IC025 as a lower limit. So based on that lower limit, that means nothing below that. At least if we are getting 0 or more than 0 value, that means we can identify some positive association between the drug and reaction. So
this has kept as a standard value.
It's the standard value given by UMC, Uppsala Montic Center, for understanding the signal between a drug and reaction.
Okay. Thank you, Krishna.
Yes.
Yes, sir.
So really a very nice presentation. At the need of the hour, say the students have to understood the concept how the pharmacovigilance aspects been going on. Okay, without such elaborative things you have been given about the signal detection, how say we can easily first slide itself we can see signal. Okay, how important it is. Okay, so like pharmacovigilance also, say we have been dealing with an enormous number of drugs, and the CDSCO, every aspect you have been covered is really a eye-opening for all of us. Everyone should know what is exactly happening, and you have covered about the EMA process and also Uppsala monitoring process. So that is a very good thing. So I don't have anything because you have done an excellent job. You have presented very well.
Thank you, sir. Yeah. Thank you, sir. Thank you very much. Thank you very much. And there's one more question I can see here. Can preclinical safety data considered a signal in pharmacovigilance? Usually, no. Whatever the data we are identifying in the preclinical studies, that's only the basic data which can be helpful for us to get the approval for consumers. But basically, the pharmacovigilance means it is only the post-approval, the post-marketing, most of the times, even though we are identifying some safety signals during the preclinical and clinical studies. But basic pharmacovigilance, very broad area of pharmacovigilance mainly covers after the drug is releasing into the market. And we are identifying the signals by using a method called spontaneous reporting, like whatever we are doing in AMCs in hospitals. All those things comes under the spontaneous reporting. So through that, we are identifying the reactions. And by analyzing all these reports, we are going to detect the signals. So basically, the pharmacovigilance means it is maybe after releasing drug into the market, post-marketing surveillance, PMS. And for all the audience who are here, maybe who are interested more in pharmacovigilance and signal detection, I mentioned here one website called OpenVisel. It's a recent one made by one of the researcher, OpenVisel. I typed it here in the chat box. So we can go to that web page. That gives you more understanding on signals identification, especially by using the US FDA data, FERS data. Like earlier, what we need to do is we need to go to the FDA website and download all that information in the Excel sheet. Or now even the platform is available. You can go and search for the reaction. But here in this OpenVisel, if you can go and type any medication, any reaction, with so many filter options are there. Like you can use and or options so that you can have more and more information, more and more filters. By applying all these things, just by clicking one button, you're going to get all the signals from the FRS data. That means you don't need to do analysis on your own from the FRS data. Just by entering that information in the OpenVisel, you can get all those calculations very easily. I think you are not getting the IC value, but you will get PRR and ROR values there, so that you can get more information on signal detection from this website. Maybe you can follow if you're interested.
Thank you. Thank you, Krishna.
Thank you. Thank you, sir.
Yes, sir. Yes, sir. Thanks a lot, Dr. Krishna Undele, sir, for this outstanding presentation. You just nailed it. Your session was very knowledgeable. It has updated a lot about detection. Thank you so much for helping us and making us aware about formulas, reactions, and drugs. I appreciate you for that, sir. And for our panel, Dr. V. Murugan, sir, and Dr. Karthikeyan, sir, thank you so much for chairing this session up.
Okay, thank you very much.
Thank you, Doctor.
It's a pleasure to.
Thank you, Dr. Sakina.
Thank you, Murugan, sir. Thank you, sir.
Thank you.
Thank you,
Murugan, sir. Thank you, Karthikeyan, sir.
Thank you. Thank you, Krishna. Thank you.
Yes. So without any delay, let's move towards our next session, which is industry section. The topic of session is artificial intelligence in healthcare. The speaker of our session is Mr. Hardik Dev. Mr. Hardik Dev is a chartered accountant and CS secretary, and I am honorable to invite sir. I am honorable to invite Mr. Hardik Dev, sir. And here is a quick introduction about Hardik, sir. Dr. Hardik, sir, co-founder of Indica Oil in 2021 and grew it onto one of the fastest growing and sought after AI data solution startup. Basically, Mr. Hardik is chartered accountant and company secretary and graduated from Jai Hind College, Mumbai University. Before founding Indica AI, Hardik worked with Ernst & Young, Baker Tilly International in the areas of financial and corporate tax law consulting as well, accelerating digital transformation and technology adoption in these areas. He also served as National Director for Entrepreneurship for World Youth Heart Federation, a non-profit organization fostering interdisciplinary collaboration in healthcare. Indica Artificial Intelligence specializes in enabling data-centric artificial intelligence companies across all industries to build high-performing artificial intelligence solutions using its state-of-the-art artificial intelligence data training, testing, and validation expertise. I heartily welcome Hardik sir to please speak up about your session, and I invite our panel in this. I invite sir to please take over the session.
Sure. Thank you, Dr. Sakina, for the warm introduction, and a very good afternoon to all the participants here. This is Sunday afternoon and post lunch session. So I hope you're awake, right? Good afternoon. If you put this right, hi, hello, something in the chat box for me to be sure that you're all awake. I'm ready for the session. AI in healthcare, right? A very interesting and dear topic for me, very close to my heart. I would love to hear from you about, are you excited for the session or not? Right? So looking forward to your messages in the chat box. The chat box is open to all of you. Are we all there? Good afternoon, Shaiba, Ashok. I'll share my presentation screen in the meantime. Host, you would have to allow me to share the presentation. Dr. Sakina, if you could allow me to share the screen, please.
I have stopped the sharing. You can share now. Sorry.
Okay. I think your screen is still visible, Dr. Sakina. You would have to stop sharing.
Yes, Hardik bhai. एक second. I think there is some. Yes, Hardik bhai, sir, please go ahead.
Perfect. Okay, please confirm when you're able to see my screen.
Yes, it's perfectly visible. Go ahead, please. Sir.
Awesome. So good afternoon, everyone. A warm welcome again. While my introduction is already done, I'll take a minute to briefly introduce and to set the context right, that what is the PA doing in the discussion around clinical decision and everything around healthcare. So as introduced, my full name is Hardik Dave, and I'm the co-founder of a company called Indica AI. What we do is we work with companies that are building artificial intelligence solutions for all the industries, right? It includes healthcare. So we have been fortunate to work with several AI companies across the globe, I would say, and also across multiple verticals within healthcare who are building AI solutions, right, from imaging analysis to radiology and a lot of literature review and digitizing the medical prescriptions and other documents, a lot of these. So based on that, like based on these experience for last almost 2 years, I believe now we are in a position where we could share some insights about what is happening in the healthcare AI industry. Even before we go about understanding what's there in the AI industry, let me take 30 seconds to briefly introduce what is AI, just to set the context right. We all know, I'm sure it was a topic which is already covered, that artificial intelligence basically means that we are training the machine to be as intelligent as we humans are, right? In the healthcare context, if you have to understand this, say as a doctor, if you can read CT scan reports, or if you can read some sort of medical literature and you can understand and find meaning out of it, how do we train an AI machine to do the same thing, right? So whatever insight, whatever input the healthcare professional can give you from reading some sort of medical documents or symptoms or anything, if a machine can also be trained to the same level, we call it an artificial intelligence-based solution. Why do we need that? The context is very clear. The healthcare industry has always been facing three major challenges. Number one is around, is the healthcare facilities available? Number two, if they are available, are they accessible to all the people? And number three, if they are available and accessible, are they affordable for everyone? right? So if the healthcare facilities are not available or they're not accessible to the last mile within our country, and if they are accessible but they are not affordable for a common citizen of our country, there is something which has to be done. And that's where new interventions are always required. AI as a technology which leverages automation at its core gives you a chance to bring down the cost, right? And make the technology, make the facilities available. So think of, I would like to share a very interesting story here that it was about a client which used to work with us. And the client was building solutions around wound health annotations, right? So, for example, if I get some sort of injury on, say, my hand, now I go to the doctor. The doctor tells me that, okay, for now I'm giving you like whatever treatment, but you would have to revisit in say 1 day from now, then 3 days and 5 days and so on. For the next 2 months, I have to regularly visit the doctor. Assume a scenario where the doctor is say 100 kilometers away from where I live. It's not possible for me to take time on a very regular basis from my work and travel that far, right? So that's where the accessibility of the facilities become a major challenge. So to take care of these kind of cases, the client, which was a Hong Kong-based healthcare AI startup, was building a simple application where I can say click photo of my wound using a smartphone. A very smart, like very budget smartphone would also work, which can just clearly capture an image of the wound. And then I could upload that wound image on the application, and the AI would suggest me that is the wound improving or not improving. If it is improving, then is there anything which I need to be concerned about? And if over a period the AI finds out that the health of the wound is not improving and it is rather going on the negative direction, the doctor immediately gets an alert that, okay, you need to pay attention to this particular patient where things are going south. Right? So that's the kind of impact which the AI can make, because it saves me now. For me, it makes my life easier that I don't have to go to the doctor every now and then. Only when there is something critical, the doctor would automatically get an alert, and that's when I have to go to the doctor in person. Right? So it is how the impact of AI would be felt. Right? I know we are now in the era of ChatGPT, where almost everybody has at least heard of what is AI. If they have not been able to see the magic of AI, right? But the need for AI in healthcare is basically addressed, I would say, on these principles, that our common objective should be to make the healthcare facilities available, accessible, and affordable for everyone. If we have to look at some of the recent trends of AI in healthcare, the numbers are huge, right? We are sitting at an industry, and we are talking about an industry which is valued roughly around 14.6 USD billion in 2023. And the growth rate is one of the highest, right? No other industry has almost 50% of CAGR, right? I think that's the annual growth percentage. So in the next 5 years, we are crossing this annual growth rate, like at the annual growth rate of 47%, the industry would be valued more than $100 billion. Now that's a massive number. And why that number is important, because that's where you can see how much relevance and how much work AI, like the healthcare industry, would be witnessing in the next 5 years. Right? So that is where every healthcare professional, irrespective of what you do, which domain of healthcare you are into, right? You need to know about what is AI and how does that affect your profession, your career, and what kind of opportunity it offers for you. My presentation today would mainly be focusing on these aspects. What are the major use cases of AI, which is something you should be aware of, how these AI solutions are built, and what are the core challenges and opportunities in this domain. Right. So firstly, talking about, to give you a broad picture about what's there in the AI for healthcare industry. Right. Some of the very common use cases that you would have heard, it's a lot of noise and news which keep coming up, that now robots would be assisting making the surgeries. We can have AI to make personalized meal plans, treatment plans, all sort of things. Now AI can help you detect diseases. It can do all sort of imaging analysis of all sort of CT scan reports and sonography reports, X-rays, MRIs, everything. So that's also there, right? A lot of interesting work is being done around using AI for clinical development, drug discovery, clinical trials, right? And a lot of interest in it. So I would say, if we have to look at the human body from head to like bottom, there is hardly any area where AI is not touched yet, right? So that sort of impact is there. It doesn't matter which domain you would be into. Within, like, are you studying pathology or radiology or clinical science or whatever it is, there is something which is happening now as we speak in the field of AI, some sort of AI research, some sort of commercialization of the product, some sort of new product development is happening, and you need to know about this. Why AI is important? As I said, the three major objectives, which is a golden standard, a golden principle, a rule of thumb, you can say, that whatever we do with AI, it would be ultimately aimed at making the healthcare available, accessible, and affordable for everyone. Right. So that's where several operational efficiencies and financial benefits of reduced cost of healthcare is what we can offer. Right. Whether it is about study design or using AI for pharmacovigilance or site identification, patient recruitment, clinical monitoring. I do not need to go in detail because about these things. What are these things? Because I'm sure it's already covered in the sessions over the last 2 days. But the slide where I would like to spend most of my time into this discussion is to give you an understanding about how these AI solutions are built. Right? Let's try to understand this whole AI development life cycle into step-by-step process. Right? So say if we want to build, let's take a sample use case in our mind. Right? So we want to build an AI-based solution, which can, as simple as that it can read CT scan report and it can identify is there some sort of a tumor or some sort of defect is there or not there. So that's the problem, that we want to build an AI solution which will be able to do this imaging analysis and tell if there is some defect, some deficiency, something unusual which needs to be noticed and taken care by the doctor. So if we have to build this solution, how do we go about this? How it would be approached in the whole development process? So the first step would be that we are defining the problem. Now to define that, the problem is defined that there would be a software, we can create new data to that software, that new data would be processed, and if there is anything adverse, it would be reported and flagged to the doctors or like the radiologists and pathologists who are doing that analysis. Why? Because so that it does not go unnoticed, so that it does not take that amount of time which is now being taken when the whole analysis is done manually, right? So that's where, how do we train this thing to do? So that's where a lot of training data sets are needed. So once the problem is defined, the next step is to define a AI training strategy, collect the relevant data, which can be used as a study material for that AI algorithm. That, okay, see this particular report. In this report, these are the 10 points which were adverse and which have been annotated by the doctors. So next time anything of this sort is coming up, you need to report. It's basically giving an instruction to that AI software that, okay, these are the things which needs to be noticed, needs not to be noticed, so that it can identify the patterns over the period and it would accordingly start learning those patterns. So that's the second step, where we are basically collecting the relevant training data sets and pre-processing them in a way. Once that is done, now it's about how do we train that, how do we feed that data which has been collected and prepared to the AI algorithm. And based on that, the training would happen. Once the training has happened, now we need to validate how well the AI has learned so far, right? Is it giving us accurate report? So that's where we would prepare a small validation or testing data set. So think of it this way, that we wanted to build this AI-based software that I talked about to read CT scan reports. Now we've created, say, a data set of 200 such reports. Out of that, 150 we initially used to train the solution. Now next 50 we are using to test that, okay, whatever training is being given based on those 150 reports, how well the AI has been able to learn so far. And if not, we would constantly give more and more feedback that, okay, this was an adverse thing which you have not been able to notice. Please take care of this next time, right? So the process of training an AI algorithm is very similar to how we human learns everything new, right? And once this solution is able to reach to a particular level of accuracy, that's the last step where we can now deploy the solution in the real world and we can start using the AI, right? Think of it as a step-by-step journey that we first have to identify. Now identify what we want to build. Based on that, how would we train that, whatever data is needed, how do we prepare the data set for AI training? Then we make the AI algorithm, we train it, we then test and validate how well the AI has learned. And once we have got the comfort that, okay, now AI is good enough, then only we deploy it into the real world environment. So what's the problem, right? If you might be wondering that, okay, it looks like a very simple process, why then AI development is so much hype. So the hype is around the challenges, right? The whole step. So in this slide, if I have to say that out of the 6 steps, which is the most complicated step, that is step number 2. AI is all about data, right? So if we are not training the AI with the right kind of data, the AI model would not work accurately. If the data that we covered was not comprehensive, not sufficient to cover all sort of possibilities, again, AI would be lacking somewhere. So every time we talk about how well the AI is, it is essentially discussion about how good the data was. And that is where it becomes very important for every AI and healthcare professional to understand the importance of data in AI development and also the corresponding challenges which are there. Right. So let's talk about the four major challenges which are there when we talk about AI in healthcare. The first one is availability of the data. Right? So where is the medical data available? So if I want to start building a new use case, I do not run a hospital. I neither run any sort of medical lab or something. I do not have simply access to the data. Right? How would I train my AI algorithm for something? Similarly, say if we have to predict that, okay, there is possibly a new epidemic or pandemic which might come up, right? We have found out that, okay, these sort of symptoms will be there and all. But how do we train an AI for those future possibilities? Because there is simply no data available, right? So a lot of the work in the healthcare AI has not been done so far only because the data was not available. The problem with AI is never that there are not like that the AI algorithm or the softwares are not available. There are plenty of software for possibly all the problems to be solved using AI. But why AI does not accelerate within the healthcare industry is because the data is not available, right? But you might say that I work at so-and-so hospital or I work with so-and-so lab or drug company. We have n number of data. Where is the problem? So once the problem of data availability is understood that, okay, the data is either not available or it is available with somebody else and not with the people who are actually building the AI solution. So how do they get the data? Or why the data cannot be shared by the hospitals to the AI startups who are building the solution? The problem is around regulations, data privacy, right? You must be aware how stringent the data privacy law is when it comes to healthcare data, right? We're talking about, say, we are all sitting in India now, where there is still some gray areas around healthcare data protection and all. But think of a country which is governed by GDPR, or say, think of USA, which has very, very strict data protection laws. Think of a startup in, say, California or New York, which wants to build healthcare solutions. Where is the data? How would they get the data? So that is where the regulatory aspect of being able to access those data becomes a massive problem, and it gives opportunity to new type of solutions, which is data anonymization, right? So is there a way that I can anonymize the data so that the identities of patients, that whether it is your blood report or my blood report, whether it is your CT scans or my CT scan report, right, that can be anonymized, but it does not affect the insight which would have been there from those data if I would have done some sort of analysis on the actual data with identity, right? So it needs a very strong domain expertise that, okay, what all data points are relevant for analysis and what is not relevant, plus a very strong capabilities in AI and data science, so that you can very intelligently anonymize these data sets in a way that the purpose is solved, but the regulatory requirements are not broken; they are complied with thoroughly, right? So that is where the second challenge, that is where a lot of, like, it actually becomes a bottleneck if you're not able to anonymize the data in a way that it can comply with the regulatory requirements. It becomes a massive bottleneck. I cannot tell you, I cannot emphasize on that point that how many AI projects have never seen the light of the day only because the data was not terrible, right? They're both the sides, like the hospitals do want to build these AI solutions. There is a startup, there is a team which want to build a solution, but they cannot coordinate for data sharing, and therefore there is no next step which are being taken care, right? So once the data availability is understood and the anonymization part is understood, the third step is data preparation, right? How do you make a software understand what is tumor and what is not a tumor, right? Or what is adverse or what is favorable? What is right, what is wrong within a medical document, within a medical image, or whatever data which is in a data set which is there. So that's where a data preparation process, or I can say very simply, I can say data annotation process is involved. And this is also the step where large number of job opportunities are being created and would be created over the next 5 years. Right? So remember the initial slide where I talked that the AI healthcare industry is going to rise from 14 billion to more than 100 billion. The major value addition would be happening at this stage, which is around data preparation. That how do you make the data ready to feed it to an AI solution so that whole data annotations can happen and the AI model can learn the patterns which are there. And I'll talk more about this with a few examples in my coming slide. But for now, to move to the next step, that's the biasness. Right? So whatever data set that you had, if it had some sort of biasness, or if it was not comprehensive enough, right? So for example, if I do testing of a new drug based on only one particular gender or only one particular demography of patient, my results are not accurate, right? Similarly, if I do AI training based on one particular type of data set, it has an inherent bias that the other people might not be suitable for this. So these are the data challenges, that the data is not available. If it is available, it is not shareable because of regulatory concerns. Even if it can be shared, there are massive investments to be done on the data preparation part. And even after the data set is prepared, you need to be double sure that whatever data you are using, is there some sort of biasness involved. Trust me, 30-minute session for making these points very clear is not sufficient, but I'm just trying to throw some light that you can possibly awaken some interest on these topics and try to read more about these, right? For now, let me go into one slide, which is more important and probably could be the core takeaway for you from this discussion, is where does the opportunities lie for healthcare data professionals in AI? And the answer is data annotators, or we can also call them data professionals. Data annotation is simply a process. So for example, look at this first image, which is CT and MRI scan analysis. Now think of it as that we are training a software to find out these particular spots within the scan report. How would we do that, right? Similarly, for dental images or dermatology images or mammography images, ultrasound or whatever reports there, or say there is some medical documents, literature available, we need a team of domain experts that understand these kind of data, right? Which basically means that they understand this data in the domain so that they can work with AI professionals, the techies, which would be building the AI solutions as a joint team, right? So this is where we as a company also employ a very large number of data, like healthcare professionals in the process called doctors in the loop, where they are basically working with us as a data annotator and helping us to build these AI algorithms with the right sort of domain expertise, right? Now, the next takeaway could be a few interesting companies that you should probably note down and read more if you have an interest to do something on the healthcare AI. There are 4 names when I think about Indian startups: Cure AI, Ticktuple, Miramide, and Redbrick AI. Redbrick is like a company from Indian founders, but based out of USA. Rest 3 are Indian companies only, right? These 4 companies, of course, not necessarily, I'm not saying that these are our clients, but these are the companies which are doing fantastic work when it comes to healthcare AI in India. Similar, a few companies in the international scenario is Space AI, Orchid, AI Cure, and Verisend. Again, you should definitely check out their website and try to develop more understanding about what exactly these are doing, right? So in case you want to take a screenshot of this slide, feel free. I will also try to share this presentation through the organizers so that you can have something for future reference. Moving to the next slide, in case you are interested to work as I call data annotator, feel free to go to our website and you would find this link, work with us, where you can register with us to be working as a data professional. And that's where we keep coming up with several exciting opportunities across industries, not just healthcare, and working with state-of-the-art AI teams across the globe to make the highest possible quality of data sets for AI training. So this is end of my presentation. I hope I've been able to wind up this in the due time. I'll be more than happy to answer any queries which might be there. Thank you so much. back to the host. I think I'm done.
Hi, thank you, Hardik bhai. Wonderful presentation and definitely, eye-opening things as we are always looking forward. The AI in clinical research, in healthcare, in all the domains as well, right? So as today we are talking about the AI healthcare, I would like to add on. So guys, anyone, if you have any kind of innovative ideas in your mind, any kind of things you want to pursue in AI, or for the students, we have amazing internships and amazing trainings at AI. You can just go to our Indica AI website, share your CV, and you will get the internship. You will get I think this is totally a wonderful platform to learn AI and to share your ideas, to execute your ideas, come up with the ideas, or if you want to think pursuing something in AI or healthcare, pharmacy, anything, research development, anything, please come to us. We are here to serve you, and we are working with other multiple organizations in partnership. So we are going to help you in any ways. So I and Mr. Hardik Dave, my friend, we are in Indica AI, we are taking up multiple projects. We are working on national and international projects. And as I told yesterday as well, we have listed in top 50, AI companies, and we have, presented recently in, All India AI Fair and Technical Fair. So, you can come up. And if you have any questions to Hardik, please, guys, the session is open for the questions as well. Anyone have any questions?
I think we're all good.
We are all good. Perfectly fine. Hardik bhai, thank you. This is an amazing session. Yes.
Yes, sir. Thank you so much, Mr. Hardik Dave, sir, for your wonderful presentation on artificial intelligence in healthcare. We have learned a lot in your session, sir. Thank you so much for enlightening us with your knowledge, with your presentation.
Thank you so much for having me. Have a good day. Remaining one.
Thank you, Hardik bhai. Thank you. Bye bye. Take care. Yes. See you, Hardik bhai.
So let's start our next session. This section is a keynote session. Topic of this session is grant proposal writing, that is ICMR and NIH. So I'm very honorable to invite our speaker, Dr. Ajit Singh sir, research scientist for ICMR, Department of Medicine, Kasturba Medical College, Manipal. Let me give introduction about Ajit Singh sir. Ajit Singh sir have done PhD in cardio and heart failure, co-founder and chief executive officer at Climate Research Solution India. A young entrepreneur and scientist, has completed his PhD in cardiology from the eminent Manipal Academy of Higher Education. He is also serving as National Director for Research at World Youth Health Federation. Currently working as research scientist for ICMR at Kasturba Medical College, Manipal. In July 2021, he has been appointed as Vice President of the Medical Artificial Intelligence Wing at Indica AI, Mumbai. He has published more than 40 papers in different streams of medicine, including cardiology, oncology, and infectious diseases. He is the investigator for many international clinical trials. Dr. Singh serves as honorary reviewer and editorial board member for more than 20 reputed international journals. He has received several international research and travel grants for esteemed organizations, including the European Society of Cardiology, the Indian Council for Medical Research, DST, STRB, and the American College of Cardiology. He received the Young Achievers Award 2019 and Young Scientist Award 2020 in cardiology research from reputed international organizations. We are very honorable to have you, sir. Thank you so much for your time and for this conference. And please start your presentation and enlighten us with your knowledge.
Yes, thank you, Sakina. Thank you very much.
I'm heartfully welcoming. I'm heartfully welcoming our panel, Dr. Mahendra Singh Rathore.
Yes, sir is joining in a minute. No problem. Sir will be joining. So let me start. Guys, thank you very much. And this session is very important for all the students and academicians. And when we are talking about, medical writing, when we are talking about scientific writing, and am I audible?
You are audible.
Yes, perfectly fine. Thank you. Wait. Yeah. I hope it is visible now. Yeah, right?
No, sir, it is not visible. Yes, sir, now it is visible. We are able to see.
Great. Thank you. So, this is the last session for, first of all, guys, I would like to thank you all and the people who are joining. There are a few people in YouTube as well. So I would like to thank you all. Thank all the stakeholders, our speakers, our guests yesterday, and all the teams who were participating behind the curtain. And most importantly, our, the people who were in scientific team. So they all are working for last 3 days, right? So that vote of thanks I'll definitely do at the end, when we'll be going towards the valedictory. So let's come to the presentation of now. So I kept it this special presentation, guys, because this is very important for students and academicians. Everyone, whenever I talk to someone or they approach me, they ask about the grants. As I'm, definitely there is, such things. I'm working with ICMR and MAHE, and we at CLIMAT also we support the researchers in different ways. So this is a big question. This is a big question from everyone who are going to pursue the PhDs or masters, or who want to come to the research field, or the academicians who want to grow in their academia. So they always ask, what is the plan for the grants? Can you tell us what are the formats for the grant? Can you tell us what are the agencies for the grant? Scientific grants, right? And the major part in this is, if they come up also with the names of the agencies, if they come up, if they know about the grants as well, if they know everything, at the end the question is how to write the grant. And this is also a part of scientific writing and medical writing. Okay, this is very important part, because this will lead you to the grants. This will lead you to the funds to pursue your research. This will get you the funds, right? This will impress the judges. This will impress the funders. This will impress the sponsors. So I feel this is one of the very, very important document and very, very important format. What you should understand, how to write, what you should keep in mind, what you should not write, do's, don'ts. There are multiple things you should keep in mind. So to discuss that only, I'm here with you all, and I'm going to, discuss all the things, all the possible things, what I can discuss in next 30 to 45 minutes. This session, usually I take 5 hours, frankly, right? So I have, cut short many of my slides for today's session because it's restricted, but I'll still, try to justify myself and try to justify the topic as well. Okay, so let's start. Let's not, wait. So what I'm going to cover today is, we will start with our, grant and funding sources, then, selection of appropriate grants, and what are the purpose of grant proposals, what is the research question, and importantly, what are the components of research proposal, like budget estimation, timeline settings, and what are the samples what I get from ICMR and NIH. There are multiple DST, DBT, EU, and multiple other like NH. There are different Bill and Melinda Gates, Reliance Foundation. There are multiple sponsors in India itself. Okay, Bill and Melinda Gates is USA, but based in India as well. We have the entire research center in India for Bill and Melinda Gates. We have NIH center it's itself in India. Okay, NIH center from US also. We have the evaluation center in India itself. Okay, so you can write the grants to anywhere, but there are certain things what you have to keep in mind. You cannot just come 3 days before the grant deadlines and come to me, and you cannot ask if I can submit a grant proposal, right? So there are multiple things what you have to keep in mind, and we are going to discover today in this presentation. Okay, so let me change. So first of all, what is grant? Very important part is this is like a giving of funds for a specific purpose to an individual or an organization. So first of all, let me define the individual or an organization. Individual means you cannot sit at home and do a research. So that is not allowed in that part. Individual means if you have your own laboratory, if you have your own research center, if you have like your own clinic, if you have like your own hospital, right? So that is called as individual. Being an individual scientist, I cannot go for the grants if I'm not affiliated to any center, if I'm not affiliated to any resources, infrastructure, facilities, right? These are very important part. So it is not necessary that you have to be attached right now, but if you are going to work somewhere, if you are going to tie up somewhere, you have to disclose that okay, after 6 months, once I'm expecting the grant, I'll be in this hospital, I'll be in this, and I have taken the permissions, entire permissions for the infrastructure, for entire things from this organization, and I can pursue this research there. So I'm asking for such grants from your organization, from your sponsor, right? If I'm writing to ICMR, if I'm writing to suppose DST or CSIR or DBT or Bill Melinda or Reliance Foundation or anywhere, right? I have to give them the supportive letters. I have to give them the permissions, entire things which is supporting my background. Being single, alone, and without any affiliations, individual cannot apply for the grants. Okay, then this is generally obtained through a competitive process. Definitely, it's a competitive process because for one grant, for like one topic, one grant, there will be hundreds of the applications with the sponsors. So they have to scrutinize, they have to go through, they have to check everything, and then only they will give you the grants. And they will, they have the entire process of assessing the proposal, all the proposals, all the grant proposals. They have the criteria to assess the grant proposals, and based on that only, if you are perfectly fine with them, if you are aligning with their criteria, then you can get. And I'll talk about those criteria as well. So those criteria, if I discuss now, those criteria are nothing but if you are aligning with their condition. Condition means how, like, see, ICMR every time does not open the grants for all the diseases. They coming up, they usually come up with seasoned infections, sometimes cardiovascular problems, and sometimes they just come up with the non-infectious diseases, sometimes they come up with the infectious diseases, right? So they have the different departments in ICMR also, which takes care, like someone sometimes they're coming up with just the diabetes, sometimes they're just coming up with the cardiovascular problem, sometimes they're coming up with only influenza. So you have to align with their condition, you have to align with them. You cannot just simply, if your specialty is cardiology and you are applying for influenza grant, that's not suitable. If you are applying, then definitely there are the high chances of rejectance. So they look at everything, the suitability of the person. If simply just being enthusiastic and applying for different streams, you might not get that. It should be aligning with your specialty. It might align with your work, your previous work, your published work, your specialty. That's important, right? So first, when they open the calls for the grants, read them clearly. It's very important to read them clearly. Like if we are really aligning with them or if we are not. Just looking at the amount, if they are offering 1 crore, 2 crores, 5 CR or 10 CRs, we should not go for that. Because if we are doing that, there are the high chances of rejection. Means we are wasting our time, we are wasting their time as well. Because they should establish one, what we say, the trust factor is also there. Okay, in that criteria there is a trust factor as well. If they feel this guy has the good proposal but not having the good support, but not having the good infrastructure, but does not have the good background in this field, they will not give you the grants, because they have to assure the timelines, they have to assure the future of the grant, they have to assure the future of the project what you are writing. Maybe you are very enthusiastic, you show everything perfectly, but your background is not at all messed. You don't have the collaborations, you don't have the perfect team with you, you don't, so you are failed to show that perfect team in the proposal. High chances of rejectance, very high chances of rejectance. Okay, so definitely competition is there. This is not a gift at all, not a charity or not a loan. If you think like if you cannot do it, you have to return it, I mean you will return it, not at all. Or it's not a gift, you have to work on that. If you get it, you have to justify the work on that, right? You have to justify the work on that, otherwise you will be blacklisted and you will not get the funds again. If you are not fulfilling the demands, if you're not fulfilling that research grant criterias or the requirements, you will not get it again, and that particular agency will not entertain you again at all. Okay, so definitely it's a relation between the grantor, as I'm telling, and grantee. Is an exchange relationship, as I'm telling. Very much it's a trust factor. It is a trust factor based. If they trust your proposal, and trust does not come with only your proposal, with your good idea, not at all. It should match your background, it should match your, if you have a perfect team, perfect collaboration, perfect infrastructure, everything, they look at it. It does not mean that they do not give to the new colleges, it did not give to the new researchers, or they did not entertain the new newbies. They entertain, but at a certain extent. Okay, at a certain extent, they start, they build a trust factor then, and then they start giving you the funds, right? And then definitely it is exchange with the very type of grants. So exchanges means some sponsors, they want the reports, they want the presentations, they want the publications. Some grant sponsors, they want like in return, as if you are getting the grants from the private company or the pharmaceutical company, they definitely need the data. If you are getting the grants from some government funding agencies like ICMR, DST, or DBT, they just need the reports and the final report. They don't want your data, they don't want anything from you. Okay, they just want the justifications, reports, how have you spent the funds, where did you spend the funds, how did you collect the data, everything should be transparent. But they don't want your data. Okay, they don't want anything from you in exchange, right? But if you are dealing with some pharmaceutical company, they need your data, they may ask for the data analysis by themselves, they may need some what we say, some kind of exchanges, so not mention that, right? So different funding agencies have different exchanges varying. Okay, now what are the funding sources? Definitely if we start with here, internal, let me come up with the pointer, guys, then it will be great. Yeah, so if we start from here, first internal means you can get it from your university, right? You can get it from university. Universities also give the grants, like I'm at Mahe gives every year to some 15-20 students, and more than 20 students, they really fund internally to their students as well. If you have the good idea, you're not getting funds from anywhere, then university itself start giving you the funds if your proposals are really good. That is internal. Sometimes it is self-funded also. Okay, but here we are not talking about the self-funded. Here we are talking about the grant proposals for the sponsor. So in that, first number is internal. Internal means your university, your college, your institution. Okay, then state schemes. Then there are certain state schemes as well, like Karnataka Bionest. Here working in Manipal, so I know Karnataka Bionest is the state scheme, and different states also have their different schemes for like non-infectious diseases, infectious diseases, to encourage the innovators, to encourage the new scientists, to establish the labs and all. So states also have their own schemes to give the funds. You just have to explore. That's important. Okay, that's important. You have to explore the ministry sites, you have to explore the government sites, then you will get it. Okay, then the federal government comes. Like in India, we have federal government, and government of India, and in that we have Department of Technology, we have Department of Biotechnology, we have Department of Science and Technology, DST, DBT, Department of Biotechnology, we have CSIR, Right. And we have, I mean, CSIR is for our industrial scheme, and then we have ICMR, Indian Council for Medical Research. So these are we have AICT, we have our University Grant Commission. They are the federal government things. They are the federal government agencies which provide the funds in India, right? And industry means your pharmaceutical industry or AI industry or technical industry, any kind of industry. They give the funds and they support the research, right? As in India, Novartis is there, Sun Pharma, Lupin, Cadila. They all fund the people, and majorly, as we can see, Novartis and Eli Lilly and Pfizer, they are giving a lot of clinical trials. They are conducting a lot of clinical trials and giving the funds to the students and academicians. And then, as I was telling about the foundation interest in India, Reliance Foundation is there, Tata Foundation is there, Tata Research Foundation is there, and they have their own travel grants as well. They have their own research grants. Reliance also have that. And then there are others as well. I forgot the name of Mother Teresa grants are there. Okay, Mother Teresa Foundation is there in India. Okay, and then international, if I talk about, there are NIH. Okay, and there are Bill Melinda Gates, and there is NH. There are different agencies and different foundations which gives you the funds for your research, right? So these are the funding sources. Let's not talk on that much. And then there is now how to select an appropriate grant. That is the first step. That is the first major step and important step while you are going for the selection of the grant. So it always starts with, will obtaining the grant aid your studies or career? Okay, so first question, I really believe that this question is very important. Okay, this question is very important. If it is really something which will aid some good career or good achievements or good success in your career or job or in your studies, then only go for it. Just for fun, research is not for fun. Okay, research is not for fun. If you think you will write a grant and unfortunately, I'm telling unfortunately word here, if you're just getting it unfortunately and not completing it, it will be a wastage, because the people where it is not usually needed, they are not interested in that. They're not interested in pursuing that. They're not interested in completing that. Okay, so that is very important, that you should have some interest in the grants. So before going for any kind of grants, you should have the interest. You should have some kind of what we say, the motivation, if it is aiding something to your studies or career. Then do you meet the criteria? Very important thing, as I was telling. Criteria means there are the specific requirements, there are the specific criterias from every funding agencies. There are certain agencies which are opening once in a year for everyone, like ICMR is opening once in a year for everyone. But there are other add-on grants as well. They are opening in January, February, March, April. They are not for everyone. They are for specific people. They're like opening in usually January, February. They open for the MBBS MD students, STS. Okay, there is a support of 50,000 for only for MBBS MD students, right? And sometimes they are opening only in the non-infectious diseases. Sometimes they are opening only for telemedicine. They are opening for new innovations. They are opening for waterborne diseases. So if we are working in that field, then only we should apply. If we are matching their criteria, sometimes their matching criteria is not just the field or the stream they are applying, they are calling for. Sometimes they are looking for the scientists with well experience as well. Sometimes their criteria is very clear, like someone who has published 5 papers, someone who is working for more than 5 years can only apply. So if you're not meeting their criteria, if you're like, oh, I have done 4.5 years of half years I have spent in the research, but I did not spend 5 years. They're asking for 5 years. Maybe till this research proposal will reach ICMR, I'll complete my 5 years. They will straight away reject it. These are the top criteria they are looking for, because they want someone experienced to spend their money in the right way, in the right direction, right? So you have to go for the discipline first. If you really belong to the discipline, if you're like, as I'm telling, if the diseases which they have mentioned are belonging to infectious disease, if they're asking for support and they're calling for influenza, and being in cardiology, if I'm very much relevant to influenza, I'm working on something influenza and heart failure or influenza on myocardial infarction, then only I should go for it. Simply, I should not correlate the disciplines. Simply, I should not waste my time and their time, right? So matching criterias are usually disciplines. Their intentions, what is their intentions? Like their intentions is like 5 years data I want. This should be the like population study. This should be the community study. And you are thinking you will do in the hospital. So their intentions and your intentions, their outcomes and your outcomes are not matching at all. If these are not matching their intentions and purposes, you should not go for it. Okay. And if you are meeting the criteria, then only you should apply for it. Main and important part is execution. If it is really feasible and achievable, look at these two, feasible and achievable. If your facilities, as I told you initially, if you have the perfect facilities to pursue that research, it's very important. If you do not have the infrastructure, what they're asking for, if you do not have the instruments, if you do not have the laboratory, if you do not have the facilities at your center, right, or where you can collaborate and pursue easily. It's not necessary to have everything at your center only, guys. No need to get demotivated. But if you are not able to fetch those things either at your center or outside collaborations are very important and as a positive point. If you do not have something at your facility, at least basic infrastructure you should have relevant to that project. Like someone is asking for data collection, you should have the laboratory where you have computer, telephone, printer, Xerox machine, everything, and you have the patients, main part is patients or the participants where you need, right? So that is basic infrastructure. If you have basic infrastructure and then you want some advanced infrastructure, if you do not have that advanced infrastructure with you, you can collaborate with someone. You can collaborate with some college, you can collaborate with some what we say hospital or lab or research center. That's perfectly fine, and that is a positive point, right? But if you are not able to achieve that, if you're not able to get it, if you think it is not feasible at my capacity, okay, if you do not have the proper teams, like if you, as I just given another example, influenza and cardiology, if I am thinking like I have a perfect plan to correlate influenza and heart failure, because every year the severe heart failure patient has to take the injections or prophylaxis for influenza, right? If I'm thinking a perfect match, correct, but I do not have any infectious disease expert with me, I should not go for it, because I cannot handle the entire part with me. If I do not have the proper nursing staff to give the injections or whatever I'm planning, if I do not have the proper team with me, I should not go for it. You should have the feasibility check. Feasibility check is very important. If you think you can gather it, if you think you can catch it, if you think you can make it, definitely you should apply for that. But if you really think that it will not be possible or it will be really tough to get it, then I believe you should not go for it, right? So do you have the sufficient time to allot? Another very important question. Sometimes people overburden themselves. They are already doing 4 projects and they want to take further projects, whether they have time or not, right? They are involved in multiple other projects. They are involved in teaching. They are involved in their extra classes. They are involved in academics. They are involved in what we say the administration. They are involved in multiple other things, cultural committee, a lot of things, right? And they do not have proper time to invest to the research. Don't go for it. You should think these questions before applying to any grants. Like if I can sacrifice my rights, if I can sacrifice my time, if I can sacrifice my personal space as well, sometimes personal time as well, because research means it needs time. Definitely, if you are in the clinical scenario and all something, then you may not need. But if you are in the laboratory work, if you're working on pharmaceutics or some pharmaceuticals or some innovations, then you may have to spend 24 hours in the laboratory. then you may have to spend 36 hours in the laboratory only. You will not get the sufficient time to do that, right? So always ask this question if you have the sufficient time to allot, right? And if it is achievable. So these things are very, very important to go for selection of the appropriation of the grant. Okay, and definitely trust funding agency, trusted funding agencies only. There are some fake funding agencies as well. There are some small companies which just catch you, and they will give you some kind of greed that okay, you do this research and give us the positive results. I was talking in the morning session, positive results, modifications, data fabrications. Don't go for such agencies, don't go for such brands, or don't go for such pharmaceutical companies as well. Always get the trusted funding agency. And then contract and agreement. Before going into the contract of research, going into the contract of agreement of the research, you should read it properly, and then only you should go for it. Otherwise, they will give you a lot of burden and a lot of other things. Sometimes they what we say, they trap you in certain agreements that you cannot come out later, right? So better you go for the trust agreement and trusted funding agencies only, right? Then next part is research grant proposal. So a research grant proposal is nothing but a document or the set of documents that is submitted to an organization with the explicit intent of securing the funding, right? It should be, there are two things. One is short proposal. Sometimes ICMR also and multiple these funding agencies or sponsors, they look for the short proposal first. They scrutinize the first these short proposals, and if they find out something good, they call for the full proposals, right? So purpose of the research proposal, simply from the investigator perspective. From the investigator perspective, this is the idea, or the research proposal is nothing but your idea, and you have to sell it to funding agency and convince them. Okay, research proposal is nothing but definitely selling your idea to the funding agency and convincing them like you can significantly tackle the problem. Okay, I mean, first you have a significant problem, worthwhile research project with you that is feasible at your center. Perfectly you can pursue it. Okay, perfectly you can go for it, perfectly you can execute it. You are competent enough to do that, and you have a proper work plan, team, collaborations, everything to complete it. And you can complete it with the new milestones. Okay, and it will give the sponsor also a new milestone and new research or a new outcome, which will be like a breaking, what we say, the breaking news or breaking research, right? So those kind of things that will give you you have to convince the funding agencies on. Like, first come up with a significant problem, so them like you can tackle it. You have this worthwhile research project, it is feasible for you 100%, and you are enough to do that. You are the perfect person to do that. You have a perfect work plan, teams, collaborations as well. Okay, and it will come up with a great opportunities and new research outcomes, right? Then selection of the research idea. First comes up, there are multiple things how you can come up with the research idea. Suppose like once you get the suppose that there are the funding agencies and they have come up with the grants, grant proposals and grant calls, right? So what you have to do is you have to come up with the research idea first, because they don't give the research ideas, they will give you the topics, they will give you the areas, they will give you the research fields, but they will not give you the research idea. You have to come up with the proper idea, aims and objectives. So it comes usually like, first of all, it comes with the discussions. Okay, discussions means discuss this with your people are usually discussing in the seminars, conferences. As this conference is going on, multiple new things I came to know since yesterday. What can be done in AI, what can be done in pharmacovigilance and AI, what can be done in medical writing, what can be done in in the research. We have evaluated almost 180 abstracts. We came to know a lot of new things which are going on around India and in different centers. So we got a lot of new ideas, right, in different fields. So sometimes if someone is coming up with a new research proposal, I'll have that in mind. Okay, something okay, we can also do something, explore, we can something what we say, the explore something in that area, or we can extrapolate that part in with some modifications or in some new way, right? So these things are always discussed at such platforms, the international, national platforms, national conferences, workshops, seminars. Always you should attend those things, right? And that's why I always advocate people to attend the seminars, always go for the conferences, and majorly look at the abstract submissions, abstract presentations, poster presentations, because you get a lot of idea from that, right? And what people are speaking, they are giving new implications, right? Since yesterday, everyone is talking about AI, how to tackle AI, how AI can help us, how AI is not helpful, so a lot of things are being discussed, right? So these things are very important to discuss, and these things we can learn from the discussions only, right? The new research ideas. And then literature, definitely you have to go for the research literature, and in the literature you have to find out what are the implications written at the end of the papers, end of the review papers, end of the systematic reviews, end of the original papers. What are the limitations they faced? So if you can tackle those limitations, right, if you can extrapolate those things, if you can go further with those things, and there are always implications, go with those implications, what they couldn't do, if you can do those things, how you can extrapolate those research projects. So this review of literature gives you always the research gap, and on that research gap only you have to hypothesize, and you have to come up with a new research question, guys. Okay. And then always you can read the news and magazines, science magazines, always give the new ideas. And then definitely the social needs, as we have seen the pandemic, when the corona came, now there are lakhs of the new researchers have come into this area. They are working on corona pathophysiology, corona related long corona, so and medicines in corona, repurposing of drugs in corona and new onset of diabetes, corona and thrombosis, corona and pancytopenia, corona and heart attacks. A lot of new things have come. So social needs, right? You have to just look around into the literature, discussions, conferences, media, social needs, right? And you'll get the wonderful topic. So once you have a topic, develop a good strategy for the addressing all the relevant components, right, guys? Now, defining the research question, your research question should be very much feasible, reasonable, significant, and novel. And then always beware of scope creep. Scope creep is nothing but you should not extrapolate in such manner that is not acceptable. Okay, and you should not look into such topics which are not at all acceptable by those people, right? So these are the things what we usually need to accomplish in the research question. First of all, feasibility, we have discussed this. Reasonable probability of success. Based on the feasibility, based on the execution, we can definitely define what is the probability of the success. If you are having the good probability of the success, definitely go for it and don't look back. Okay. And then if it is novel and significant, so definitely, as I always tell my students as well, always look for the significance of the research and novelty. See, novelty does not mean that if the research has been done in Macedonia and somewhere in Madagascar, as you cannot do in India. If you are doing in your population, that could be novelty. Okay, because it will give you the different outcomes in your different population, various populations. Okay, if it has done in different geographical conditions, you can do in your geographical condition. See, innovation is different than novelty. Okay, novelty is something you are doing it for the first time in your population. Okay, with your data, with your samples at your site in your area. See, if someone has done the research study or research registry in cardiology, in heart failure in New York or in Spain or in somewhere in Greece or somewhere anywhere in Japan, it's not like I cannot do that heart failure registry in India because I'm representing India. That is geographically different. That is environmentally different. Pathophysiology of the people is different here. So that is also the novel idea. Okay, and novelty is something which you are extrapolating from which has already been done, right? And it should be different from others as well. But if you are doing like, okay, my seniors also have done the same thing, I am also doing the same thing, that is not novelty at all. If you are doing at the same center similar things, then you are wasting your time, wasting your research, wasting your resources, right, guys? and your research question always should be smart. So what do you think by smart? Smart is nothing but it should be specific, it should be measurable. Measurable means it can be easily executed, okay, and outcomes are measurable. Achievable, definitely. Relevant to the society, to the topic, to the idea, whatever given to the organizers, anything, it should be relevant, and then it should be time bound. You have to fix a time bound things, you have to fix a timeline for your research. Okay, so smart idea, always go for the smart questions and with the research feasibilities and everything. Right now, research question examples, I'll okay, these are not related to medicals, but these are really amazing things. Okay, so the weak research questions and strong research questions outcomes. So what are the differences in weak research questions and strong research questions? Always the wide outcome questions or the wide scope questions are weak questions. Either very narrow means like if you are just targeting orphan disease or orphan conditions, orphan drugs, or such things for the proper research, that will not work. If you're just targeting like one very specific thing, but that is not clinically or statistically significant, that will also a weak question. Weak question means either it is too wide to do, or it is too what we say too specific to do. Okay, so when you go for a strong question, you will see that it has every component. It has a good timeline also. It is something which will give the good outcomes also. It will include the good population also, and it is reflecting or relevant to some social needs as well. Okay, so like they have given the examples over here. Will holding workshop reduce the school violence? Okay, so they have not given like what kind of school workshops, what kind of workshops they want to do, like one school, two schools, multiple things, what kind of violence, and they have not defined properly, right? They have not defined properly. Their angle is too wide. It is like holding the workshop will reduce the school violence. What kind of workshop they are looking for, what kind of workshop they're talking about, and what kind of school violence they are talking about. Okay, school violence is not only one type, school violence is multiple types. Okay, workshops are also multiple types: psychiatric workshops, physical workshops, self-defense workshop, multiple workshops are there, right? So what kind of workshop you're looking? This is a very wide what we say the question to do, right? So that's why this is also one of the weak question. Will counseling reduce the mortality in heart attack patients? What kind of counseling you are looking for? Drug counseling, lifestyle counseling, food counseling, what kind of counseling you are looking for, right? That's also a very wide question. And for how much time, right? What is the follow? Well, I mean, lot of things. And the question is, we do not have to write the how much follow-up and all, but we have to give the specific terms. We have to write the specific things, lifestyle counseling, or drug counseling, disease counseling, complications counseling, anything, right? So this is a wide angle question, right? And then if you look at the strong research questions, will 1 hour hands-on car seat training session reduce child motor vehicle injuries in Mumbai? Yes, very specific. 1 hour hands-on training on car seat sessions, how to pull the car seat, your what we say, and then how to reduce the belt, how to pull the belts, and how to keep the belts, and everything will be covered. And it is targeting the very specific population like child motor vehicle injuries, and in a specific area, Mumbai, right? So it is one of the strong questions. Then, will the patient counseling on medication non-adherence? Now look at this. Will the patient counseling on medication adherence? So as I told, multiple types of patient counseling is there. So medication non-adherence, if you're doing a counseling, will improve the outcomes in heart attack patients after angioplasty, right? So that is very important, right? Strong questions. Components of a research proposal. So if you look, there are multiple. So first it starts with the title of the project, then summary of the project you have to submit. Summary means It is like an abstract of the project. One page, sometimes they write 500 words, sometimes 400 words, 200 words. Summary also you have to present with the proposal, entire proposal. So first title of the project, then summary of the project, then accomplishments, whatever you have done in your life. Okay, whatever you have done in your life, you have to write there. Publications you have done in your life means the publications, your achievements, your presentations, your field experience. Field experience means the experience in that field, experience in that area, experience in specific conditions, specific NCDs or CDs, communicable disease, non-communicable disease, cardiology, infectious or psychiatry, wherever you are applying. If you belong to pharmaceutical company, pharmaceutical analysis, or you are from pharmacy practice or anything, pharmacology, you have to be very specific and you have to mention the accomplishments. Okay, and then definitely background about the project, literature review, proper literature review, novelty or innovation, how it is innovative, how it is novel and different from others, what you are doing for first time in this project, right? And then aims and objectives. Methodology means where are you doing, what you are doing, what's the population, what's the sample size, what is the randomization technique, what is the sampling technique, multiple things. Ethical approvals, then what are the expected outcomes, limitations of the study, what would be your limitations of the study, and what you are looking after this project. What are the basic timelines when you are completing the data collection, when you are completing the data analysis, when you are submitting the reports, right? And what institutional support you are getting, like teams, infrastructure, labs, computer, whatever you are getting from the institutional support. And then finally budget, right guys? So let's discuss one by one and finish it off. Importance of the literature review. It will give you the idea of your novelty, right? It will give you the novel idea or novelty idea. As I told you, always look for the research gap. That research gap will take you to the perfect idea, perfect aim and objectives. Then your sources should be always academic literature and gray literature for the ideas. Okay, and then you can, ideas can come from anywhere, but usually the good idea usually we get from the gray literature, because gray literature, they publish it with a lot of new implications. Where are the research gaps? Be critical thinker. So your integrate and synthesize the existing literature. So always the title, if I say the summary of this slide is, you should always look for the implications. You should always look for the future perspectives, whatever is mentioned in the literature, and always construct your hypothesis on the research gap given in the paper, in the book, in the idea, in the summary, in the conceptual papers, wherever it is, wherever you get from. Okay, then methodology. How will you answer the research question? Like who to participate, like who means your population, what means what you are looking for, outcome, when means the duration of the study, timelines. Okay, where means the site of the study, which hospital, which laboratory, where you are pursuing your study, why, what are means again what you are looking for, your aim and objectives, and how means your entire methodology, how you are executing this, like what are your sampling criteria, what are your interventions, what are your counseling parts, whatever is included in that. So these are very simple questions: who, what, when, where, why, how. So who, your population, your inclusion criteria. What means outcomes, when means timelines, where means study site, why means your aims and objectives, and how means your methodology. Okay, so simple to do that. Okay, these are the components to methodology. As I told you, study design, facility, population means who, population means why, and how you are including and excluding the criteria in the population, inclusion and exclusion criteria. Then sample size, then study design and facilities, then very important implementation and ethical considerations, what you are from where you are getting the ethical considerations. And very important part guys, if you are including human in your research, you have to take the ethical considerations. Okay, and then data collections and what kind of statistics you are using, that's important, right? Then while writing the methodology, methods must be very thought out and comprehensive. Research design means, if you look at this research design, whether it is qualitative or quantitative or mixed methods, whether it's a cross-sectional study or longitudinal surveys, whether it's a primary or secondary data analysis, prospective, retrospective, whatever is there, you should be very transparent, clear. And then what kind of tools you are taking, pilot studies are done or not, or if you are going for the questionnaire or something, whether it is validated or not, right? What are your proper inclusion exclusion criteria? What are your race for the participant, individuals? What are the groups? If you have multiple groups, then always mention those groups: group A, group B, control case, anything, based on that. And what are the tools, instruments, equipments you need to pursue your research? So methodology means you should take care of these all things. You should be very precise, very transparent, very clear, and coherent. Everything should be mentioned in that. How descriptive you can go ahead. Okay, in the methodology part, there is no restriction of the words. There is no restriction of the words from NIH, from ICMR, nowhere. There is no restrictions of the words in methodology part. They may have the restrictions in introduction, background, discussion, everywhere, but there is no word restrictions in methodology. As much as possible, go clear, go transparent. Okay, and teams and collaborations are always appreciated. If you have multidisciplinary approach research, then that's benefited for all the researchers and high chances of getting accepted, because doing a research in different domains, including different teams and multiple sites, is very much appreciated. Okay, like suppose I'm doing something in medicine right now in diabetes. If I include our biochemistry department as well for testing of GlyHB or testing of PPBH or testing of some calcium or calcitone in diabetes, then it will be multidisciplinary. I'm taking help of them. I'm using the multidisciplinary approach in doing that, right? And get a good team, always good data collection, data entry operators, and good team to perform the test, good team and a good statisticians in your team, right? If you have a good team, definitely there will be no hurdles, right? And choose the appropriate peoples to perform the task. As I told you, collaborations and associations. Don't be greedy. Okay, don't be greedy means do not keep everything for yourself. If you think research you can do at by yourself, all alone, then you are at greedy side. You are greedy, completely greedy, and you cannot pursue the research completely. Okay, don't overburden the staff. As I told you, in the research, always keep time for yourself. If you are overburdened, don't go for it, or get the more staff to pursue the tasks. Okay, determining the timelines. So here, as I was telling, go for these things means you have to plan everything and you have to write in the proposal. Okay, these things are appreciated in the proposal as well. If you are writing these clearly, like, okay, they don't hesitate to give you funds. If you are asking for a good amount, also they will give you, but you should be clear why you need it, why you want this much associates, how many associates you want, how many data entry operators you want, how many data collection people you want, research assistants you want. They will give you, but if you are very clear and you are justifying those things, they will give you. Okay, so don't think that you are writing only by yourself, you will be doing everything. That also they will think and they will not give. They will think like he is greedy, he don't want to give it to other people, and he don't want to perform by other experts, and we know he cannot do that. So that is these are the negative points, guys. These are the negative points. If you think that I will write less funds and I will only do everything, that is also not correct in the research proposals. Okay, always be appropriate. Okay, determining the timelines, proposed timelines, there, like how much time must be allotted to write the introduction, how much time is there to complete the overall task, and if it is something, then break them down differently. Like, okay, I'll write introduction part, this will write this part, and this expert will write about the institution, and in that part, to decide the timelines, Gantt chart is very important. So here is the Gantt chart. Like this is for the like suppose for a full you have to provide the full Gantt chart for entire research. Like, okay, in first we will write the proposal and give, then we will submit to the ethics committee in the March 2014, like in first month, then second month presentation of the thesis proposal, third month preparation of the application to the ethics committee, then we will start data collection in June 25. I mean, this is just a timeline, so you have to keep clear everything. Data collection, data analysis will be done in this term, and then submission of this will be done in this term, presentation of and data project winding will be done this time. So Gantt chart will give them the entire schedule. Always prepare the correct Gantt chart and submit it with your proposal. Okay, and then budget estimations all inclusive. It must be Research how much all supplies, material, and overall cost, everything should be there. For personal, you must factor in benefit, vacation time as well. As I told you, you should keep everything intact, your personal time, vacation time, your personal benefits as well. You should be very clear in that. Okay, it's not like that you will tell them I will complete everything in 6 months, just give the funds for 6 months. They will not believe and they will not give. Okay, always be reasonable. Okay, always be reasonable. No padding. No padding means like you cannot write even extra cost. If you think like they are giving, you should not keep the extra cost. If it is a work of 3 people, if your proposal says this is a work of only 3 people and you are writing that you need 12 people or 10 people, they will not give. They are also assessors, they are also researchers, they are also top people. Okay, so they will evaluate everything. Some reasonable things are okay, but unreasonable things, unreasonable less amount or unreasonable high amount, padded amount will not be acceptable. Okay, so we can include the student charges, hirings, supplies, travel cost, everything you should include. Okay, and you can include in that. So these are usually the flow charts. So first is like we're starting from here, identification of the needs and focus. Then final prospective grants, which grant you are, find the prospective grants, which grant you are going to. Then develop the general protocol, proposal and budget. And then, in that, like some of the letter of inquiry. If you are not aware of something, then call them and ask, write the letters, write the emails and ask what's the problem. Okay, find out the full with the clear idea. Okay, then prepare the specific proposal and submit it. Okay, after submission, it will go to the agency review. Agency will review it, and then you will get the award letter if it is sanctioned. And then it is up to you if you want to accept or decline and carry out the project. File the submit. File the reports at the end of your research projects to the funding agency. So this is the major flow chart, research flow chart. Research proposal flow chart, right? Now I'll take you general tips, very important. So know the culture of the funder, like what kind of fundings they have given before. Okay, that's very important. Follow all instructions. Follow all the instructions which is given by the funder or the sponsors. Very important. Read the entire proposal. If it is 20 pages, 30 pages, 100 pages. If they took time to write 100 pages, why can't you read the 100 pages? Guys, people, this is the major problem. This is a major problem with investigators, majorly with the new academicians and new researchers. I'm telling you, with the new researchers and new academicians, there is a problem. They think, oh, this is 100 pages. They just read the eligibility and try starting. Okay, start writing the proposal. That not work. If they have taken the time to write 100 pages, you should read all 100 pages properly. Okay, that is very important. Each and every instruction is important in funding criteria. If you are writing a good proposal, writing the great proposal, but if you are not aligning with their one instruction, they will straight away reject your proposal. Got my point? They will straight away reject it. Know the assessment criteria. Usually they give the assessment criteria. ICMR also have the assessment criteria. NH also have the assessment criteria, and based on that only they assess your things. Treat the application like an argument and present it with the confidence. That's important. Argument means you are justifying. Argument means you are not making the argument, you are justifying, you are convincing them. Okay, you are convincing them and presenting that your convincing power with the confidence. Okay, use the appropriate disciplinary concepts and methods. Okay, and then 4 C's. For the proposal writing, it should be clear, coherent, compelling, and competitive. Right, guys? It should be clear, coherent, compelling with your the instructions and all, and competitive. And then last, I think, general tips for grant writing: macro over micro details. Okay, don't get caught up into the little details, very little details. When you are writing a proposal, I'm not talking about the methodology here. Methodology should be clear and detailed. But big picture means your outcomes, as I told. It should not be very, very specific, or it should not be very vast. Okay, your outcomes should give them a good, what we say, the implications after, or good future after the research. Okay, write and cite and cite and cite. That means plagiarism. You should not go for it at all. You should not copy the ideas which were published or something. You can extrapolate, but not plagiarize. Okay, do not plagiarize that at all. Get some parameters, comparisons, like, if it is very, if you really want something, then, give in the review of literature or in that, proposal, in your, aim and objectives, try to compare the things, then, it's easy for, easy to get grants, because you are looking at the wide picture and you are giving them direct outcome, you are giving them direct comparisons. So they usually look at such types, but usually in clinicals we do not get much scenarios as North versus South or Asia versus EU, until unless we are doing multi-centric and multi-million-dollar research. Then definitely know your agency's mission, read the instructions carefully, write with confidence, but don't disregard others' ideas. Have a great scientific ideas. If in doubt, contact the program officer. It's important. You should not hesitate to call them and ask them what's the problem. Okay. So with this, always keep in mind the page limits, word limit, budget limit, abstract format, reference format, PI and co-PI eligibilities, and submission methods, what type of files you have to submit, what should be the size of file, okay, and what's the font size, minimum resolution of the pictures, table of the content, research objectives, tables and figures. Okay guys, so with that, I'll end this. This is, I think, last slide for me. Common errors in a proposal writing. Usually people do not fit with the agency's mission, still they apply. They violate one or more agency guidelines, as I'm just focusing on these only since starting. Beyond the capabilities they go, okay, and lack of proofing, like grammar, spelling, formulas, numbers, math errors. They missing the page, figures, tables, signatures. Sometimes they're poorly organized. Their methodology or their entire proposal is poorly organized, unfocused. They have low personal, or they are just keeping low personal budget, not enough people, or they are low impact, no publishable and all. So in such traps, do not trap yourself. Do not go to such traps, guys. Okay, this is very important to avoid such errors. Okay, so with that, thank you very much, guys. And I just want to show you one more paper which I have opened for. This is the ad hoc project for ICMR, format for a research plan. As you can see here, title of the proposed research, summary, keywords, abbreviations, background 500 words, literature review 1000 words, novelty 250 words, 250 words, study objectives, methodology 2000 words. So including these following subheadings: study design, sample size, project implement plans, ethics review, data collection, statistical plans, limited expected outcomes, limitations of the study, future plans, timelines. This is the ICMR ad hoc proposal guidelines. Okay, guidelines means the proposal sample. So such samples are always available. Do not get scared that you have to write by yourself and you have to like you don't know anything. Everything is well explained in the NIH or ICMR, DST, DBT sites everywhere, guys. Okay, so with that, I will end my session and over to Mahendra sir and the audiences.
Yeah, if anybody is having any question, they can ask Dr. Ajit. Dr. Ajit, first of all, I will congratulate that it is really very wonderful session. And in the 45 minutes, approximately 1 hour, you have just summarized and concise this very vast topic. Because as you correctly said that it can take 5 hours for you to explain the things in the minute details.
Yes.
So really, this is really great information for the participants and very nice initiative. So I would ask any participant if any question they are having, Dr. Ajit would be glad to answer that.
Sorry, sir. Yes. Guys, anyone can ask the questions if you have any kind of questions about grant proposals. And don't worry, I'll be, taking the 5-hour sessions as well soon.
Yeah, but that really want the patience and telling the things in detail. There's a great effort.
Okay, so
see, Dr. Ajit, I would like to ask one question from the
yes, sir,
from the viewpoint of the budding researchers who are there to file the grants for the funding agencies, particularly to the government agencies.
Yes.
So,
what are the things basically that you have narrated all the aspects and regulatory eligibility criteria? Of course, these are the criteria. But apart from that, what are the things that they are looking into the young researcher? Novelty of idea is a very big term. If we can make some specific or some narrow down.
Yes, definitely. So for the budding researchers, they look for their experience. Now you will tell me that they are budding researchers, but how they will come up with the experience? They will look for, when you are writing a proposal, they ask for your master's thesis. They ask for the master thesis title, or if you have done any publications, there is no such masters where you cannot publish anything. Right. If you are a PharmD student, always try to publish your project. Always try to focus on your project, how you are doing. If you are just taking it lightly, just writing the thesis book for the namesake, for the submission sake, it will not help. If you really want to do something, do it properly and try to publish it. Whether it's a small impact factor journal, whether it's just Scopus indexed or any index journal, publish it. Okay. And don't leave it until it's not published. When you are in internship, you have a great chance 1 year to do multiple research. Wherever you are going to medicine, you are going to surgery, you are going to dermatology, do some 2 to 3 months project. Write them in your project. If you are not able to publish it, also write in your project. You have that experience of doing research. They do not look that you have explicit research experience of 5 years after the degree or not. For the budding researchers, they just look for your enthusiasm in the research, how much you have done in your studies. That is more important what they look for in your MPharm, in your PharmDs, in your, if you are in the PhDs, what you have done before and what you are looking for. Sometimes we are not able to publish in our master. Sometimes we are not able to publish after MPharm. But at least we have done something. If we explain that only nicely, it is very, very important for them to understand, and they will they get that vibe from that proposal only, like how you have written that, the methodology. If you have done something research, if you have written the proposal, proposal means the protocol only, right? Proposal means the protocol. When you are in your masters, in your PharmD, you must have written the proposal, the protocol for the ethics committee. So the way you are writing that proposal, see, I can understand that guides definitely help them and do that. So there are 3 things for the people, what I suggest for the budding people. One is, like, if you have the experienced mentor with you. Okay, it is very important to have the experienced mentor with you who have done the relevant work to the field. That's important. If you also have not not having any kind of experience in that field, your guide must have, and where you are going to pursue that project or the research, that affiliated institute or the medical school or the hospital must have those facilities, right? So these are the major thing what usually they look into the budding researchers. And budding researchers always, I'm telling you, sir, should not go all alone. If they are writing all alone with their for the first time, even some of the professors also, they write for the first time, and associate professors also, they write for the first time. They should not go for the multi-million projects. They should go with the small projects. A small project is not like 50,000 or 1 lakh. Small projects are 20 lakhs, 50 lakhs. These are the small projects. So first, start your journey from somewhere. And if you're not getting grants also, just continue the research. And if you are continuing the research, if you are showing that, because that will have some outcomes, your projects, your results, your some publications, your conference presentations. So it is applicable for the new buddings. If you are not getting published, you have presented somewhere in the conferences. That also they ask. There is a particular section in the field, like how many conferences you have presented as a first author, or these things. So that shows your enthusiasm of doing research. That's very important for the budding researchers.
Yeah, this is really very informative. And any participant, if they are having any question, so I can see that many 50-60 participants are there. So
yes, sir.
There are a lot of questions that can be asked, and then we
definitely.
are having expert here. That we are having the man who has written many proposals and got so many grants from the ICMR, and this is the only thing I know, that you must be having different sources also.
For
So
all type of grants, from Novartis,
yes.
2.5 CR for MHFR,
Yes,
and
yes,
from
yes.
ICMR, from DST,
So
travel grants,
you are the
from
perfect
European
blend of the
societies, American College of Cardiology.
you are perfect blend from the industry and as well as from the government agencies. So these are the things.
If guys you have any questions, please ask.
Yeah, I think this is या तो ये clear हो गया या all clear हो गया.
I think that's fine then, sir. Thank you. Thank you, everyone.
Yes, sir.
So
Yes, sir. Thank you so much, Dr. Ajit Singh, sir, for this awesome presentation. It's really outstanding. We had a lot of update in our knowledge with your session, mostly about grant proposal writing. Thanks for helping us become more aware of problems and the ways we can solve them. Really,
Dr.
it was
Sakina,
outstanding.
Dr. Sakina, I'll take over from here from you. Okay. Thank you very much.
Thank you, Mahendra.
Thank you for our host, Dr. Sakina. And now there are last 20 minutes. I will take over from here from her. So guys, you're welcome. And thank you for attending this. And we have Dr. Mahendra Singh Rathore with us. Sir is the principal at GIP, Geetanjali University, Udaipur. And sir is not only the academician, but he is the NAC instructor, and he's the public speaker and neuro-linguistic educator and trainer. So I would like to invite sir now to be our chief guest, as I have invited him initially also. So to be the chief guest for our valedictory function, and sir will be announcing the results as well. So first I would like to invite sir to say a few words, and then I'll go for the vote of thanks and the awards.
Yeah, Dr. Ajit, am I audible? Yeah.
Yes, sir, you are audible, perfectly audible.
So this is really a great conference that you have organized, and rather as usual, it is a perfect one. And then this is a perfect blend. I was going through the resource persons who were there for the past 2 days, and then these really were very renowned people. And I'm damn sure that the participants would be having the great benefit from this conference, and the insights about the research, insights about the different pharmacovigilance aspects, insights about the artificial intelligence aspects and the medical research, medical writing, pharmacovigilance. So we all are really in touch with that. So thank you very much for inviting me and as a chief guest. And I'm not having much to say about this conference. And rather, I would say one thing from my experience of past 18 and 19 years. That one thing in your presentation, that last presentation, I found that you have mentioned about the goals. That is the SMART goals. So this SMART goal is applicable not only to the research grants, but it is applicable to, in fact, every area of the life. Let it be your personal life, let it be your career or business life, let it be your health, let it be your inner life, and or let it be your relationship also. So these are the things that we can apply everywhere. So one thing, a small exercise that I used to suggest to my students, that whenever you wake up in the morning, you should ask one question, that why I have waken up today. And immediately after that, the moment this question comes in your mind, in answer of that question, one should say that I have to write down 3 goals at least on the paper, and will decide to and commit to myself, make a commitment that before sleeping, these 3 things at least are to be accomplished.
Yes.
See, because today's world is designed for the distraction. And every moment we are distracted by the shiny objects, we are distracted by the social media. So it's remaining focused on a particular task is a really challenging one. And if someday, if it would permitting, I will tell you that our mind is also designed to be distracted, and why it is so. So that we will discuss the thing, and some small I don't know how much time I have having. So
Sir, you please go ahead. Please go ahead. We have 10 minutes. No problem.
Okay, so that I will just put some light on that. So let's discuss this thing. I told you that how our mind is designed to be distracted.
Yes.
So I would like to explain this a little bit more. That in the previous era, since evolution, our human brain was evolving. And at that point of time, the human body was there and human brain was there. There was no social structure. There was nothing like as the modern era. So only the thing and the brain was having the two functions. That is, one to keep the body himself protected, and the second one is to make that comfortable. These are the two primary functions, and these are still the primary function of the brains. And the brain do not want to do a lot of exercise. So wherever they will find the comfort, they will go there. Yes. So our brain will find the comfort. So early, before that, we are having 5 senses. And in that earlier era, there were the threats in the jungles, like lions or snakes or this different animals. They can harm the person. And our mind is designed that our senses, little bit, thodi si bhi awaaz hui, our senses would be attention to that. And then you can see this and correlate this example with the today's mobile phone message blink. The little bit blink is there, that our attention will be going whatsoever the focus work we are doing. So the little bit blink is sufficient enough to have the distraction of our mind. So our mind is designed like this. And then keeping that into the focused area requires a lot of efforts. So this is the thing that's why I told you that the smart goals are to be kept, and these are very much useful in all areas of it. See that we are having 5 areas in our life. That is, one is our career and business. The second one is money and finance. I kept both the things differently because these are the 2 different aspects. And then the third one is the health, and then the fourth one is the relationship, and then fifth one is the inner life. That is that whatever we are talking to ourselves continuously. And these are the things, so that remaining focused is an art, and that can be learned. And there are a lot many techniques are there that how to get the focus mastery, and how to, despite of all the distraction in the environment, how we can remain focused on our task. So one of the factors that you have mentioned correctly here, that smart goals. And then see, one thing is that the goals are being set by our conscious mind. Conscious mind means that we are talking, we are observing from our senses, and the goal achieving is done through the subconscious mind. There is a secret behind that. So until unless this thing will not reach to the subconscious mind, nothing would happen. Whatsoever the goal we are deciding. See, we are keeping target to go to the gym daily. So tomorrow I will wake up. Yes, I will go to the wake up on 5 o'clock. I will do this thing, exercise regularly. So these are really but how many of you we are able to make commitment fulfilled? 2 days, 3 days, 4 days. So there are deep concepts behind that. Why our brain is doing this, and how this can be overcome. So this is just the glimpses that I have told you, that you discuss about that, how the neuro-linguistic programming can help out one person to achieve the goals in all the 5 areas that I mentioned here. So there are very much important techniques and the things.
We'll definitely
really
specific session on that, sir.
Yes, it would require, though, as you told that on the research you can speak 5 hours, I can speak 10 hours for this topic.
Yes, absolutely.
Yeah. So, but this is not the right platform, so that just I conclude my talk here. Yeah.
Okay. So thank you very much, sir, and thank you for really amazing motivating things, motivating words, and how we can organize ourselves. Definitely, we should have the targets and goals in the life, whether they are short-term goals, daily goals, weekly goals, monthly goals, yearly goals, or long-term goals. We should always have the goals in our life. That's true. Someone wants to speak? Okay. So now I think this is the time to announce the winners, and then we will go to the vote of thanks. Right, guys? So excited? All Okay, let me share the sheet, what I got from my team. Okay. So this is the sheet for MCPCON abstract presentation winners. So first we have poster case reports, like case reports, poster presentations, and the first award goes to Ane from Government Medical College Baroda. He presented a large and rare schwannoma of the musculocutaneous nerve, the case report, in the poster format, and the first award goes to him. The second award in this category goes to Dr. Karisma Rathi from Dr. D.Y. Patel Institute of Pharmaceutical Sciences and Research, Pimpri. She talked about Carolese disease associated with autosomal dominant polycystic kidney disease, ADPKD with acute pancreatitis. She also presented the poster and case report. Now I'll come to the original category, oral presentations. The first award goes to Dr. Anusa N. She is from Jipmer, Pondicherry. She presented the oral presentation, the paper, original paper on subclinical hypothyroidism and plasma levels of commonly prescribed antiplatelet drugs. And the second award in this category goes to Rohit R. Nair, and he presented on assessment of mental health status among transgender and gender diverse population. He's from Acharya and BM Reddy College of Pharmacy, Bangalore. And then I'll come to the review papers or innovation or conceptual papers on innovation. In this, the first award goes to Priyanka Guha. She's from Naipur, Hajipur, and she presented an insight on recent developments in tuberculosis diagnostics in the era of the artificial intelligence. And the second award goes to Deepti Chandran. She's from Sri Chitra Trinul Institute of Medical Science and Technology, Trivandrum. She presented neuronal derived exosomes from blood for identification of biomarkers in Parkinson's disease. So these two were very good papers. Again, everyone has presented nicely, but these two innovations and conceptual papers were really amazing. And then in oral case presentation, only one was selected, others were really below the marks, frankly. So in oral case presentation, all the based on all the marks given by all the scores given by our abstract evaluators, they have selected only one. In this category, Dr. R. Gauri from GRT Institute of Pharmaceutical Education and Research, Trutani, and she presented assessment of text neck syndrome among college students. And then final original poster presentations. First award goes to Ruqayya Fatima Begum, and she is from SRM College of Pharmacy, SRM Institute of Science and Technology, Tamil Nadu. She presented on pharmacological investigations of vitamin E with combined oral contraceptives on INHBA gene against PCOS that intricate through melatonin PKC pathway. And the second award goes to Mohammad Kass from Engineering University, Lucknow. He presented to evaluate the ulcer protective effect of Mangifera indica kernel extract in indomethacin induced gastric ulcer in experimental animals. Poster oral original papers. So I appreciate your efforts, guys, and I appreciate the efforts of our abstract evaluators also, and I'm very much thankful that we had around 180 abstracts we have evaluated. And with that, I congratulate every winner here. I congratulate every winner for the poster presentations, oral presentations, conceptual papers. Your presence was amazing, and you have contributed to the conference in a different way. You have presented your science, and it is an amazing way to showcase the science in front of everyone, right, guys? So and again, I would like to announce that the first presenters, the first awardees who got the first rank will get we have decided, as we have decided, 5,000 rupees cashback and 3,000 rupees for the course. So there are two courses. They can choose any. One is on medical coding, and one is the 6-month PG diploma course, PG certificate course in clinical research and project management, and 2,000 rupees they will get the cash. Okay, we will GPay you, and with your certificate, rank certificates, and the second ranks, they can get 3,000 rupees worth course. These course, any of these course you can take, because these courses are just recently launched, and these all are value-added courses for 3 months. One is for 3 months and one is for 6 months. Okay, so you can take up any course of your choice, either medical coding or our postgraduate certificate course in clinical research and project management. Okay, guys, so they will get 3,000 rupees worth in a course. Or if you don't want to take either of these, we are coming up with multiple things. You can take anything worth of 3,000 rupees from our CLIMAT anytime within 1 year. Okay, so it is not like we are just forcing you to take out of these two. You can take up any course of 3,000 worth, or rest you can pay if it is higher than that, or you can take it up to 1 year, any course from us, right, guys? So these are the awards, and these were the presenters who got from us. So I would like to thank everyone of you, and then I would like to start our major part. So I would like to start thanking with our valuable partners, IFMTM, International Federation for Medication Therapy Management, and Indica AI. Mr. Hardik Dave, my friend, is very cooperative, and he always support by us, and Indica AI was always supporting in conducting this MCPCON virtual. And the theme of this conference was also AI in healthcare. Embracing AI in healthcare. And I would like to thank our guest of honor for this conference, Dr. Ranjit Barsikar sir, and chief guest for inauguration, Dr. Pramod Kumar Rajput sir, and chief guest for valedictory function, Professor Mahendra Singh Rathore sir. Thank you very much, sir, for joining us. And Mr. Hardik Dave again, and special session discussions was done by Dr. Hit Sharma and Dr. Shivani Desai. And it was paneled by Dr. Jamal Beg and Dr. Anupama Koneru. So Dr. Jamal Beg is Director of Pharmacovigilance at Sanofi. Dr. Anupama Koneru is Principal at Sultan College of Pharmacy. Then we proceeded with Mr. Apoorv Soni, Project Manager at JSS Medical Research. And that session was presided by Dr. Kiranmai Mandwa and Dr. Hamantachar Joshi, Principal and Professor, St. Paul's College of Pharmacy, and Principal and Professor at Sarda Vilas College of Pharmacy, respectively. And then we had a wonderful debate session yesterday on AI in medical writing. That session was moderated by our own Ms. Sarabjit Sidhuji. She is the manager at Clinical Trial Transparency, GSK Pharma, and we had debaters, the best debaters, Dr. Kanav Khera. He is a professor of pharmacy practice at Lovely Professional University, and Dr. Sameer Dhingra, registrar and associate professor and head of pharmacy practice at Naipur Hajipur. Then we proceeded with the next topic on ICH role in globalization with Dr. Kanav Khera, and the session was presided by Dr. Isaac M. Deer, Professor of Pharmacology, Department of Pharmaceutical Sciences, and Director for Center of Career Planning and Counseling, University of Kashmir, and by Dr. Jamal Beg again. And then we had Mr. Ashish Kumar Verma, pharmacovigilance expert, and our CLIMAT Research Solutions at CLIMAT Research Solutions. And that session was presided by Dr. P. Rajsekhar, Principal, St. Peter's Institute of Pharmaceutical Sciences, Hanamkonda, Telangana, and Dr. Mansa Deepa, Professor and Head HOD at East West College of Pharmacy, Bengaluru. And then the next session, the final session for yesterday, was taken by Mr. Ravi Jogai, Associate Director at Syneos Health, India. And the session was presided by Dr. Ankumar Mishra. And today we started with a wonderful session by Dr. Ashok Sonoy sir. He's the professor of pharmacology at Kasturba Medical College, Mangalore, Mahe. And he took the New Drug and Clinical Trial Rules 2019. And that session was presided by Dr. Rakesh Somani, principal and professor at Dr. D.Y. Patil University, College of Pharmacy, Mumbai. And then we had another session by Dr. Akshima Sahu, who is also our pharmacovigilance expert at CLIMAT Research Solutions. And that session was presided by Dr. S.N. Sriharsa, Principal and Professor of Hillside College of Pharmacy, and Dr. Giriraj Kulkarni sir, Professor and Principal at Goparaju Rangaraju College of Pharmacy. And then the session was on clinical trial transparency by Sarab Sidhuji. And then the session was presided by Dr. M.S. Lauddin sir, Principal, Alamin College of Pharmacy, Bengaluru, and then Dr. P.K. Lakshmi Madam, HOD Pharmacy Practice, Jeepula Reddy College of Pharmacy. And then you have seen that we talked about result biased in clinical research with Dr. Kanav Khera again. And then after lunch today, we had a wonderful session by Dr. Krishna Undela on signal detection, and the session was presided by Dr. V. Murugan, Professor and Academic Director, Narmad College of Pharmacy, Bengaluru. And I'm very glad that I'm going to that college tomorrow. I'll be presenting and interacting with their students tomorrow physically in Bangalore. So Nirgun College of Pharmacy, and then Dr. Karthikeyan sir is the professor and HOD of Pharmacy Practice at School of Pharmaceutical Sciences, Vistas, Chennai. And then the next session was by Mr. Hardik Dave, my friend, founder and CEO of Indica AI Mumbai, and he took the industrial session on AI in healthcare. And then last session, you must have seen, you are seeing that I took the session and presided by Professor Mahendra Singh Rathore, sir, who is our chief guest for valedictory as well. So thank you very much, sir, and thank you everyone. And this session and this conference was not at all, successful by the without the participants. We had around 300 plus registrations. Though due to I think Sunday and vacations, there are a little less population who were joining, a little less participants who were joining, but there were around 300 plus registrations and 180 plus abstracts. And now I conclude, and I thank my teams as well. Dr. Siddesh, Dr. Sakina, Manish, and everyone who are working, day and night to make this conference successful, guys. Thank you. So thanks again. With that, good evening and take care, guys. It's 5 o'clock. Thank you. Thank you, sir. Thank you, everyone. Thank you. Certificates, yes, guys. Certificates will be given to you by Tuesday morning. Tuesday morning, not before that. Nobody is going to ask me about certificate before Tuesday morning. On a lighter note, but certificates are in the preparations. Maybe by tomorrow afternoon, you all will get your certificates. Otherwise, by Tuesday morning, to definitely sure. Okay. Take care, guys. Take care. Good day. Good day. Bye-bye.